Right.
Yes.
So please open your PPT and then share your slide.
Right, madam. Right.
Next speaker, I think Chippy is also there. Please be ready.
Okay, ma'am.
Dr. Sampath?
Yeah.
Dr. Sampath, are you there?
Yes, I'm there.
Dr. Somnath? Dr. Somnath? Okay. Sir, over to the chairman and co-chair, sir. Manjunath ji, please put a slideshow. Dr. Manjunath, please click on the slideshow.
Now it is visible, madam.
Yes. Click on the slideshow option.
It is visible now. Is it visible?
Oh, no.
Madam, it is visible. Madam,
Oh, hello, sir.
it is visible.
I am ready.
It was visible, now it is not visible.
Oh,
Miss Chippy, please.
okay. Yes, I am here, sir.
Madam, it is visible now.
Now, Manjunath ji.
Oh, okay.
Yes.
Is it visible?
Okay, sir.
Yes. Manjunath, Dr. Manjunath, please click on the share button. Open your PPT first on your desktop or laptop.
Yeah.
Then next to the video, stop.
Visible.
Visible now?
Yes.
Okay, now I am going for the presentation mode. Now it is visible. Visible now?
Dr. Manjunath, you have 10 minutes time and 8 minutes for presentation and 2 minutes for discussion.
Fine, sir. Fine, sir.
Thank you, sir.
Shall I start?
Please start.
Please, yes, please, sir. Please start.
So, good morning, all. I'm presenting my talk. The title of the presentation is Metabolome, that is particularly the phenol propanoids as a reciprocal defenses in the pomegranate and XAP interactions. And as the component of the metabolome in case of pomegranate, we worked on particularly the phenol propanoids, how that can be induced and that how they are modify the resistance level in susceptible and the tolerant varieties in the interactions of the most invading pathogen in the pomegranate. Before that, I like to introduce the pomegranate. As we all know, pomegranate is the most important cash crop in the country, especially in the Karnataka and the Maharashtra. And it has been touted as a super fruit, and it is a cash crop, and it is a rich source of the nutraceutical. And recently, it is also being used as a cosmetic source, and it is one of the most in the ancient the folk medicine, and the most of the processed food are prepared from the pomegranate. And the major constraint for the pomegranate production is the two important diseases. One important is bacterial blight, which is Xanthomannus axonopodis or punicae, and which causes the losses up to from 10 to 100% depending on the weather factors. And it is becoming the devastating the disease, particularly in the Maharashtra and Karnataka. So this is just to show the pure culture of Xanthomanus, how it looks on the culture, and why metabolome study is required in pomegranate means we have very limited options of synthetic molecules in case of pomegranate disease management. Where, currently the antibiotic streptomycin sulfate and oxytetracycline combination, and the bronopol is another the component is the only source for the chemical the nature, and that's why to be integrated in the management options we need the new generation of molecules where the metabolome really can be an answer, and where an engineering of the metabolic process is the key activity under the rapid changing pathogen diversity, where in the normal process, the pathogen is getting tolerance over the already used chemicals like streptomycin sulfate and oxytetracycline, where earlier we are using 200 ppm, now we are 500 ppm is also not, I mean, it is becoming tolerant up to 600 ppm. That's why under the rapid changing pathogen diversity, where the new generation of molecules are required, which is, that's how the metabolome studies are required. And as we all know, the metabolites are very smaller molecule with a diverse biochemical origin, can be programmed to elicitation activities. These metabolites can be elicited by array of compounds under the pathogen inoculated condition. That's how the metabolites can contribute for the new generation of molecules. And during the host pathogen recognition and reception, these metabolites serves as the direct signature which defines the dynamic equilibrium of pathogen and correlated phenotype as resistant and susceptible. And this is the workflow how we studied the phenylpropanide pathway in the XAP and pomegranate interactions. So to elucidate the biochemical process involved in pomegranate responses to XAP infection, a liquid chromatography mass spectrophotometry based untargeted metabolome study was designed using the metabolic composition of aqueous methanol extracts analyzed through high performance chromatography system coupled to high definition mass spectrophotometry. And the retention time up to the 31 minutes we studied, and this is the workflow of the whole process under the study, and this is what the sample collections of the resistant susceptible to proceed for the metabolic analysis. And this is what the number of metabolites eluted at different retention time. And this is the principle in the normally for metabolite analysis, the principal component analysis we adopted here and visualize the different the metabolites. And the left panel indicates the very significant the metabolites we studied in pomegranate, and the right side panel is the array of metabolites and the untargeted the metabolome of the phenylpropanoids out of our attempt. And this is the table to show how the resistant related induced phenylpropanide identified in the pomegranate leaves upon the XAP infections, where the many precursor molecule of the PPP like the cinnamic acid at the 72-hour post inoculations 29.6 and the 16 host after the pathogen the infection 76.9, and another the precursor molecule in this pathway is 4-coumarate, which represents at the 12 hours it was 10.8, and which is the other the metabolites in case of upregulated in case of the phenylpropanide pathway is the ferulic acid. The where ferulic acid upregulations was seen up to 15-fold changes at the 216-hour post inoculation time, and similarly the syringine where 32.2 at 48 hours and 72 hours again 29. It is again upregulations were seen at 46 the folds with the 216 hour post inoculation, and also the similarly the other the two the metabolites were also upregulated, the p-coumarate acid and camphor fold. And this is to show that how the resistant related induced metabolites belongs to phenylpropanoids against bacterial blight at the different the time points like 12. This is the different time points 12, 24, 48, 72 and 216 hours. This is what indicates here. And this is the metabolic pathway and correlation based network analysis that revealed that the multi-component defense responses is characterized by the functional metabolic web. So wherein this was used, the network was analyzed by using a cytoscape, and their FC was visualized by node color. So where the cinnamic acid at the 48 hours and picomeric acid from all the time points were very visible. It is a qualitative picture over the previous one, and this is to show the network. And in case of the upregulated one, the syringine and the eugenol and camphorol and the picomeric acid upregulations were very visible from the 24 to 48 hours. And this is the point where the pathogen and the host, the interactions were very decisive at the 24 to 48 hours. At the time, the eugenol, syringine, and the other metabolites upregulations were very visible. That indicates that these metabolites are correlated to the phenotype of the resistance in pomegranate against ZAP. This is the photograph to show that how these the metabolites like cinnamic acid, coumarin, eugenol, ferulic acid, syringine were cross-checked in the in vitro conditions, and this is very clear again. So eugenol were shown the maximum the antibacterial property which the metabolite recovered from the LCMS studies, and followed by another the ferulic acid. And the eugenol, the antibacterial properties were much more higher than the streptocycline what we are currently using. So that indicates the eugenol will be the potential molecule to treat the pomegranate bacterial blight under the field conditions. And again, we check that the eugenol, and we also use the clove oil as the component of the plant, the extract, where the clove oil has containing the eugenol to the 80%. That's how we compared with the clove oil. And the eugenol and clove oil are the more potent to reduce the disease with the maximum protections of the 70.69 in the eugenol treatment, and which is also similar to the streptocycline. That's how this eugenol and clove oil can be replace the currently using streptocycline molecule against the bacterial blight. This is the in vitro antagonistic activity of the eugenol under the laboratory conditions. At 0.1% and 0.2% were shown the very clear the inhibition mark. That's how up to 0.1 and 0.2 can be used. Beyond 0.3 and 0.4%, when we used for the plant the studies, they are showing the phytotoxic symptoms. That's why we choose to study 0.1 and 0.2% as against the pathogen under the in vivo studies. This is a relative quantification analysis we done for the ZAP DNA in pomegranate leaves, where the less the fold change of the pathogen was reported in T2, that is the eugenol treatment, followed by T1, that is a streptocycline. So that indicates that the abundance of the pathogen, the growth was largely inhibited by the eugenol after the treatment, and in the control, the fold change is more than the 200, whereas in case of the eugenol, it is less than 10 to 12. Whereas the streptocycline also recorded the higher the fold change. So these are
Dr. Manjunath, you have another 2 minutes. Manjunath?
Yes, sir. Yes, sir. So this was again implemented with the marker defense genes like the PR protein and the other the defense protein. All these were upregulated, complemented to the upregulations of those metabolites in the plant system, where the PR4, the upregulations are 122, the PR1 at the 120, and the PR10 is at the different time points up to 120. And the most important, the gateway enzyme, the phenylpropanide pathway, was also noticed at the different time point, and the PAD and the chitinase. So this indicates that at the point of those metabolites, the defense proteins are also upregulated, indicate that these were complemented together. And in the summary, I like to conclude that there are array of molecules we have identified as the defense activity by our the metabolome analysis. And the phenylpropanides are the potential to elicit the defense responses, the in vitro and the in vivo. And the priming of those identified molecules elicited with the respective preceding chain of the molecules, which can accomplish the resistance in pomegranate against the Xanthomonas axanopodes tunicae. And this can be explored for the chemical synthesis and characterization, which are now currently undertaken. And those specific metabolites, rich plant derivatives like the eugenol, can be used as the eco-friendly options in IDM approach, where the clovile and combinations of tetracycline, the combinations of the copper are much more potent than a tetracycline to reduce and suppress the bacterial blight under the field condition. That's why this gives the better cue to replace the tetracycline in pomegranate. And furthermore, very important, these studies contributed to the ongoing efforts towards the comprehensive understanding of the regulation and reprogramming of the plant metabolism under the biotic stress. Now, where the genome approaches are being undertaken in NRCP, we are complementing this data to them to have the better understanding of the resistance, where the resistance is the very meager against the xanthomonas in the country. So this is I like to conclude my presentation.
Thank you, Dr. Manjunath, for your nice presentation. After all the 4 presentations are over, now I request Ms. Sippy to share her slides and start her presentation on antioxidant enzymes plays crucial role in piriformis spora indica mediated tolerance to Bendy virus. Manjunath, you start sharing your screen. Sippy.
Whether the PPT is visible? Hello, sir.
No, your slides.
No,
It's visible?
Chippy, no.
Okay.
You first open your PPT on your laptop. Have you opened? You minimize this. Yes. Open. Now click the share button
Yeah.
in the WebEx. Next to the stop or on video, there is a share button. Can you see the share button?
Yeah, ma'am.
Click on the share button.
Yeah, ma'am. It is visible?
Now,
Yes, it is fine. You go for the slides.
now, yes.
Yes. Hello, ma'am. Hello, ma'am. Whether it is visible?
Yes, you can
Very
start your
clear.
presentation now.
It is visible.
Okay, ma'am. Ma'am, can I start?
Yes, you can start.
Yes, you can start.
Good morning to all. Respected chairman, faculties, and other members of the conference. Let me welcome all to my presentation. Antioxidant enzymes play a crucial role in piriformospora indica mediated tolerance to Bindi yellowing mosaic virus. Myself, Chippy, from Department of Plant Pathology, College of Agriculture, Velayani, Thiruvananthapuram, under Kerala Agriculture University. Bindi or lady's finger or okra, it is one of the most important vegetable crop grown in tropical, subtropical and warmer temperate regions of the world, and it plays a vital role in human diet according to its higher nutritional quality, particularly high content of carbohydrate, protein, vitamin C, etc. Yellow disease is the most devastating viral disease that affects among the various diseases, which cause major constraint in the production of okra and causes heavy losses to the farmers. Bindi yellowing mosaic virus is transmitted whitefly by Mysia tabasi in a non-propagative manner. And the major symptoms include vein clearing, vein thinning, complete yellowing of leaves, and the symptoms on fruit include complete malformation and discoloration of the fruits. The current management of Bindia lovi mosaic virus include use of resistant varieties and control of vectors through chemical or cultural method. Use of beneficial endophytes, that is fungal endophytes, is a novel, durable and sustainable method for the management of viral diseases. Piriformospora indica is a beneficial root endophytic fungus that is non-host specific, which was isolated from Thar Desert in 1998 by Ayama, and it confers tolerance to biotic and abiotic stress. Moreover, it enhances growth and also several other growth promoting activities in croplands. This I may introduce my topic, that is to evaluate the fungal root endophyte Piriformospora in the management of Bindia lovi mosaic virus and elucidate the role of antioxidants in this tripartite interaction. Coming to the first part of the study. As a part of the study, various fields were visited and the various symptoms observed in the plants were recorded and studied. The major symptoms in all field locations were vein clearing, complete yellowing of leaves, and stunting of plants. In severe cases, there was bleached appearance of fruits and deformation of fruits. However, vein clearing on flower buds was also observed. This was confirmed by molecular detection using specific primers to the code protein of Bindia lovi, that was AVAC and dung primer. PCR was carried out using a specific condition. For this, total DNA was isolated and PCR was conducted, and the result revealed that amplicon size of 575 and 520 base pair was yielded with AVAC and dung primer in all the isolated, which is depicted in the lane 1 to 4. Moreover, the presence of DNA was confirmed in the pollen grain of infected samples, which is depicted in the lane 5. Coming to the second part of the study, that is co-cultivation of okra with beneficial fungal root endophyte Piriformospora indica. Co-cultivation of Piriformospora indica with okra was carried out both in vivo and in vitro conditions. Under in vitro condition, the carried out in modified plant nutrient medium, and the microscopic examination revealed the presence of indica chlamydosmore inside the root 20 days after co-cultivation. There was also presence of chlamydospores under in vivo co-cultivation of P. indica with okra. Was carried out in potting mixture. For this, P. indica mycelium was cultured, or P. indica was cultured in broth for 15 days, and the mycelium 1% weight by weight was mixed with potting mixture and kept aside for 1 week for fungal growth. This medium was filled in pot trays, and the surface sterilized seeds were placed on this medium, and it was maintained under temperature and humidity controlled conditions for 1 week. And the growth attributes were assessed at various intervals of co-cultivation. There was a significant increase in the shoot and root biomass of okra seedlings under in vitro conditions, and the prominent increase was noticed 20 days after co-cultivation. Moreover, there was also increase in the root length and number of secondary roots in the P. indica colonized plants compared to the non-colonized plants, and the result was more prominent 20 days after co-cultivation. And there was it should be noted that there was a 49% increase in the formation of secondary roots in case of P. indica colonized plants. We move on to the last part of the study, that is evaluation of P. indica primed okra plants against natural incidence of Bindia lovi mosaic virus. For that, P. indica primed okra plants or seedlings were raised, as we have already discussed earlier, and the plants were transferred to pots at 1 week after 1 week of sowing. A pot culture experiment was carried out in Department of Plant Pathology, College of Agriculture, Vellani, for evaluation of the P. indica primed okra plants against the natural incidence of Bindia lovi mosaic virus. And around the plot, infected organisms were placed, which serve as a natural inoculum for the spread of the disease. And the disease incidence which was calculated in case of number of plants divided by total number of plants, and disease severity which was represented as vulnerability index was also calculated 40 days and 55 days after sowing. From the data that there was a significant reduction in the disease incidence and vulnerability index of P. indica colonized plants under natural incidence of Bindi yellowing mosaic virus compared to the non-primed plants. There was 57% reduction in the disease incidence and 56% reduction in the vulnerability index in case of P. indica primed plants after 40 days of sowing. It is clearly evident from the picture there was a reduction in the leaf symptom in P. indica primed plants which was challenged with the disease. It is also clear from the picture that plants, the P. indica colonized or P. indica primed plants showed less symptom, and the root architecture or the root biomass was higher for the P. indica colonized plants under the natural incidence of Bindi yellowing mosaic virus. Effect of P. indica priming on various growth parameters was also assessed, and there was a significant increase in the plant height and leaf area in P. indica colonized plants which was challenged with Bindi yellowing mosaic virus. Moreover, there was also increase in shoot biomass and root biomass in case of P. indica primed plants, which is clearly evident from the graph. P. indica colonization significantly enhanced the dry weight of root and shoot biomass, which is clearly evident from the graph. Total DNA was isolated from the P. indica primed and non-primed plants and subjected to PCR using dent primer. And from the result, the P. indica virus titer or virus concentration was less in P. indica primed plants compared to the non-primed plants. So, what are the biochemical mechanisms that are activated in this mediated resistance to Bindi yellowing mosaic virus? For this, biochemical analysis was carried out. Total chlorophyll content, total soluble protein, and antioxidant enzyme activities like catalase, peroxidase, polyphenol oxidase, phosphate activities were assayed. Coming to the assessment of chlorophyll content. Infection of okra with Bindi yellowing mosaic virus significantly reduced the content of chlorophyll, that is chlorophyll A, chlorophyll B, and total chlorophyll in case of virus infected plants. But P. indica priming could significantly increase the content of chlorophyll A, chlorophyll B, and total chlorophyll content. Total soluble protein, peroxidase, and polyphenol oxidase activity was also assayed. There was a significant increase in the total soluble protein P. indica primed plants under natural incidence of Bindi yellowing mosaic virus compared to the diseased plants. Peroxidase, which is a major enzyme that is involved in the detoxification of H2O2, that is a reactive oxidation species that lead to oxidative burst of the activity of this enzyme was significantly increased in the case of P. indica primed plants. And activity of polyphenol oxidase, that is another enzyme that is involved in the defense mechanism, was also in case of P. indica primed plants under natural incidence of Bindi yellowing mosaic virus. It is clearly evident from the graph that the peroxidase activity was significantly high in P. indica primed plants under
2 minutes
natural
time.
yellowing mosaic virus. And similarly, in the case of polyphenol oxidase activity also, there was a significant increase in the PPO activity under natural incidence of Bindi yellowing mosaic virus, which was primed by P. indica. And the activities of catalase and phosphatase enzyme was also assayed. A crucial enzyme that plays a major role in the scavenging of H2O2 and lead to systemic resistance in plants. Effect of P. indica priming on catalase activity revealed that there was a significant increase, that is 48% increase in the activity of catalase in P. indica primed plants under natural incidence of Bindi yellowing mosaic virus compared to the diseased plants. Activity of phosphatase was also assayed. Phosphatase activity was assayed under 40 days after sowing, as phosphatase is a major enzyme that is involved in the phosphorus metabolism in plants. From the graph, it is clear that virus infection, Bindi yellowing mosaic virus infection, significantly reduced or drastically reduced the content of phosphatase activity. But P. indica priming maintained the level, or it inhibited the prevention of phosphatase activity in virus infected plants. It maintained the levels of phosphatase activity in naturally infected plants. So let me conclude my words by saying that P. indica colonization significantly promoted the plant growth, and P. indica priming could significantly reduce the natural incidence and severity of Bindi yellowing mosaic disease. And the decreased disease incidence and severity is attributed to the increased chlorophyll and soluble protein content with enhanced activities of antioxidant enzymes like peroxidase, polyphenol oxidase, catalase, and phosphatase. So let me conclude that. From this present study, it revealed that the P. indica or Piriformospora indica, which is a beneficial fungal root endophyte that can be effectively used or can be exploited for the management of many diseases, including Bindi yellow and mosaic virus, which is very difficult to control. But from this study, it was clear that this P. indica can be used for the management of Bindi yellow and mosaic virus, as it increases the antioxidant capacity of the plant, and also it promotes the growth of the plant. Thank you. I thank Kerala Agriculture University for providing facilities, financial support, and fellowship for the research. I also thank you, the organizing committee of the conference, for giving me an opportunity to present my topic here. Thank you. Thank you all.
Thank you, Sippy, for your nice presentation and very good slides. Now,
Thank you, sir.
Dr. Sampath Kumar,
Yes, sir. I am ready.
you are going to speak on diversity of virulence determinants, affectors, or AVR genes in Xanthomonas citrei pathova malvacearum isolates causing the cotton bacterial blight.
Yes, sir. Yes, sir. Yes, sir. Is it visible, sir? Hello.
Select your
It is.
PPT, select your PowerPoint.
Yes, sir.
Dr. Sampath?
Yes,
Yes.
madam.
Can you share your PPT?
Yes, madam.
Okay, madam. Okay.
Is it visible, madam?
Slideshow. Yes, yes. Make it slideshow.
Okay.
Select the slideshow icon.
Okay, okay, okay.
Got it? Slideshow icon.
Uh,
Down. Your presentation, you make a presentation icon or slideshow icon.
okay, madam. No, it's not showing.
ಇಲ್ಲಿದೆಲ್ಲ. Got it? Down, slideshow button. Yes.
Okay. Okay, is it visible,
Yes,
madam?
done. Yeah. It's visible now.
Okay, madam. Thank you. Respected chair, co-chair, and organizers. Good morning to all. Is it audible, madam?
Yes, it is
It is
audible.
audible.
Okay, thank you. Myself, Sambat Kumar, plant pathologist from Central Institute for Cotton Research Regional Station, Coimbatore. So I am going to give a talk on the diversity of virulence determinant effectors, that is AVR genes in Xanthomonas cryptopatha or Malvaceae isolates, the causal agent of cotton bacterial bite. We know that the cotton, the white gold called as finger fibers, is one of the most important commercial crops of India. So it is India. It's grown around 125 lakh hectare with an production of 360 lakh wells with a productivity of 486 kg per hectare during the year 2019 and 20. So it's mainly grown in three regions of India. That is divided into three regions: North Zone, second one is Central Zone, and the third one is South Zone. So North Zone consists of Punjab, Haryana, and Rajasthan. Central Zone consists of Maharashtra, Gujarat, parts of Orissa and Madhya Pradesh. South Zone consists of Telangana, Andhra Pradesh, Tamil Nadu, and Karnataka. So among these pest and diseases, cotton is affected by several pest and diseases. Among the diseases, leaf curl is a major problem in the entire North Zone and restricted to only North India. But another leaf spot disease like bacterial bite, Alternaria leaf spot, and another wilts and root rots are spread all over the cotton growing zones of India. So this bacterial bite is one of the most important disease where it's prevalent all over the world. So in Australia and all, it's mandatory to breed resting for this bacterial bite. So in India, we have surveyed the bacterial bite incidents and we have studied the diversity effectors. So during the survey, we found symptoms bacterial bite have the five types of symptoms. So among them, four type of symptoms we have observed, like what we are seeing: angular leaf spot and vein blight. Apart from that, we also observed petiole blight, black arm symptom, and bold rot. So we have collected around 34 isolates all over India from three cotton growing zones. Mainly five isolates from Punjab and 10 isolates from Maharashtra and five isolates from Karnataka, three isolates from Telangana and Andhra Pradesh, and eight isolates from Tamil Nadu. The positive control PCCAs are obtained from CACR Nagpur for this study purpose. So these isolates were isolated on mutant agar medium, and the pathogenicity was proved on susceptible cultivar LRA 5166. This race identification was performed using 10 cotton host differentials among these isolates. Around 5 different races were identified. Among them, race 18 was major one. That is a major problem all over the world. All the rescue feeding programs are directed towards the race 18 because that is a highly virulent isolates all over the world except Africa. So among these isolates, we have studied the effector diversity among those isolates. So by seeing the plant pathogenic effectors, these are all the proteins secreted by bacteria host cell, usually type 3 secretion system and others. So in general, bacteria, we have around 12 secretion systems, type 1 to 7 and 9. Apart from that, we have sortase and injectosome and general sex system, that is general secretion system. That sex system is further divided into sec A to sec B, sec, and further all those things. So generally, in plant pathogenic bacteria, this type 3 secretion system was studied very well, and these effector proteins are determining the race of the particular bacterial pathogen and its aggressiveness. Apart from this, these proteins are essential for their bacterial virulence and host specificity. So, in general, these effectors are further divided into tall effectors and non-tall effectors. These tall effectors, that is transcription activator like effectors, that is basically called as Avirulence BS3 family. So that is the most important, and these tall effectors are highly present in all the Xanthomonas pathogens. Apart from that, non-tall effectors like ZOPs and HOPs are also studied. ZOPs means that is Xanthomonas outer proteins, and HOPs are HRP dependent outer proteins. HRP means hypersensitivity response and pathogenicity genes. So if the pathogenic bacteria attacks the plant cell, if the plant is susceptible, the pathogenicity will be proved, that is through HRP genes. If the plant is resistant, it shows hypersensitive response through programmed cell death, where the hypersensitive through hypersensitive response. So in general, this number of effectors have been studied in the Xanthomonas. So from 2000 onwards, a lot of papers appeared about these bacterial effectors. So most of the studies were directed towards this AVR BS3, that is tall effector family. In tall effector family, so many families are there, like hops, zops, and AVR BS3. So these are all, but most studied was the type 3 secretion system effectors. So this, generally this type 3 effector proteins are directly injected into host plant cells, where it in susceptible reaction it affects this defense signaling of the plant cells through different pathways. Xanthomonas citrei, there are a number of few papers are available. So among them, so many previously it was called as AVR genes. Then slowly the people have shifted to study more on these bacterial effectors. So in 2013, Joy and co-workers, by doing the complete genome sequencing of these two highly virulent Malvacearum isolates, they identified 181 and 178 potential virulent related factors. They classified that type 1 to type 4 secretion system, type 1, 2, 3, and 4. So in 2017, Phillips and co-workers studied the 4 complete genome sequence of the pathogen HCM along with the 13 draft genomes of this pathogen, and they could identify 24 conserved and 9 variable type 3 factors. So this type 3 effectors, mostly specifically this highly virulent race 18, the 3 to 4 by 5 more effectors than others. So right now, after this BS3 family, that is AVR BS3 family, that is tall effectors, now the people have started the more concentrating more on the tall effectors. That is in this year, in 2020, people Hug and co-workers from China, they have identified 6 tall effectors from these Xanthomonas citrei or Malvacearum. So generally, these 8 to 12 tall effectors are Xanthomonas citrei or Malvacearum. So right now, 12 complete genome sequences and a number of draft genomes are available at NCBA. So for this, so we have found this, we have analyzed several papers and other things, and we identified 5 different effectors which are responsible for a different type of symptoms in the plants. For example, in general, these ZOP effectors and HOP effectors are mainly responsible for the pathogen infection and multiplication, that is survival in the host. This AVR BS3 is responsible for the pathogenicity of the pathogen in the host plant. So we have selected these 5 effector protein genes, and these primers are designed based on the sequences available in the NCBI database. So we have downloaded this Xanthomonas citrei or Malvacearum whole genome sequence of the AR81009 and specific gene, the specific sequence of the particular gene, that is AVR-BS3, ZOP1 and ZOP-K were derived from the NCBA, and primers were designed using Primer 3 software in the NCBA website. So for this ZOP-AD was designed from the sequences retrieved from the MS14003. Then HOP-G1 was primers for HOP-G1 was designed from this strain XM M1003. So these primers, so Primer 3 software gave a number of primers for this amplification of specific effector genes, but we have selected the primers which has a high number of GC content along with the melting temperature around 60 degrees Celsius. So these were PCR using these primer sequences. We amplified the sequences using our isolates with the handling temperature for each gene. So finally, we got around 13 positive bands for these 34 isolates for AVR-BS3, where the AVR-BS3 is mainly responsible for pathogenicity of this Xanthomonas citrei or Malvacearum in the host plants. Apart from this, after that we have identified 8 isolates are positive for this ZOP-K, and 7 for half G1, and 6 for job AD, and 5 for job I. So by correlating this the presence of different effector genes in the pathogenic isolates along with their race, mostly this race 18 are highly virulent isolates. Most of these race 18 isolates have this ABR BS3 gene along with some other genes. But these are all happened only in general zone. Among the in south zone isolates, so this even this race 18 did not have the effector genes, but some other effector genes like HOP G1 and ZOP I and ZOP AD were present. So based on this study, so we conclude that these Xanthomonas citrei have different variable effectors in each isolate. So this generally number of isolates have been identified like ZOPs, HOPs, and AVR basically like tall effectors. So more number of primers which is used for to screen further of this to find out the number of effectors will further elucidate the specific involvement of these specific effectors in function in implant analysis also will help further analysis. Thank you.
Thank you, Dr. Sampath Kumar, for your nice presentation. Now we will move on to the last presentation of the session. Dr. Somath,
Yes, sir. Yes, sir.
please start sharing your slide. You are going to speak on transcriptional analysis of Tetraedra cyperi, the causal agent of the sugarcane systemature. It reveals its adaptation to parasitize monopod hosts.
Okay, sir. Sir, I'm trying to share now. It's showing that we can't display your shared content. Make sure that you are allowed permission.
Somnath,
Yes,
hello?
sir. Yes, sir.
Wait for 1 minute, just a second. We have to make you as a presenter.
Okay, sir.
You can now.
Okay, sir. Share. Make sure that you have allowed permission to share the content and then try again. Okay, permission.
Given permission is given.
Okay, sir. Just a minute. I'm trying to. We can't display. It's showing that we can't display your. Okay. Yes. Share application.
Somnath.
Just a minute. Switch. Reclaim the host role.
Have you opened your PPT on your system?
Yes, ma'am. Yes, ma'am. I opened. I'm trying to share. So
Yeah, done.
it's showing that sharing. Then you have not allowed permission to share the content. So I don't know how to.
1 second.
Setting. I guess there is something to do with the settings.
Please log out and log in again.
Okay, sir. Just a minute. I'll log it out.
ಇಲ್ಲ last.
Hello.
Enter, no?
Hello.
Sir.
Somnath.
Hello.
Yeah, hello, sir.
Yes, yes. Am I audible now?
Yes, yes.
Yes, sir. Audible, sir.
Yes, but still it's showing that you have allowed, make sure that you allowed. Okay, share content.
Somnath, what is your account name?
Sir, Somnath Pokhre.
Yeah. It's showing. There is a problem with your bandwidth.
So I'm connected to LAN now. No, it's
showing
showing
some-
that I have not allowed something to share the content. So I
No,
need to,
we have allowed actually.
not from your side. Most probably it's from my side.
Yeah.
Yes. Exactly. There is some problem from your side. Please check again.
Okay, okay, ma'am. I'm trying now.
I think that PowerPoint can be collected by the host and present it, no? But that person can talk. And,
Yeah, if he mails to us, we can do it, sir.
yeah, that would be better.
Just a minute, sir. Ma'am.
Let him try one last time. If it doesn't happen, then we can do it, sir.
You will give to next person. By that time, you will get it.
The last one.
Sure. Okay, just a minute. Next. Sending audio.
Wait.
Share content. Yes. I'm trying hard, ma'am. It's not able to share the content. So maybe I can email the meeting.
Yeah, quickly, if you can email us your PPT, we will see if we can upload it here.
So on which email?
Minute, if you can do it.
In which email?
Plant Pathology COH Bengaluru. Plant Pathology COH Bengaluru. Yeah, quickly.
Yes. UHS Bengaluru. At the rate gmail.com.
Yeah.
Dr. Ali, can you please close your PPT? Yes. Yeah.
I'm sending it, madam. Just a minute. Sorry for the trouble. Madam, I sent it. Hello? Hello? Can you hear me?
Yeah, please wait. Please wait.
Yes.
Just a minute, we are checking.
Hello.
Somnath.
Yes, yes.
Dr. Somnath. So we still not got the mail.
Okay. In that case, just I'm still trying to connect also. Sharing.
Ah.
Madam, there is something issue with that. I got an email also. Bounce back. So plant pathology.
COH Bengaluru.
COH
Bengaluru. BENGALURU.
Yes. Yes.
Not possible. We'll switch over to next session.
It's already sent, sir. Can you check it now?
Not received.
I'm sorry. Should I maybe
so
we can
later?
Yes.
Chairman and can take it.
Dr. Devappa sir, already we are behind schedule 20 minutes.
Will start. Chairman.
Thank you very much for all the speakers who participated in this session on and all the invited speakers. They delivered a wonderful lecture on perspectives of plant immunity and role of nitrogen nutrition and the defense system, plant pathogen infections. The other three speakers who presentations also did a very nice presentations, and the slides which they showed us was excellent, and the information they provided was very nice. And I'll request now the co-chairman of the session to make his remarks.
Thank you, sir. We had totally 6 presentations and 2 keynote presentations. They were very important presentations on emerging perspectives on plant immunity, and wherein the tobacco rattle virus gene silencing has been explored for the plant-triggered immunity. The buzzwords that we see in the recent science, the PAMS, MAMS. The work of Professor Dinesh Kumar on autophagy is an eye-opener for the recent happenings that are there in the plant pathology, especially with respect to plant pathogen, host pathogen interactions. And Dr. Jagdish Kumar gave a very good detailed account on the per se performance of the biocontrol agents under stressed situations, especially with respect to the nitrogen nutrition as well as nitric oxide per se with respect to the performance of the different biocontrol agents. Definitely we can draw good recommendations from this session. There are totally 4 oral presentations listed. However, we could able to have 3 presentations and 1 presentation will be adjusted to the remaining session. The presentation on Manjunath was Dr. Manjunath was very good, and I will take one of the comment from the chat box Shalini Yerkulla, she wrote, research on pomegranate is very nice and your presentation was really informative. I am from USA working at University of Tennessee. I am interested to know more about your research. My email ID, so definitely Shalini, we will share your email ID with Dr. Manjunath and you will be in a position to interact. And we have one or two quick questions also. One question, maybe all the questions listed in the chat box, I will share with the respective the speakers so that they can answer you wherever it is possible. And we had a presentation by Ms. Sippy wherein it is the exploration of the endophytes in the management of virus disease, which is a very good approach. And the work of Dr. Sampath Kumar on effector genes, I feel he has reported a good number of effector genes governing the cotton bacterial blight determinants for virulence factor. So all the four presentations, the five presentations were really excellent, and I hope the rapporteurs have made some points. We will, Dr. Sadanand as well as Jayasri, we will send the proceedings to the organizers at the earliest. So I thank the organizing secretary, Dr. Devappa, and his team for giving me an opportunity to co-chair this session along with Dr. P. Prasad sir. Thank you, and now we will close this session. Thank you.
Thank you, sir.
Thank you, sir. I thank the chairman and co-chairperson, and the rapporteurs and all the delegates and participants, and even the presenters for the nice session. Now we move on to the fourth theme, sorry, climate change and pathosystem, where the chairperson for this session is Dr. R.D. Ravan sir, and Dr. M.S. Kulkarni sir is the co-chairperson.
Yes, I'm here.
Sir. And our rapporteurs are Dr. Gurudat Tekde and Dr. Kiran Kesi. First of all, I would like to introduce the chairperson, Dr. R.D. Ravan sir, who has worked as a principal scientist and head, Division of Plant Pathology, IHR Bangalore, and whose main specialization is fungal plant pathology, who can solve any field problem with within a seconds, and he has more than 300 publications. He's a life member of many societies like Indian Phytopathological Society, Indian Society of Plant Pathologists, and many more. He was awarded Dr. M.H. Marigoda National Award in the year 2003, and he's presently working as Principal Consultant, Crop Mantra Consultancy Private Limited. This is a very brief data of sir. There is a lot more to tell, but because of the time constraint, I would restrict myself to this. Then I would like to introduce our co-chairperson, Dr. M.S. Kulkarni sir, who is presently Dean, Postgraduate Studies, UHS Bagalkot, who has extensively worked and specialized in management of viral diseases in horticulture crops. And he has served in different capacities as Dean, DSW, and presently is the Dean of Postgraduate Studies. He has been a member of the task force and chairman for the management of viral diseases. He is a fellow of Virological Society, and he has guided many PhD and MSc students. Welcome you both, sir, for this session.
Thank you.
Now over to the chairperson and co-chairperson. We have two keynote speakers here. One is Dr. Vasant Kumar and from Sri Lanka, and Dr. Anti Kumar from Delhi. So over to Raval sir.
Thank you, madam. Thank you very much for introducing me. Well, friends, my dear Dr. Devappa, Dr. Krishnappa, Kulkarni sir, I welcome everybody. And for this session, this is very important because we are talking about climate changes and all these things. As Dr. Sangeeta has told, there are two keynote addresses and 4 are the oral presentations. And already I think we are running behind the schedule, maybe half an hour or so. I request my speakers to please maintain the time and all these things. The keynote speaker will be getting half an hour, where the oral presentation will be 10 minutes. And I think at 25 minutes, we give a tinkle that yes, you have to wind up and all these things so that we can maintain the timings. And with this, I must start. Now, first keynote speaker is Dr. K.L. Santakumar. He's from, as Sangeeta, she's from Sri Lanka. Santakumar joined University of Ruhuna in Sri Lanka and as a lecturer in 1994. Then as a full-fledged professor, he became in 2009. And he was head of the department of plant agriculture biology faculty and agriculture university of Ruhuna, where he has served for a long time. He graduated from the University of Ruhuna again in 93 and started working there only. And he got his PhD from our university.
Rahul sir, you have muted yourself, sir. Rahul sir, please unmute. Rahul sir, we cannot hear you. You have to unmute.
Yeah, he has to unmute. Yeah. Yeah.
Yes.
It became automatic mute. Okay. Then he did his PhD from University of Agriculture in Bangalore. He did from Bangalore, and then he has many research accomplishments. He is a leader in the many international coordination, and now he also has worked on sabbatic leaves in Tea Research and Education Center, and he is also having collaboration with many international organizations. At present, he is specializing in mushrooms, and he has got a good experience of his topic will be plant pathogen interaction under the elevated atmospheric carbon dioxide and concentration. I request Dr. Vasant Kumar to please take the stage and start your lecture. Dr. Vasant. Some problem with Sant Kumar?
ಹಲೋ ಸರ್, ನಾನು ಪಂಕಜ ಮಾತಾಡ್ತಿರೋದು ಪ್ಲಾಂಟ್ ಪಥಾಲಜಿಯಿಂದ. ಇದು ನೀವು ಅಟೆಂಡೆನ್ಸ್ ಇದು ಕಳ್ಸಿದಿರಲ್ವಾ ಸ್ಟೆಪ್ಸ್, ಅದರಲ್ಲಿ ಲಾಗಿನ್ ಆದ ತಕ್ಷಣ ಸೆಟ್ಟಿಂಗ್ಸ್ ಒಂದು ಮೂಲೆಯಲ್ಲಿ ಬರುತ್ತೆ ಅಲ್ವಾ, ಅದ್ರ ಓಪನ್ ಮಾಡಿದ್ರೆ ಮೀಟಿಂಗ್ ಸೆಟ್ಟಿಂಗ್ಸ್ ಬರೋದಿಲ್ವಲ್ಲ? ಒಂದ್ ನಿಮಿಷ ಇರಿ. ಒಂದೇ ಒಂದು ನಿಮಿಷ, ಶಟ್ ಡೌನ್ ಆಗ್ಬಿಡ್ತು. ಒಂದೇ ನಿಮಿಷ.
Dr. Vasant sir, yeah. Open the PPT on your laptop or desktop.
ಲಾಗಿನ್ ವಿತ್ ಗೂಗಲ್ ಅಂತ ಬರುತ್ತಾ? ಅಲ್ಲ, ನಾವು ಗೂಗಲ್ ಇಂದಾನೆ ಲಾಗಿನ್ ಆಗಿರೋದು ನಾನು. ಒಂದೇ ಒಂದ್ ಸೆಕೆಂಡ್ ಓಪನ್ ಮಾಡಿ. ನೀವು ಮಾಡಿದ್ರಾ? ನೀವು ಟ್ರೈ ಮಾಡಿದ್ರಾ? ಹೌದಾ? ಕರೆಕ್ಟ್ ಹಾಕಿದಿರಲ್ವಾ? ನಾನೇನೋ ಅದೇನೋ ಸ್ಯಾಂಪಲ್ ಎಲ್ಲಿಂದ ನೆಟ್ಟಿಂದ.
Open your PPT on your desktop or laptop.
ನೆಟ್ ಬೇರೆ ಇಲ್ಲ.
Then click on the share button.
ಒಂದ್ ನಿಮಿಷ, ನಂದ್ರಲ್ಲೇ ಜಾಯಿನ್ ಆಗ್ತೀನಿ.
ಡಾಕ್ಟರ್ ಸಂತ್ ಕುಮಾರ್,
ಈಗ 12 ಗಂಟೆ ಕ್ಲಾಸ್ ಇತ್ತು, ಅದಕ್ಕೆ.
Vasant, Dr. Vasant, we are not able to hear you.
ಈಗ ಸೈನ್ ಇನ್ ಅಂತ ತೋರಿಸ್ತಾ ಇದೆ. ಸೈನ್ ಇನ್ ಆದ್ರೆ ಥ್ರೂ ಇಮೇಲ್. ಸಾರಿ, ನಾನು ಥ್ರೂ ಗೂಗಲ್ ಸೈನ್ ಇನ್ ಆಗೋದು. ಓಕೆ,
and then come back to Vasant. Vasant, Dr. Vasant.
Vasant Kumar, are you audible?
ಒಂದೇ ನಿಮಿಷ. ಒಂದು ನಿಮಿಷ ಫೋನ್ ಮಾಡ್ತೀನಿ ಇದು ನೆಟ್ವರ್ಕ್ ತುಂಬಾ ಸ್ಲೋ ಇದೆ.
Dr. Sangeeta, he's not able to
ಹೌದಾ
get
ಸರಿ,
connection
ನಾನು
or what?
ಲ್ಯಾನ್ ಕನೆಕ್ಷನ್ ಮಾಡ್ತೀನಿ ಒಂದೇ ಒಂದು ಸೆಕೆಂಡಿಗೆ.
He's not able to get or what? Now his video is also not there. नहीं आवाज आ रही, ये ऑटोमेटिक म्यूट हो जाता है। नहीं अब तो खुला, बीच में करना पड़ता है। What, Dr. Sangeeta, what should we do?
One minute, sir. Sir is come there. Sir will inform you. ಇಬ್ಬರನ್ನ ಮಾಡಬಹುದಾ ಪ್ರೆಸೆಂಟ್?
Can we exchange the next person if he is ready?
Should we go with the next person then? Dr. Kulkarni sir.
No, if he is ready, that will be better, sir.
Yes, sir. I think Dr. Kumar is there. Dr. Kumar, please unmute yourself.
I am unmuting. Is it audible?
Yes. Yes, sir. Yes, sir.
Yeah.
Yeah.
Sir. Sir and Kulkarni sir. Kumar sir is ready.
Can I upload my presentation?
Yes, sir. Maybe the chair and co-chair will introduce you.
Yeah. In the meantime, I will upload.
Yes, sir. Please open your presentation, sir, and then click the share button. Done. Sir, Raval, sir.
Yes, I'll now speak about Dr. Amdi.
Yeah.
Yes, sir.
I'm just uploading. Is it visible on this?
No, sir, not yet.
Yes, yes friends. Hello. Friends, there's a problem with the Dr. Vasant Kumar's PPT and uploading. So we'll go to the next speaker. Our next speaker is Dr. Anand Kumar. He is from PhD Principal Scientific Plant Pathology. He is working in the Division of Plant Pathology at IRI, New Delhi. And he has worked 23 years research experience in various institutes of ICR like IASR, Calicut, and then he was moved to IARI. And one of the he has got one project on DBT where he has done a lot of research output and all these things. He was also in the DBT fellow in Germany. And then currently he is working on plant microbes and plant immunity microbial volatile genes and research here. And he has got a very brilliant record of the research, and he is a fellow of many societies. We'd like to hear more from him, and he has got award of MJ Narasimha Award in PhD Best Research Paper. J.S. Pruthi Award he has got. He has got also a research publication 128 citation in 1370, and he has other teaching experience also. Now, the topic will be genomic tool to track the boundary movement of the globally significant plant pathogens. Dr. Andy, please.
Yeah, please. Thank you, sir. The is it audible, sir? Here, is it audible, sir?
Yes, sir.
Yeah. Yeah, fine. Yeah. Yeah. Thank you, sir, for your nice introduction. I'll just get into the presentation, but before that, let me thank the organizers for inviting me to share our experiences, what we gained in the last few years on very important plant pathogens. The transboundary migration of some pests from Africa. That was quite known to everybody. It was quite also in the environment. But what is quietly happening in global agriculture is the transboundary movement of pathogens. They are quite invisible. So, of course, you need different kind of gadgets to see their movement or migration and their invasion in a country. The picture what I showed here is a fungal pathogen. Of course, you cannot see them, but you need sophisticated gadgets. So this talk is entirely revolving around the recent developments in genomics and also the genomics-driven technologies, how it can help us to understand the transboundary movement of important plant pathogens. So I'll move on to the very important slide. The global connectivity, all of you must be knowing. You can reach any part of the world in less than 24 hours. So this global connectivity, coupled with rapid movement of commodities across different continents, is a serious issue. The trade-related acceleration of these different commodities are the serious problem. And they have the potential to change the global landscape of plant pathogens. Please be reminded of several pathogens which got introduced in a different part of the world. It was not there before. So the commodities, in particular, agricultural, horticultural commodities have got the succulent nature of these commodities. Horticultural commodities can really transfer the pathogens from one part of the world to another part of the world. And coupled with the so-called climate change effects, the problems are really compounded here, and the disease landscape of different countries are getting changed over the period of time. It led to rapid introduction of different pathogens. Often it goes unnoticed, and certain pathogens came to limelight. We will see in the coming slides what are those pathogens. Of course, I highlight vulnerable is the horticultural commodities because of their succulent nature and high moisture content that paves a wonderful opportunity for this pathogen to survive for a long time. Let me take you to the history of plant pathology. It plant pathogen, phytophthora influenza. So it's called as blast. It is still happening, and it is continuing to potato that was introduced into European continent somewhere from Central America or South America that has created a havoc, and all of you must be knowing very well. And the recent wheat blast, the outbreak that happened in Bangladesh. And these two pathogens are, I would say, a reminder for all plant pathologists that the plant pathogens can move any part of the world because they do not recognize any boundaries. That is what they are, transboundary plant pathogens. So the impact of late blight, all of you must be knowing, the impact is felt everywhere in the world, right from, I would say that the socio-economical impact of this late blight is enormous. And the very strange thing between among these two pathogens, the late blight of potato, these two pathogens, they origin in South America. So this is very a coincidence, something maybe 200 years ago or 150 years ago, and what has happened a few years ago. The pathogens, they do migrate from one part of the world to another part of the world, and they can create havoc in the country where it is getting introduced. So I would say that the introduced pathogens are more dangerous than the native pathogens for different reasons. So the connecting point between these two pathogens are the material which is introduced into the country. In the case of late blight, it is the rudges, the Irish potato, which came from upon a different kind of selection from South America. In wheat blast, it is a wheat seed which was imported into Bangladesh for different kind of purposes. And these two commodities from a distant land that is right in South America. So two pathogens, of course, really highlight the importance of the transboundary pathogen. Get into the tools, I would like to really impress upon the audience who is sitting here some of the impact of this late blight of potato. This is popularly called as Iris famine. In fact, Iris famine is synonymous with this late blight of potato. And this is introduced into from South America into the entire European continent, in particular Ireland. And of course, it led to quite a lot of issues in those countries: hunger, migration, unemployment, poverty, and even death. So, of course, this pathogen is still evolving, still an open challenge for all plant pathologists. And of course, the positive side of this late blight was it paved the way for the modern plant pathology subject, what we practice in the world right now. And it's also contributed towards a theory, which is now the foundation for the entire microbiology and plant pathology, that is the germ theory. The germ theory born out of this particular discovery, particular issue. And DeBerry has contributed enormously to bring this theory into science. So the picture what I'm showing here is a statue that reminds all of us that plant disease can create hunger, and this hunger should not be there forever. That's what the statement is very important here. Never again should a people starve in a world of plenty. This is very important point for all plant pathologists. I'll take you to the next slide, some of the globally important bacterial plant pathogens, which is reported to be having the potential to move from one part of the world to another part of the world. So it is highlighted here. We have Dickia as a major genus. It is transmitted through the succulent tubers. We have, of course, Clavibacter michiganensis. We have Ralstonias. We have Xanthomonas, both by seed as well as through the horticultural commodities. And these pathogens are have the potential to get introduced into any part of the world if an opportunity is given. So, of course, the bacteria are ubiquitous. They can survive. They can colonize any part of the plant, both interior and exterior. The picture what I'm showing here is a different compartments of the plant is colonized by one or other bacteria. And remember, most of these bacteria are plant pathogenic bacteria. And of course, the bacteria not only habitats, they also move on its own and also assisted by different plant parts and plants as well. This is some of the thought process what they survive, and plant is getting transmitted. They are getting different pathogens and ginger rhizomes and stem cuttings of commodities such as a banana. Every commodity is having its own associated microbiota, which includes the pathogens as well. So with this, I'll just move on to some of the classical fungal pathogens. In addition to what I already spoke, that is late blight of potato, we have several other pathogens. All of us have studied them in the different kind of textbooks. and I just updated it with the recent one, that is magnoprotective rhizae pathotype triticum. It got introduced into Bangladesh in the year 2016. So this is a kind of, again, globally significant fungal pathogens. They got introduced into one or other country from another country. The origin is different and got introduced from another country, and where it got introduced, and those countries really suffered a lot in terms of crop losses and the crop loss associated issues. It may be a social issue, or maybe sometimes it may be even a political issues. So these are all the impact of the transboundary plant pathogens. Of course, the banana, the is again a reminder to all of us that, again, the story is not yet changed. So what was happening in 1960s, it replaced popular banana cultivar that is across the story. Serious crop losses. And that I just move on to issues why this transboundary movement happens. This transboundary movement happens because of already told global connectivity and internationally agreed export and import, and of course planting material exchange. And the third important is very the inadequate implementation of quarantine measures. And of course, I already told the pathogens do not identify the boundaries with the other support system. And once it is introduced, they have the potential to evolve. One of the case studies I will be highlighting, after getting introduced into a land mass, these pathogens can, in its own, to create different kind of issues. So, different kind of dispersal mechanisms. I think all of you must be knowing there are two kinds of dispersal mechanisms we studied. One is the auto transmission and also assisted transmission. So what is more important here is the assisted transmission can be legally contained through different kind of mechanisms. So, but the auto transmission, you cannot do anything with that. So here comes the role of plant pathologist, and which one is getting really auto transmitted and which one is getting assisted transmitted. We need to segregate them and come out with a kind of policy how to contain these pathogens in different part of the world. And this is some of the issues I just flagged it here. I'll just move on to the most important, the core aspect of my presentation here. That is how to track them. We know that the pathogens are getting transmitted from one place to another place. Is there any way to monitor them? Unlike the pest, what we witnessed in the few months back, the pathogens, they cannot see them through naked eye. You need some kind of sophistication to see them and to track their movement from one place to another place. So I just put this the entire scenario under the title Geospatial Tracking of Plant Pathogens, and this need to be really taken up as a major I must say that any country. So here the monitoring the spread of plant pathogen through geospatial tracking must integrate different pathological approaches, starting from the conventional, that is the culture-based tools. We have the recent electronic gadgets, for example, aerial vehicles, sensors, and image analysis, and of course the molecular methods, what we call them as the genomics assisted pathogen surveillance system, or simply called as GAP2S. The GAP2S system, which has the assistance from electronic, electronic systems, and this must be in place in future to assist, or to rather track the movement of the pathogen from one place to another place. So, and that's going to be a reality in future. So I'll just take you to a slide which is really emphasizing the culture-based methods. They are very important to characterize pathogen, and different variants of these methods are now available. I just put a highlight here. We have a phenotypic fingerprinting by biolog. We have a FAME-based for bacteria, and we have diverse taxonomic strategies for identification of different kind of pathogens, including fungi. And of course, the culture method has got its own difficulty in understanding the nature of pathogens. So we need some kind of sophistication. This is a snapshot of different kind of tools what is available for pathologists to come out with a very sophisticated surveillance system for plant pathogens. And under the title, what is called as genomic assisted pathogen surveillance system, we have a different kind of tools. So here I must emphasize there are two parallel things are needed here. One is the technology per se, and second one is the databases. Because most of the time we need databases to compare the results what we are generating in our own lab with elsewhere, and to come out with a kind of conclusion. This is so important for us, because if you're dealing with the transboundary plant pathogens, certainly you need to have the global database, which is going to help us to understand how this pathogen, whether the pathogen what you encountered in your home country is same as somebody reported in some other country. So the databases and tools are so important here to meet the objective of the so-called genomic assisted pathogen surveillance system. So I just put everything in a perspective, which tool we can really use to have a complete understanding of the pathogen, or to come out with a thorough genotyping of the pathogen. I just put it under two categories. One is the pre-genomic era. If you put 1995 as a cutoff year where the genomic subject born, and now what we are experiencing is what is called as a post-genomic era. And the pre-genomic era and post-genomic eras come out with the different tools. And certainly the tools what appeared in post-genomic era are having wonderful resolving potential. You can even mechanisms even up to isolate level. And in particular, the tools like multi-locus sequence typing, surface protein profiling, multi-spacer typing, and of course the full genome sequencing, they have the potential to characterize an organism even up to isolate level. Even single base pair variation between two isolates can be differentiated with the help of these kind of tools. And not to forget, some of the pre-genomic era tools are also very, very important to know more about the phenomics of the plant pathogens. So that is already highlighted here. So I'll just take you to one particular tool which I will be focusing in the rest of my presentation. We have an array of genomic tools, which is science. I am not going to talk the entire spectrum of that. Instead, I will be focusing one particular tool on how this tool has assisted us to resolve or to answer some of the biological questions we had in our research agenda. To start with the multi-locus sequence typing, in a couple of slides, I will be telling you the technology, how it should be done in a laboratory. So you need to have certainly the whole genome sequence information of the pathogen. Of course, they are already, most of the global pathogens are already sequenced, and the information is already uploaded in public domain. So it's nothing like you need to really sequence your organism. The data is already there. Since the data is already there, now it gives you a platform to carry out a variety of experiments in plant pathology. For example, if you want to characterize an organism, if you want to characterize a pathogen which got entered in a territory, if you want to characterize them and come out with a kind of interpretation, you can use this technology. So in multi-locus sequence typing, this is a typical workflow, internationally accepted one. And here you need certain genomic loci that need to be sequenced, number one, and the sequence need to be compared with the database, and you need to find out the polymorphism at a single nucleotide base level, and you start converting that polymorphism into a single digit number, what is called as allele numbers, and this allele number become an input data for making different kind of interpretations in transboundary pathogens. So I'll just move on. Am I audible to all of you?
Sir, audible, sir.
Yeah, okay,
Audible, sir.
yeah. So, yeah, fine.
you are
So
audible.
yeah, thank you so much. Yeah. So this, the flow is something like this. You need to sequence that specific genomic loci. Usually it is a coding sequence, we take it. And those coding sequences are either they are participating in housekeeping gene, a housekeeping function. Sometimes it is effector coding genes also. It's always a combination of housekeeping genes and effector coding genes. We take it and we sequence them using a simple, the classical Sanger sequencing technology, and sequence typical workflow, the highlight of this work is you need to assign allele number for the sequence polymorphism. That's going to be your input data for any kind of interpretation. And this is the flow. What is that? It's a simple cost-effective sequencing technology. We are bypassing the cloning-based sequencing here. Rather, you go for PCR product sequencing because the technology itself is integrated so that everybody can do this work in their laboratory in the shortest possible time. The length of the sequence, what is prescribed for multi-locus sequence typing is anywhere between 400 base pair to, it can go up to 800 to 1000 base pair. So it a single pass or two pass reactions, you can come out with the sequence information about that particular loci. So once it is done, the number is allotted for the polymorphism. And these numbers are very important for us because what we do, suppose if you're handling around 7 genes or 8 genes, the genes are sequenced and the polymorphism in those 8 genes are converted into 8 different numbers. And these numbers are stitched together to form or I would say a string of this allele number. The string of this allele number is what is called as allelic profile. And this is allelic profile from the pathology perspective, we can call them as a sequence type. So this sequence type is ultimate in what they call in the taxonomical hierarchy down below. And the sequence type reveals a lot more information about any other technology which is present in our science now. For example, if 2 isolates, what you encountered, maybe one from your home country, one is already there in the database. If all these 2 isolates are sharing all the allele numbers, that means these 2 isolates are duplicates of each other. This is a simple hypothesis. Suppose if 50% allele numbers are common between 2 isolates, then you can make an interpretation that these 2 are, they were what they call sharing a common ancestor, and they evolved from a common parent in the last 100 or 200 years. So this is a simple logic we work in this particular multi-locus sequence typing. So with that, I will take you to the tour of 3 case studies, what we did in the last few years to address some of the biological questions, and they are already published. And the first one is Ralstonia solanacearum. It is a transboundary plant pathogen. We have several evidences already generated. This is one of the most notorious plant pathogens reported worldwide, perhaps except North America and Canada. And there are different varieties of these pathogens are there, creating problems in different kind of crops, and predominantly the horticulture crops. So what we did, we have a very serious problem of Ralstonia in ginger. This is caused by race 4 strains. It's very important in several ginger growing countries in Asia and Southeast Asian countries. Very serious problem. And we compared a population, what is prevalent in our country. Our country, we have 2 contrasting locations, the southern India as well as the northeast. They are separated by over 3,000 kilometers. So we had a population from these places, and we just analyzed the population genetics of these 2 populations, and we used around 8 different genes. They were sequenced. The polymorphisms were converted into allele numbers, and those allele numbers were matched among the isolates. And what we got is very interesting. This is the database, and I must tell you here, to house these databases, we have a global consortium of pathogen databases. The present one is located in the United States, and you can be in touch with the curator. Only thing is, you cannot send the sequences there. You need to send the chromatographic file of your the Sangus sequence reads. That is curated by the curator to assign the allele numbers. So you can do it yourself also, but for the beginner, it would be better if you take assistance from the curator. So this is the data what I have. The data says the isolates what we found in Kerala, the isolates what is prevalent in the northeastern very remote location, for example, Sikkim, they were sharing around 50% of the alleles among each other. So this is very interesting finding what we found. And the interpretation what we made was, the population, we cannot say that where the pathogen really got evolved, but one thing is very sure that there is an exchange of the so-called pathogen or the transmission of pathogen is happening between these 2 locations, which we really don't know what is the mechanism by which this pathogen is moving from one place to another place. And we attribute the rhizomes as one of the major carrier of this pathogen, which is getting moved from one place to another place. So the pathogen what is prevalent in southern India, the pathogen what is prevalent in northeast, they were sharing several alleles. So you may wonder whether it's a true with maybe another country isolated. It may be true also. You are going to see the same kind of
Kumar, Dr. Kumar, you have 5 minutes to carry on. Yeah, please wind up.
I'll find this answer.
Yeah, please carry on.
Yes, sir. So this is this paper we published in Plant Pathology from BSPP. And the second story is, again, the transmission between mainland and the island, what is there in Bay of Bengal. The story is once again same. We got the alleles, which is shared between the isolate, what is prevalent in southern India and all in mainland India and also the island. And again, this is a suggestive of the so-called transboundary migration. This paper we published in Plant Disease as a full-length article. And the third one is the race 3 by war 2. Dr. Raval must be knowing it very, very well. Race 3 by war 2 evolved in South America once again, and it got introduced in the different part of the world. And if you type this strain using MLST tool, you are going to get the same allele number. For example, the Indian strain what we encountered in West Bengal is almost the same as what is prevalent in Bolivia and other South American continent. So I must say that this tool has given us an idea that this pathogen can really, the transboundary movement is always happening. The second case study is pomegranate. All of you must be knowing very well. It's a very important country.
PowerPoint.
Is it visible, sir?
No. Please share it.
Yes.
So I shared it, sir.
It's nothing is visible.
Okay. Over?
I think there is something happened, sir.
I see.
Is it visible now? Is it visible now?
Now it's visible, sir. Slideshow.
Yes. Yeah.
Yeah, sir. Oh, okay, sir. Yeah, what I was telling is the story was same again between the mainland and also island what we encountered. So I'll just move on to the third case study, that is Xanthomonas. And in this study, what we did the same kind of work, but the genes are different, completely different from each other. We did a comparison of different isolates, what we got from different states of our country, and the numbers were compared. And what we found was very startling revelation from this particular pathogen. That is a clone, that is ST3, was predominant in the entire states of our pomegranate growing places. So we made a kind of conclusion here. A minor pathogen in 1950s has become a major pathogen in 2000, just because of two major reasons. One is unregulated popularization of a few elite kinds of pomegranate for different kind of reasons, coupled with the clonal nature of this pathogen that has aggravated the spread of this pathogen from one place to another place. And this paper we published in, again, Plant Pathology BSPP last year, and it appeared this year. And I'll just take you study. Before that, I must make a statement here. We made a conclusion that the diversity in plant pathogen is a reflection of actually the host diversity. This we come to a conclusion because we do not have much of a diversity in the case of pomegranate as a crop, and also the pathogen. And the third case study is the last one. And here, the Xanthomonas oryzae, Pethovar oryzae, a population we compared between mainland and also island, and that is in Andaman. And the finding was, again, very interesting finding what we got. The finding is this pathogen displayed very high level of genetic diversity, which is exactly opposite to what we found in the case of Xanthomonas axinopodes punicae. So it is extremely diverse. And again, we must be recalling that rice is also one of the most diverse crop. And what we did, we constructed the global Xanthomonas tree using a novel approach which needs allele data, that is GO-EBUST. And this data is clearly revealed that the population what is prevalent in Andaman Island is actually the population what got introduced into this island from mainland, perhaps through the infected seed. So some of the isolates what we found in the mainland is having same alleles as what we found in the island. So this conclusion also made us to think that the transboundary migration of this pathogen is a reality, and it is this is revealed with the help of MLST tool. And these are all some of the statements what we would like to make here. And again, this paper just appeared in the Journal of Applied Microbiology just this month itself. So here it talks about the migration of pathogen into a territory, and in that location it gets diversified because of various agriculture related operations. For example, if you deploy a resistant cultivar, the pathogen is going to evolve with the time. So the introduction and diversification of pathogen we have confirmed in this particular work. So with that, I will summarize my lecture. There are 5 key points or take homes. One is the genomic assisted pathogen surveillance system, or GAP2S, can be exploited in plant pathology to understand the transnational or interstate movement of pathogens. I would say that they are monitoring pathogen molecular epidemiology to elucidate the diversity within the classification. What we pathology to say that strain identity or even isolate identity is quite possible here, to develop a DNA barcode so that the data portability is ensured here across the different nations, and to explain the host adaptation of the pathogen. For example, the pathogen which is coming at a new host, you can do this MLST analysis to know from where this particular pathogen has entered into the new host, both in terms of geographical expansion and the genotype expansion of these pathogen. This MLST tool is very handy to develop high sensitive and of course very selective molecular diagnostic tool is also possible with the help of this kind of tools. So with that, I conclude my talk by thanking all my students, past and present, who contributed directly or indirectly to come out with these papers, and all of them are indirectly or directly working on these projects. I am again thankful to different funding agencies for supporting our projects. So thanks for your time. Yeah, thank you so much.
Thank you, Dr. Kumar. You have a long time I'm seeing you. Thank you very much. Very good talk. And
yes.
Now we can go to the previous speaker again.
Dr. Vasant.
Dr. Vasant is online.
Dr. Kumar, sir, can you close your PPT? Dr. Kumar, sir, can you close your presentation? Dr. Kumar, sir.
Dr. Kumar?
Getting close.
Okay.
Thank you, sir.
Not responding. No, that's why she's asking again and again.
No.
He is a very sincere worker, so she's very particular about all these things.
Yes, sir.
Yeah.
Yes, sir. Dr. Vasant, I will share your slides from here. Is that okay?
Yeah, let me see, ma'am. Just give me a minute. I know the time constraint.
Yes. Yeah, no problem, sir. No problem.
Yeah, now it has come. I will unshare it.
Dr. Kulkarni, sir. Dr. Kulkarni, will you please manage the time? Dr. Kulkarni, will you please manage the time?
He's in time.
It is gone. The presentation is gone.
It will be okay, sir.
Okay.
It is gone now. Presentation is still there.
Yes, sir.
No, it's gone. It's gone, sir. It's gone.
No,
okay, thank you so much. Yeah, thank you, organizers, for giving me opportunity.
Thank you, sir. Dr. Vasant?
Dr. Vasant? Dr. Vasant? Is he not? He's not online?
Yeah, he can't, but he will present. He has sent a chat that he will present now.
Yeah.
Yeah.
Raji?
Yeah. Is he online, Sangeeta?
Yes, sir. Dr. Vasant, I was able to see him.
I'm not seeing.
Dr. Vasan?
Presentation is there now. It is slide presentation has come.
Slide I am sharing, sir, from here.
Yeah, okay. Then. Asant Kumar, are you hearing? Is it audible to you?
I think his face is also not there. He's not visible also.
Earlier he was there.
Yeah. Yes. Developer, some problem with sharing or what?
No, we don't have any problem here. Problem with
Yes, Dr. Vasant Kumar.
Or should we take this in the end? We can take the word presentation.
Yes, sir, Chairman, sir.
Line Brana.
Sir, can we finish off the oral presentations and then take Vasan? Raman sir, please unmute yourself, sir. Yes, sir.
Dr. Kulkarni, can we move to oral presentation?
Yeah, we'll finish the oral presentation because we cannot. Time is also important, sir.
That's what. So, friends, now one keynote address is left out that I think we'll take in the end.
Yeah.
So that we can start the oral presentation. There are 4 oral presentations. Each one will get 10 minutes. Please keep to the time because already we are running very short of time, and I want cooperation from all of you. So I invite the first speaker, Dr. B.R. Kulkarni. Effect of weather parameter on the development of alternate leaf spot and the gray mildew in cotton. Dr. Kulkarni, please. Guru Dutt is sharing.
Yeah.
How come? You are a co-author. Hello.
No, he is in Dharwad.
Okay.
Now it is Dr. V.R. Kulkarni, sir.
But Guru Dutt is showing.
Both are in the same place, sir.
Yeah. Maybe.
That's
Yes,
what I'm
sir.
saying. They are the co-author.
Okay.
Yes, please.
Start. Yes. Unmute, Dr. Kulkarni.
Dr. Kulkarni, sir, you should unmute and then speak.
Yeah, now.
Yes. Thank you.
Yeah.
Yes.
Can I start, sir?
We are getting by
already late.
Can I start, sir?
Yes, sir.
Yeah.
Yes, sir. You can start.
We are requesting you to
Okay.
start immediately.
Okay, sir. Good afternoon, sir. Dr. Rawal, sir, and Dr. M.S. Kulkarni, sir, and fellow scientists. First of all, I want to congratulate that today the World Cotton Day. On that day only, I got the presentation of this as a cotton pathologist. I got this opportunity to present some of my work in front of the esteemed scientists. Cotton, as you know, this popularly referred as white gold. It is a principal raw material for the textile industry. In the world, India stands first in production, followed by the China and UK, Brazil, and Pakistan. Then cotton suffering from many fungal diseases and one viral disease. Out of those, the cotton is mainly affected by alternaria blight and gray mildew, and rust is also.
Yes, sir. Carry on, carry on.
Sir, these are some of the important symptoms of alternaria blight and gray mildew. These are the important diseases of cotton. Even though after introduction of BT also, we have more disease incidence in the field on BT also. This loss is due to the disease. It is necessary to forewarn the incidence of the disease in advance so that farmers can take precautionary measures to prevent the disease enhancement rather than the wait for curing the diseases. As diseases are weather induced, it is possible to forecast the disease in advance and hereby forewarn to the farmers not only on diseased incidents but also the necessary advisory for the timely management so that farmers can get ready to face the adverse situation. In Agriculture Research Station, Dharwad, we have conducted an experiment to know the effect of weather parameters like temperature, minimum temperature, maximum temperature, relative humidity, morning, evening, and rainy days and rainfall at the weekly. This is the experimental site, and we have taken 4 varieties, 2 non-BT and 2 BTs. One is Abadita out of that. One is Abadita, then Jaidhar, then Dr. Brand. Okay, sir. And this is the effect of weather data on the variety Abadita. Standard week of 31, the first symptom of alternaria, observed on 31st standard meteorological week at PDF 5. But it took 8 weeks to express or to appear the disease like gray mildew. It was noticed on 39th meteorological week, where the maximum temperature was 30.87, minimum was 19.27, and this is the maximum incidence of area of 18.3 in the week of 46th week of meteorological standard weeks, and gray mildew was 18.85. Like that, in Jaidhar is also susceptible to the gray mildew, and the same week, that 31st standard meteorological week only, we have noticed alternaria blight, and in 37th week of standard meteorological week, we have noticed the gray mildew. It is 2 weeks early than the previous variety, that is Abadita. We have noticed that in 46th of standard week of meteorological week only, we have maximum the incidence of gray mildew 36.29. Likewise, in case of BT hybrid, we have taken investigation, that is Bunny BT. So it was a popular and it is a little bit susceptible to these two diseases. Here also in 31st standard week, we have noticed the alternaria, and gray mildew, it was started in 39th. The peak incidence of both the diseases were on 46th week of standard week of meteorological data. And this is Dr. Brent. It is also one of the important or popular hybrid. It was there, now it is not available. It is of a myco product. Same type of observations also we have observed here also. Okay, this is the correlation values of fungal disease, like whether this gray mildew, AB means alternaria blight, and gray mildew. Here, maximum temperature, it is positively correlated significantly correlated with minimum temperature, relative humidity, and relative humidity at evening also. This is regression equations we have arrived. This R value is 92% in case of alternaria blight, and in 82% in case of gray mildew in case of Abadita variety. Jaidar, 95% of R value we have got in alternaria blight, and 92 in Jaidar. As we know, the both diseases are infecting on this Jaidar. And this is the development of prediction and the observed values. Here also we have observed same this one, and this is the graphical representation of predicted and percent disease index of the fungal disease Apadita, and this is on Jaidar variety, and this is on Bunny BT, and this next one is on the Dr. Brent. This is about my conclusion here is this alternaria blight is significantly negative correlation with minimum temperature, morning and evening relative humidity, the number of rainy days for all the genotypes. Gray mildew is significantly negative correlation with minimum temperature, morning and evening, and relative humidity in all the 4 tested genotypes. And here multiple regression analysis was performed, and disease forewarning models were developed. It will be helpful in timely management of advance to the farmers. Thank you.
Over. Thank you for very good presentation. Thank you. Yeah. Now the next speaker is Dr. Mallikarjun.
Very good.
Pardon, sir?
Yes, sir. Yes, sir. Yes, sir. Good morning,
You have
sir.
something to left, sir? So you are Mallikarjun is speaking weather influence vectors, dynamics in epidemiology of leaf curl virus disease. Yes. Yes, sir. Thanks, Mallikarjun.
Yes, sir.
Please.
Good afternoon, Chairman, sir, Co-Chairman, sir, and all the. Yes, sir. I am uploading.
So still you are uploading.
Yes, sir. It's going. Earlier participant has to exit, no, sir. Is visible, sir?
Earlier participant ನನ್ನ ಮಗನಿಗೆ ಮನೇಲಿ ಇರುವುದಿಲ್ಲ.
Dr. Dinesh Singh ji,
Dr. Kenganal sir, Dr. Kenganal sir, please open your PPT.
Madam,
Can you be able to push PPT?
Good, congratulations. ಅಲ್ಲಿ ದೇವಪ್ಪರ್ ಫೋನ್ ಮಾಡಿ ವೆಂಕಟೇಶ್ ಬೇಗ ಟೈಮ್ ಕೊಡ್ರಿ ನಂಗೆ ಅವಸರ ಮಾಡ್ಬಿಡ್ರಿ ಅಂತ.
Gurudat, please mute it.
Hello.
Yeah, please. Yeah,
Slide is visible.
Slides are visible. Visible.
Okay, sir. Okay, sir. Thank you, sir. I will start my presentation, sir. This is the research program on the weather influenced vector dynamics in epidemiology of the. We all know that change has been witnessing decline. Agriculture alone, we are losing annually more than $4 billion of rupees due to the climate change. And all of the climate change has been impacting on the pest as disease incidences, and this is one of the major components where the FU is happening across the globe. And climate change, many of you, those who are participating in might be hearing that the earlier minor disease major. They are becoming major. Metophthalablite in PCP rust in 1999 rust. Everyone. And recently, onion twisting also has been found very severely affecting the onion crop. We are all knowing that we are talking about the climate. So, in the disease development and the disease trial, what is the environment, especially talk about more important and has been playing a major role in changing the disease incidences. We all know that this viral disease also.
Okay, so that means.
Hello. We all know that these viral diseases are no exception.
Everything I use, but still I think that's an issue. Don't I use my tablet also? Maybe, but I prefer to have it a computer. But only thing is, a computer it didn't recognize the mic, so I don't sort of problem. What can we do, Sangeeta? Still the problem exists, I think. Yeah, okay. So if not, please forget about that. Sorry for the trouble. We could have probably had a session before this to sort out the problem. But anyway, now we can't do anything. Okay. Thanks, Amita. Okay. Please, okay. Good luck. Bye.
Okay. Yes.
Yes, you can present, sir.
What problem, Dr. Mallikarjun?
Mallikarjun, please present.
The message at the right time.
Mallikarjun, you are muted. Please unmute.
Now,
yes.
Audible, sir? Okay, sir. Okay.
Yeah.
These viral diseases, especially Bemisia tabaci transmitted viral diseases in case of leaf curl and tomato leaf curl in chili leaf curl and sour leaf curl. So all these are transmitted by the whiteflies. But the issue arises, how come these diseases which are occurring in different seasons are becoming so severe? And change biology dropping to climate change. This plot where we are saying it's a Mumbai yellow mosaic virus infested plot which is also one of our study area where we monitored light dynamics throughout the year. This other plot where the seed production was carried in sunflower. This entire plot was infested by sunflower leaf virus. So as we know that these viruses are being sheltered in the weeds and voluntary plots during the off season, and the whitefly which carries the viruses from these survival hosts to the main crops. So the study had the observation of the whitefly population throughout the 52 standard meteorological weeks of a year during 2016, 17, 17, 18 and 18, 19. Because of the times concerned, I'm presenting a few, 1 year board data with regard to the other year's observations, which is going to be concluded. So the weekly accounts of the whitefly population were recorded on the fresh trap, that is yellow sticky color trap, which is used for the collection of the whitefly. This was done in two locations. One was in Koppal, another one was in the Raichuri MARS. So the virus disease incidences, we know that they are going to be varying in, and we had collected the entire weather data information from our own weather station. So from the observations, what we noticed that the whitefly population was varying throughout the year in out of 52 standard meteorological weeks. So this is how the intensity of whitefly has changed from month to month. So this is during the third week of November on a sticky trap. This is on third week of the March. Then another slide you can see here, the August first week, where the intensity of whitefly population drastically declined. So it indicates that the whitefly trap during different week intervals throughout the year was changing. And that is directly having correlation with the maximum temperature and minimum temperature with the rainfall. It means to say that the highest population of the whiteflies were recorded during the winter season, March, and the lowest population was recorded during the rainy season, that is starting from July to the up to September end or October mid. You can see here the wherever the red color is seen with the data, that is the rainfall is incident, rainfall is there. Therein subsequently the decrease in whitefly population could be observed. At the temperature has remained between 24 degrees Celsius to 40 degrees Celsius. The studies earlier published, they say that the whitefly population are very determinant to the higher temperature. But the changing climate and with these outcome of the research, what we are going to find is that even in spite of the increase in temperature, the whitefly population is there. You can see here, per week, maximum of 1,456 whiteflies were counted on the each single whitefly trap, and in the increase to temperature soaring in the summer up to 41.4 degrees Celsius. Then the whitefly population was very abundant. It means to say that the whiteflies have learned to adapt the increasing temperature. So we can find the earlier publications saying that the whiteflies, the regression analysis is also showing that the temperature is positively correlating with the whitefly population, and also the rainfall is negatively correlating in both the years' observations. At the same time, we also did a study on the whitefly life cycle, that is multiplication of these whiteflies under the glasshouse using the caged atmosphere. So normally it is known that the whiteflies complete their life cycle between 16 to 24 days. But in our observations, what we found that their maximum population was observed during October, from starting from September, October, November, December up to the March. So this is how the whiteflies per plant week were counted, and maximum population was observed during, as I already said, November. And another thing importantly to understand is the earlier publications, whatever the researchers were conducted on whitefly population, they said that the whitefly population like the lower temperature. But what we found that the whitefly population can increase up to a temperature level of 38 degrees Celsius, where you can see the number of for each cycle of whitefly population was drastically. So this means to say that the total observations what we could find were taken at 20 degrees Celsius. 25 days were taken at 22 degrees temperature. Then 26 to 30 days were taken at 31 to 35 degrees Celsius. Then 35 days and above at 30 degrees Celsius. And 40 days were taken where the temperature was maintained up to 40 degrees Celsius. So all these observations, what we could find is whitefly population has altered rainfall negatively, then rainfall above 10 mm at weekly and reduce the population of the whitefly. Then dry weather is favoring the whitefly vector buildup, and more leaf curl diseases such as tomato leaf curl viruses and chili leaf curl viruses, you might be all knowing that they are increasing during the summer as well as Rabi season. So this is mainly because of the buildup of the high vector population. Then temperature of up to 35 degrees Celsius can favor the vector growth. So the highest cycle of multiplication can be seen in the dry season. So the post-southwest monsoon is most important for management of these whitefly population. So the conclusion is to say that we have to manage these whitefly population as a preventive measure before the taking up of the any Rabi or summer crops, so that the disease such as tomato leaf curl virus, sunflower leaf curl virus, or chili leaf curl virus, and even in the early monsoon, that is Mungbean yellow mosaic virus, can also be reduced by taking the preventive measures. So thank you, sir. Thank you for giving me the opportunity to present the observations. So this is totally, of course, looks to the only for whitefly population, but for 4 years, for continuous 3 years, the data says that temperature increase has nowhere giving any negative impact on the whitefly population. Perhaps the whitefly is adapting to the changing climate and the temperature as well. Hello? Hello?
Yes, sir.
Yes, madam.
Thank you, Dr. Mallikarjun, because high temperature favors the whitefly multiplication. So it's a good observation. Right.
Yes, yes, sir. Yes, sir. Even in the it is taking lesser days in warmer period for its single life cycle also.
Yes, thank you very much. Thank you.
Thank you, sir. Thank you very much.
Now, next speaker, where is Dr. Kulkarni? Sudharani.
Sudharani is not there, sir. She is not doing it.
Okay. Pushpa.
Pushpa? Kingana, sir, can you please?
Yes, madam.
close
Yes,
your-
madam. Yes, I'm doing it.
Yes,
Next is Pushpa, madam.
Yes, sir, I'm here.
Okay, madam.
Are you ready, Pushpa?
Lycopene content of bioinformatics treatment of tomato. Yes, madam. Where are you?
Okay.
Yes, madam.
Yes.
Selection of the
Just a minute, ma'am.
Yes, yes, ma'am. Yes, sir.
Please exit your slides.
Dr. Kenganal, you should close your slides, sir. Dr. Mallikarjun Kenganal?
Is he on the screen? He's not there, I think.
No, but his slides are open, sir. Dr. Kenganal?
Dr. Sangeeta, are you hearing me?
Yes, sir. Dr. Vasan?
Are you hearing me?
Yeah, we are hearing you.
Okay, that's great. Okay, so that maybe I can wait till it's finished.
Yeah, I think one presentation is there. Let it be over first.
Okay, yeah. So, sir.
Yes, sir.
Kenganal?
Pushpa is not.
Hello, madam.
Can you please?
Madam.
Jayappa sir, if you have Kenganal's number, just telephonically tell him to close the slides.
He's
Yes,
not
yes.
lifting.
Yes, sir.
Yes.
Thank you, sir. Pushpa, please share your slides, ma'am.
Yeah.
Hello.
Yes.
Hello.
Yes.
Yes,
Yes.
we are able to hear you, but we are not seeing your
Yes,
screen.
ma'am. You are
Yes.
my screen?
No.
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 Okay, 1 second, please. Ma'am, how to share my screen?
You open your PPT on your laptop or wherever you're opening.
Okay.
Okay, on your WebEx, you see the stop video button. Next to that, there is a share icon. Click on it.
Okay, stop sharing.
Still, Kangal is there. Bengal is there in this.
Dr. Kenganal,
Dr. Kenganal?
Hello.
Wait,
Dr.
Pushpa.
Kenganal,
Pushpa, did you get that icon? Share icon?
Okay, ma'am.
No. You wait.
Kengnal complete agila. He has to close.
Unless Kengnad takes off his slides, it's difficult.
No, you can make from your end. Mallikarjun Kengnal, yes, please close your slides.
Is it visible or not, ma'am?
No, it's not visible, madam. 1 minute you wait. The previous presenter has to close the slides. Therefore, ಅವ್ರಿಗೆ ಹೇಳ್ರಿ ಮಲ್ಲಿಕಾರ್ಜುನ ಫೋನ್ ಮಾಡಿ ಹೇಳ್ರಿ please.
Sir, he's online. We are talking to him, sir.
Okay.
ನಿಮ್ಮದು ಕೆಂಗನಾಳಿಗೆ ಸ್ಲೈಡ್ಸ್ ಅಪ್ಲೋಡ್ ಮಾಡಿಬಿಟ್ಟು ಬಿಟ್ಬಿಟ್ಟಿದ್ದಾನೆ. ಇಡೀ ಪ್ರಪಂಚ ನೋಡ್ತಾ ಇದೆ. ಅವನಿಗೆ ಸ್ವಲ್ಪ ಫೋನ್ ಮಾಡಿಬಿಟ್ಟು ಅದನ್ನ ಮಾಡಿ ಅಂತ ಹೇಳಪ್ಪ. Sir, there is some technical problem. Already is exited.
He has exited, but in your computer.
He is doing. Wait a minute, sir.
ಸ್ಟಾಪ್ ವಿಡಿಯೋ ಮಾಡಿ ಮತ್ತೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿರಿ.
Kulkarni sir, what is happening?
Very glad to see you, sir.
Yes, sir.
Because we remember you for immediately. We still remember the efforts for
Thank you, sir. Thank you.
Hello. Rawal sir, even I also very happy to see you after a long time, sir.
That's what I'm saying. When Devappa called me, I said definitely I have it.
Yeah, it was a really nice to see you after a long time, maybe after 20, 25 years, sir.
Thank you.
Yes,
Thank you so much, sir.
he used to come to US, Bangalore for IPS meet along with Dr. Anna Hosur and Srikanth Kulkarni, sir.
Yes, I came to your college for 3-4 times.
Yes, come. Yeah. That is also there, sir. Yes.
Yes.
So, health now, everything is fine.
Going on so far, so good.
We wish a very good health of you, sir, in future.
You people are very close to my heart.
Thank you, sir. Thank you very much.
Yes.
Now, okay. Now,
Pushpa, can you try now?
Okay.
Now Pushpa can share it.
Yes, do it.
Still it is happening.
Yes, so Dr. Ken.
Is it visible or not, ma'am?
No, okay.
no, again back.
Same problem.
Dr. Keshan, can you please exit from here?
Same problem. Same problem.
Pushpa, can you start sharing now?
There is no.
Yes, ma'am.
Yeah, can you start sharing your video?
Is it visible, ma'am?
No, it is not visible.
No, there is another PPT.
Not
already
visible.
there.
That is, yeah, not visible.
Okay. Oh, ma'am.
No.
No?
No,
Pushpa?
not at visible.
Pushpa, are you sharing from your cell phone or laptop or desktop?
Android phone.
There is some problem with it, I feel.
Okay.
No, but madam, that earlier screen is still visible.
Yeah, that is some issue they are resolving, sir. But I think Pushpa's, because of her cell phone, there is some issue. Pushpa, can you mail your PPT to us? So meanwhile, we look into Dr. Vasan.
No, wait.
Yes, ma'am.
Already clear speaker
Hello?
exited. There is a problem with mobile, Android mobile.
Okay.
Yes. So you send your this one to us. If possible, we'll share from here.
Okay.
Raval sir, can I check if Vasant is available?
Yeah, I'm here. Can you hear me?
Yes, Dr. Vasant.
Okay.
Shall I share your PPT? Let me see.
I can, yes.
Yes, I'll share your PPT.
Please see.
Yes.
No, it's not coming.
Hello.
Do you see?
Are you sharing, Vasant? Are you sharing the PPT, Vasant, or shall I share?
I'm sharing. Can you see it?
No.
Yes.
Not.
No.
Not it? Not it?
Not it.
Okay. Once again.
Yeah, try once again. Vasant, you stopped sharing. I will share from here.
Okay.
Yeah, you stop sharing. Close your PPT.
I'll close.
You close your PPT. I will share from here.
Okay, that's great. Yeah. I think that has not come, Sangeeta.
Yeah. I'm trying it. 2 minutes, sir. We are doing it.
No, Rick. Okay. So, Raza, how is everything?
Yes.
Okay. Yes.
Okay.
Yes. Yes, sir.
Slide show, please.
Okay. Thank you so much. Can I start?
Yes,
Yes, sir.
Please carry.
So I'll try to cope up with the technical problem. Okay. Good afternoon, everyone.
But you're
It's very
not
nice to have a speech at this conference. And also, I thank the organizers for inviting me and especially giving me the opportunity to meet one of my wonderful teachers, Dr. Rahul. We have been working, I was doing my PhD in Bangalore, and also to meet Professor Sangeeta, one of my colleagues, when I was studying there. And also, my special thanks for you to organize this event despite of this global COVID-19 issue affecting all the parts of the world. So that back to the business my presentation, I'm probably going to discuss a little bit on maybe there some.
Your video is not on, Dr. Vasant Kumar.
Yes, sir.
Video is not there.
I think still video I have put. I have not stopped. But if you can hear, for video will come, sir.
Okay.
Okay. Can I?
Yeah, please carry on.
Okay. So first slide, please. First slide, please. Second slide. Sangeeta, are you moving the slide?
Yes.
Okay, thank you. So, please on. Okay, yeah, please on. Okay, so the climate change, in fact, this is one of the biggest threats to the mankind at present, probably next to the COVID-19 issue. So, it is estimated that around 0.4 million worldwide, this has cost around $1.2 trillion, a big and huge amount. And as you know, around 20 to 30% of the crop production is lost because of the pests and diseases. Of course, these figures may change in different parts of the world, and also there may be some similar losses at post-harvest level also.
any future change in the climate would have either decreased or increased this loss. And it was observed, I have seen this on that economic paper, that recovering losses of major crops to fungi and especially maize, this could feed around 8.5% of today's population. So you can see how much we are losing every day because of these crop losses. Next slide. Next slide, please. Okay, IPCC predictions, everyone knows. Previous one. Yeah, so this is a prediction. The global temperature may change from 0.9 to 3.5 in between any figure of these values by year 2100. So there could be some extreme changes. For example, more frequent higher temperatures and also less frequent lower temperatures. If you think about carbon dioxide concentration, before industrial revolution, it was somewhere around 280 micromolar per molar. And today's has gone up to 9.8, that is in 2019. So this is very significant because more than 30% of the increase after industrial revolution. Next slide. So if the rate goes on this way, you can see the level of CO2 in next couple of years time. Next slide, please. Next slide. Okay, if you talk about ozone concentration, 2 to 5-fold increase has been observed during the last century. And precipitation, rainfall, it has been predicted that increased precipitation both in summer and winter in high latitude regions, but in case of Asia, it will be increased in summer. And storm patterns are very important, especially kind of diseases that are disseminated through wind. So there would be influence global movement of pathogens because of this. And more importantly, enhanced drought because of soil water content is expected to decrease in some part of the world. So these are the probable predictions due to climate change. Next one.
एक presentation हो रहा है इसको करना है और एक दिन
Next slide, please. So this is, everyone knows plant pathology, the disease triangle. And here, the host, pathogen and environment. The climate, it influences all stages of host and also pathogen. So even though the environment is not conducive for even the more severe pest and also more susceptible host, the disease may not occur. So that climate play a vital role in disease occurrence. That is what I want to put this message. Next one. Okay, this is the story. Atmospheric carbon dioxide concentrated. Picture in your website. So in 2019 September, it is 408.55. In 2018, it's 405.59. In 2017, 403 only. So that there's a big increase in the carbon dioxide concentration. This will also lead to elevated higher temperature levels. In both temperature and carbon dioxide concentrations are almost similar. Next one. So it has been observed that for production, it could differently depending on the latitude. For example, in high and mid latitude, you may expect increased yield. The lower in latitudes, the yield could be decreased. And under elevated carbon dioxide concentration, or the double CO2 concentration, the yield of C3 plants expected to be increased by 30%. So you all are probably aware that the carbon dioxide concentration has been limited factor in C3 plants at the moment. But at higher CO2 levels, definitely the yield could be 30% increase. And this probably the Agrawal's the prediction of 1%, 1 Celsius, 2 degrees Celsius, and 3 degrees Celsius in Punjab study, it has been estimated the rice yield reduction by 5.4%, and at 3 degrees Celsius it is 25%, 1%. It's a huge amount we can expect. Next slide, please. And also, the climate may influence the geographical distribution and growth of plant species. And there are a good number of examples, the prediction of these changes. And also, pathogen, it would follow migrating host and may infect vegetation of natural plant communities that have not been previously exposed to the often more aggressive strain from agricultural crops. So this would be a danger in the future changing climates. Next slide. So, do the climate, do the pathogens or the evolution or adaptation rate of the pathogen there than plants. Under change, the potentially be unable to migrate or adapt as readily as environmental conditions. Now we know that plants are not moved unless we are taking them for different parts. But in case of plant pathogens, they take the advantage of plants because they can easily move long distances, and also they do have shorter generation time. So that helps pathogens to adapt into changing climate than plants. So therefore, we may expect the adaptation in pathogens in the changing climate would be much higher than plants. So this would be the question that we have to answer in the future climate. Next slide, please. So in the next slide, we will see the impact of elevated carbon dioxide concentration on plants. So as you know, under higher carbon dioxide concentration, you can expect the increased canopy size and also density of plants. And this also generates a greater biomass production. And as a result, the microclimate may become more conducive for plant pathogens. They get more and more beneficial environment as a result. And due to assimilation of carbohydrates, there would be a very high carbon and nitrogen ratio. Next slide. So shows one of the experiments we have been doing, we have done at Horsham, Australia. So we have tested Ascochyta blight disease and with plants under elevated and ambient carbon dioxide concentration level. You see on your left side the elevated carbon dioxide concentration. The plants have been growing much better than ambient carbon dioxide concentration level. So that indicates the high canopy, and therefore there will be better microclimate created for pathogens to colonize and also infect plants. Next slide, please. In the same study, we have observed the disease, Ascochyta blight disease. You will see after day 4, the elevated carbon dioxide concentration has significantly influenced the disease index. So that in Ascochyta blight, so there would be high disease we can expect in the future. Next slide, please. And under elevated carbon dioxide concentration, you may expect greater biomass production and high carbon to nitrogen ratio. and this will lead to slower decomposition. That means the slower decomposition leader and the increased biomass and also higher winter temperature, this may increase the on overwintering crop residues, expecting more disease in the changing climate in some parts of the world. And also this will increase the amount of initial inoculation available for subsequent infection. So therefore, there could be some more and more disease conditions under these conditions. Next slide. And when we talk about host resistance, change the host morphology and physiology, nutrients and water can also affect resistance and susceptibility. For example, reduced stomatal opening and changes in leaf chemistry has been observed under elevated CO2. Therefore, in diseases like rust, down mildews, and some necrotrophs, you may expect due to reduction, there may be some lesser infection. And also decrease in stomatal density. So this may increase the resistance to pathogen because pathogen find it difficult to enter the plants to penetrate. And this has been observed in some number of examples. I have been sharing this one. For example, in geosporoids in centers, there has been the reduced germination and penetration due to tomato issue. And also some defense structures, for example, under high carbon dioxide concentration, barley plants, they were able to mobilize, assimilate into defense structures, and this will help to formation of papillae and accumulation of silica. That has increased the penetration of erythroid graminease, and this has been affected the reduction of the disease. Delay initial establishment of the pathogen because of the modification in pathogen or its aggressiveness has also been observed under elevated carbon dioxide concentration. For example, it has been observed that the lengthening of the latent period of Mavalia, that is bovine rust. Next slide. Then the next slide, I will show you one of the experiments I have done. The Hosham and the air concentration, the CO2, where we have observed that the microspherular pin nodes, the atmosphere formation was high in the initial state at the elevated carbon dioxide concentration. So that means the microspherular pin nodes is able to establish the disease condition before the stomata are closed. So that on the elevated carbon dioxide concentration, there could be a chance that the metabolite would be increased because of this reason. And next slide. And also, increased fraction of assimilated roots has also been observed constantly in crops, for example, carrots, beetroot, and also radish. So, more carbon is stored in roots, so pathogens and diseases may be reduced and the climate can change. Well, this is under elevated carbon dioxide concentration. Next slide. You can also expect the increase and decrease in the fecundity of pathogens. Next slide, please. Fecundity has been observed it increase or decrease on the elevated carbon dioxide level depending on the host and the pathogen interaction. Now, for example, in case of Pelinum, that is Timothy graph, it has been observed that at 500 and 600 ppm CO2 concentrations, the Alternaria alternata, it has produced nearly 3 times number of this graph. So this is significant, and the more the pathogen is at a more severe level under this circumstance. So the next slide show you the effect carbon dioxide concentration of plant pathogen interaction based on the type of change in the plant character. For example, we have already discussed that the crop can be enlarged under elevated carbon dioxide concentration. This may help infection sites for the pathogen, and also microclimate become more conducive for the pathogen. And also enhanced photosynthetic efficiency can also be observed, and this will help additional carbohydrate supply. This will result in increased starch and sugar, so good for the pathogen. And again, the reduced time of stomata opening has also been observed, and this will restrict the pathogen penetration as explained by Eastburn. Silicon content in rice, then this will for the disease, so that rice probably will be increased under these conditions. And then in the beta 1,3 glucanase activity has also been observed under elevated CO2, and this will reduce the leaf necrosis. And again, reduce stomatal conductance, and this will reduce the disease incidence through reduction of leaf nitrogen and CN ratio. And in this epithelial has also been observed in certain pathosystems, and this will enhance the resistance to certain diseases under changing climate. So the next slide probably will show some of the recent next slide experience on the next slide, please. Okay, different plants of pathosystems. For example, in wheat, in the elevated carbon dioxide concentration, have been observed that the stem run is expected to be decreased. So in wheat leaf rust, either is increased or decreased. The wheat stripe rust is expected as increased. In case of yellow virus is expected to be decreased, and smart disease may decrease under elevated carbon dioxide concentration. So the maize and potato and rice could be increased in some of the diseases. Next slide. Next slide, please. We have been talking about mostly the fungi. So how about the bacteria? The rhizosphere colonizing Pseudomonas and Trichoderma, they were significantly influenced by carbon dioxide, elevated carbon dioxide concentration. So this is because of the increase in total sugar concentration and the high carbon dioxide concentration. And also, in this particular example, you will see that rhizosphere colonization of Pseudomonas semidia in soil, and with relatively low carbon and nitrogen concentration has been increased from sub-ambient to elevated carbon dioxide concentration level. Next slide. Viruses, in the next slide.
Hello.
Next slide, please. Yes, sir.
Yeah. You have 5 minutes more. You have 5 minutes more to wind up your lecture, Brittany.
Okay. Fine. So on the plant viruses, tobacco plants grown at increased concentration, it has been shown to markedly decrease spread of the virus. Next slide. Next slide. And wheat plants, and they were grown at two regimes, for example, ambient carbon dioxide and elevated CO2. When the plants were inoculated with ball yellow dwarf virus at 2 leaf stage, it has been observed that ball yellow dwarf virus titer was significantly higher, over 36% in leaves with leaves grown under elevated carbon dioxide concentration. So the next slide shows, next slide please, how these affect beneficial plant microorganisms. For example, elevated carbon dioxide concentration would enhance avascular mycorrhizal fungi and also ectomycorrhizae, endophytic mycorrhizae, and plasma promoting bacteria. So these are the predicted prediction of ECO2. Next slide. Discussing about the single but what is the issue when we talk about the combined effect? For example, carbon dioxide and ozone. It has been observed that the interaction effect elevated carbon dioxide alone, or maybe in combination with ozone, it has found significantly reduced downy mildew disease by 39 to 66%. And this experiment shows the carbon dioxide concentration versus patient. You will clearly see that under elevated, there's a significantly less disease under ambient carbon dioxide concentration and supplementary irrigation. And under elevated carbon dioxide concentration, supplementary irrigation produces as much disease as strain fed. Next one. So let me brief about the research gaps in the next slide. Do the research from carbon dioxide. So most of the research have been concentrated on climate change effect on the single atmosphere constitution or meteorological variable. But we know that the environment or the interactions are more complex in the real situation. And therefore, most of the results may not predict the exact level of prediction. Next slide. And on the other hand, in the next slide, the most of the studies were on fungal pathogens. We need to discuss about the other pathogens as well. The complexity in terms of amount of information needed to adequately predict the outcomes of the climate change. Okay, next slide. And also the models that we are using to predict climate change, uncertainty and reliability of these models continues to be reported or inadequately emphasized. For example, the predictions of generally more uncertain than temperature predictions. We can easily predict the temperature increase or decrease, but other factors may be very. So this is what we have been doing in Victoria Ocean. So you will see the amount of money and amount of time we spend for this creating the changing climate. So this is the big amount of money, and we have created the very near natural environment. Every second the carbon dioxide the concentration is maintained. So this will help us to understand different aspects under changing climate. Here, but still we can change only one or two factors. So that is the main slide. And also the next that we use to detect for the climate change factors, they are also very expensive, and this may not help. Like us may not be easily afford this kind of equipment. Slide. So what we need in the future climate experiments, next slide, we need to understand, next slide, better understand the gene expression in plants and also in response to climate factors. Next slide. And also we need to understand multi-factor studies, data models we need to understand to adaptation rates. And also better data and models related to dispersal and current levels of intra-specific diversity has to be used. And next slide, you also need long-term and large-scale records of pathogen and host distribution. And models of regional processes incorporate diseases and also data and models and dispersal of propagules and spores need to be understand. How about traditional varieties? Are they respond? How they are responding under carbon dioxide concentration in the future? These also have to be studied. So therefore, created multidisciplinary and international network of data collection and synthesis have to be maintained. Next slide will, so if not, probably we may have to go to this whole era where people are struggling, people are starving due to shortage of food. So this is a very case we know about the maybe we will end up with Bengal famine or any other epidemic conditions in the future. So very much. Thank you once again for allowing me due to this technical problem. I could not go.
Thank you, Dr. Vasant Kumar,
But
for
hope that
presentation. Thank you very much. How are you? How are your family? Now, next speaker, should we call Pushpa?
Yes, sir. She's there. She's there. I will upload her PPT. Pushpa, are you there, Pushpa?
Yes, ma'am.
You can start your presentation.
Okay. Good morning, good afternoon, all of you. Myself, Puspa. I am going to present my research work, and the title of my research work is Increase in Lycopene Content of Oil Bioformulation Treated Tomatoes. Next slide, please. Yes, tomatoes. Tomatoes are the third most important vegetable and integral part of our diet. It is the rich source of lycopene, that is carotenoid, and vitamins also. Next slide, please. Lycopene. Lycopene is a plant nutrient having the antioxidant properties and the red color pigment found in tomatoes and some other fruits and vegetables. Next slide, please. Chemistry of lycopene. The molecular formula of lycopene is C40H56 and the molecular weight is 537 Dalton. It is soluble in hexane, acetone, and insoluble in ethanol. And it is stable in very high temperature. Next slide, please. Next slide, please. Yes. Uh, the procedure for lycopene extraction. We were using the lycopene hexane extraction method and read the absorbance at 503 nanometer by using the UV spectrophotometer. Next slide, please. Yes. We were conducting the field trials in 4 agroclimatic zones: IRI New Delhi, UHS Bengaluru, BCKV Kolkata, and SKUST Srinagar. Here are the some results of IRI New Delhi, in which the lycopene content is high in 4 ml and 6 ml doses, which is 26.106 and 23.495. It is very higher as compared to control. The control is 13.05 only. In the case of Alternaria solani. Next slide, please. Yes, it's a graphical representation of the lycopene content in case of Alternaria solani. Next slide, please. Yes, in case of Xanthomonas juvescatoria, the lycopene content in control is 7.96 only, but in the 4 ml and 6 ml, it's a very high lycopene content. Next slide, please. Yes, it's only a graphical representation of lycopene content. Next slide, please. Yes. Uh, uh, BCKV Kolkata. We were conducting the field trials in BCKV Kolkata. In treatment number 5, having the highest lycopene content, which is 9.9%. It's a 100% oil by formulation treatment. In comparison with control, it's a very high, and in control, only the 1.20 lycopene content. Next slide, please. Next slide, please. Yes, SKUAST Srinagar. T14 is the control without chemical or bioformulation treated bioformulation treated in which the lycopene content is 3.94. But the treatment number 12, the lycopene content is very high in which 100% all bioformulation is used. Next slide, please. Yes, it's only a pie chart of lycopene analysis. Next slide, please. Next, please. Yes, our future plan is we will extract the lycopene content from the bioformulation treated tomatoes, and then we'll prepare the tablets and capsules for making for marketing purpose. Next. Thank you.
Thank you very much, Dr. Krishna.
Yes, sir.
I think the session is over now.
Yes, sir.
Session over now. I'll request Dr. Kulkarni and
Yes, sir.
Dr. Kumar to give their comments, please.
Yeah, quickly, there were 6 presentations. All have prepared very well, and the topics are very well suited for the present climate resilient horticulture or agriculture, and very useful information has been provided for all the participants here. And I congratulate all the presenters. Only Sudharani has not presented. All 5 presentations are excellent, and I thank the organizers for giving me the opportunity to coach with Dr. Hadi Dawal, the experienced horticulturist, the horticulture pathologist. I thank Dr. Devappa and his team, and also Dean of COH Bengaluru, and also our university authorities for hosting this international webinar, which is highly successful, I think. I thank all the organizers as well as the participants. Thank you, sir.
Reporter, they have something to say?
Yeah, they
No, sir. Please go ahead, sir. You conclude it, sir.
Gurudat, I also heartily congratulate and thank the reporters, the Gurudat Hegde, as well as the other person. Yes.
Okay. Thank you, Dr. Kulkarni. Now, as a customary, I have to conclude. First of all, I thank, as Dr. Kulkarni has told, we had 2 keynote addresses and 3 oral presentations. All were having real output. We got a good data from them, especially when Sant Kumar was presenting. And it's a very good information that more the carbon dioxide, more the biomass. When there is more biomass, there will be more pathogens. It is natural because they'll multiply on that. Whereas on non-obligate pathogens will be reduced because of they cannot survive in carbon dioxide. I think this is a good generation. He has done it. And then Dr. Anand Kumar. Anand Kumar is my old friend. He has done a very good work on bacterial wilt. I remember some from India to report in ginger. I think he has given a good insight of this one, climate change with the genomic formation and all this, and how the pathogen shifts from one host to other host. That is because of that. Then there was some oral presentation. First is Kulkarni. Dr. Kulkarni has given the weather with relation to the grey light and the alternarium. So there they are giving minimum and maximum temperature, and that is but natural. minimum temperature is negative 2 in the Alton area because this is a pathogen we need a low water temperature, maybe around 18 to 20 and plus humidity. I think that's a good information here generated, which is confirming to others. Then this one, your Karge, this one, Kanigal, Mallikarjun, on whitefly observation. This is very well observed that I think Kulkarni must be knowing. More the temperature, more the insect multiplication.
Definitely, sir. Yes.
More the rain, less is the insect proliferation. It's a good confirmation, I can say. Then lycopene contained by Dr. Pushpa is a good new tech one. That means when the bioagents are used, the effect of disease is reduced. In like will increase. Yes, I thank you all the speaker and I Devappa and Gita and others and give us and hear us about him. And also I thank all the speakers. They have done a very good job. My reporters and Dr. Kulkarni, I thank you all. Hope to see you personally. Thank you very much.
Thank you, sir. Thank you very much.
Thank you.
Thank you very much, sir. Thank you for all the speakers, and special thanks to Raval sir. It's a very nice. It's an honor for me to see you here.
For me,
And
they
also
couldn't,
Vasant.
but I could not see Vasant fully. It's okay. Right then.
Sir.
Wish you have a nice lunch and enjoy yourself. Thank you very much.
Thank you very much, sir. See you again.
Thank you very much.
Thank you, Rahul sir. Thank you so much.
Well, thank you so much, sir. Wish you all the best, all of you.
Thank you, sir.
Thank you.
Thank you, sir. Thank
Okay.
you, sir. Thank you, Kulkarni sir.
Please announce the session.
Reports, I think Dr. Gurudat and
Yeah, I will send it, sir.
Kiran will give. And Dr.
We
Devappa.
will take it together, send it by tomorrow, sir, to either
Okay.
organizer or to you, sir, directly.
Okay, then I'll forward to Dr. Devappa.
Yes, sir. Yes, yes.
Okay.
We will send you. Yes, sir. Thank you very much.
Thank you very much.
Thank you, sir.
Right. Can I move?
Yeah.
Yes, sir. Thank you, sir.
Okay.
It's getting late for you. Have your lunch.
Thank you. Thank you.
Yeah.
Next time
Sir.
announcement next.
Next to 2
Okay,
o'clock
Sangeeta will announce.
sir. Kulkarni sir. Because
Now it is 5 minutes.
So 2 o'clock sharp start,
Break.
sir.
Because
Yes, sir.
now it is
sharp
1:50.
Start.
2:30 maybe.
In another 2-3 minutes, we are ready to welcome the guest. Thank you, sir. Thank you, sir.
Thanks.
Sir, good afternoon, sir. Benagi, sir.
Good afternoon.
ವಿಡಿಯೋ ಇಲ್ಲಿ ವಿಡಿಯೋ ಮೇಡಮ್.
Sir, good afternoon, sir. Benagi, sir.
Hello, sir. Good afternoon.
Yes, sir.
Hi.
Dr. Jerd Samlis, sir. Am I audible, sir?
Yes, you are audible.
Hello, sir.
Hello, namaste.
Happy afternoon, sir. Jerd Samlis, sir.
Thank you.
Hello, sir. Sir, can you share your PPT? Dr. Jerd Samlis?
Yes, of course. I take the ball.
Yes, sir. Thank you.
Screen?
Yes, sir.
Okay, I go into presentation modus. Okay?
Yes, sir. You can put it to slideshow mode, sir.
Okay.
Thank you, sir.
Does it work? Is it on video?
Yes, sir. It's visible, sir.
It's visible. Okay.
Yes, sir.
Good.
Thank you, sir. Thank you so much. Dr.
You
Benegi,
can-
sir.
Okay.
Dr. Tamaya, sir. Okay. Dr. Benegi, sir, shall we start the session now?
Yes. Please you can start now. Because
Yeah. Good afternoon, all. I welcome you all to the afternoon session of the International E-Conference on Recent Molecules in Plant Disease Management. This session is chaired by Professor V.I. Benagi and co-chaired by Dr. N. Tamaya. The rapporteurs are Dr. S.B. Gowder and Mr. Sudarshan G.K. I take this opportunity to introduce the chairman and co-chairman. Dr. Benagi, sir, he was the former professor and head Department of Plant Pathology, US Dharwad. He has worked extensively on fungal diseases of horticulture crops. He served in various capacities, starting from professor, director of extension, director of education, and finally, chancellor of US Dharwad. He has published more than 80 research articles in national and international peer-reviewed journals. He is the recipient of many awards. The prestigious Karnataka Rajyotsava Award has been deciphered to him. He is the fellow of more than 12 scientific societies. He has guided 10 PhD students and 25 MSc students. And the co-chairman of this session, Dr. Tamaya sir, I would like to introduce. He is the professor of plant pathology, UHS Bagalkot. He is presently working as Dean, College of Horticulture, Arabavi. Sir has worked for more than 2.5 decades on banana foliar diseases. He has published more than 110 research articles in peer-reviewed journals and has guided 5 PhD and 15 MSc students and is the recipient of many prestigious awards. In this session, we'll be having 3 keynote speakers. The first keynote speaker is Dr. Gerd Stambler. The second one is Dr. H.M. Venkatesh. And the third keynote speaker is Dr. M.C. Ranganath. These keynote talks are followed by 2 invited talks by Dr. Ravikumar M.R. and Dr. Suresh Ekbote with 2 oral presentations. With the permission of the chair, I would like to introduce Dr. Gerd Stambler. It gives me an immense pleasure to introduce you, sir. He is working as principal scientist on fungicide resistance research, BASF, Germany. He has published 121 research articles in peer-reviewed journals and here in this session, he is delivering a talk on recently developed fungicidal agents in the light of fungicide resistance and regulatory challenges in managing plant diseases. Dr. Gerd Stambler, over to you, sir.
Okay, thank you very much for this nice introduction. I'm very happy to give this presentation here. Can you hear me clearly and good?
Yes, sir, you're audible.
Okay, that's fine. Okay, then my title, as you already heard, recently developed fungicidal agents in the light of fungicide resistance and regulatory challenges in management of plant diseases. Next slide. Okay. You might know this sentence, farming looks mighty easy when your plow is a pencil and you're 1,000 miles away from the cornfield. This was said by a Vice President of America, Eisenhower. And I think, if you are a farmer, you are facing a lot of challenges. And one of the most important challenges you have is to keep your plant healthy. These are examples of drastic plant diseases, this Claviceps purpurea, which killed a lot of thousands of people in the Middle Age, or the Great Famine in Ireland in 1846 caused by Phytophthora infestans, where millions of people died or had to go to other countries like US. Yeah, but also I have some. Yeah. Oh, sorry. Okay, but also in our days, there are still plant diseases which endanger our food and health. And here are some examples. Soybean rust is a drastic disease in South America, and septorial leaf blotch is one of the most important diseases in Europe on wheat, rice blast. You know that in Asia, late blight everywhere. Downy mildew grapes, for example, also everywhere in every country where grapes are grown, or gray mold on different crops, and many, many more are critical to control. And we still have high losses caused by fungi nowadays. Yeah, and this is summarized by a very nice professor, Sarah Gurr. Yeah, she mentioned that still there are losses of food that would feed more than 600 people, even in our days, where we have good fungicides and where we have a high knowledge on plant diseases, but still there is many things to do. And what is also true is that the importance of fungal diseases can change over time. In our days, the Panama disease comes up again. The septorial leaf blotch in wheat was not a topic before 1970 or 80. The Asian soybean rust was not a problem in soybeans in South America until 2000. Ramularia leaf spot is spreading over the world. Yellow rust now in wheat in Europe. We did not have in Europe Amazonina, which was present, of course, in India and other Asian countries, but now it arrived and makes problems. Black spot, 10 spot, and many, many other diseases. It was also mentioned in one of the reports before that yeast blast, but there are many, many others. And travel trade changes in cropping, new varieties, yeah, overcoming variety resistance, changing in pathogens, climate changes, and others are the reason for this. 2020 is the International Year of Plant Health, and fungicides are still important tools to keep plant healthy, ensure yield, and ensure high quality. There are a lot of fungicides available. This is the poster of the Fungicide Resistance Action Committee. You can find that on the webpage www.frug.info. And this is a summary of all these different fungicides which are available. More than 200 compounds are registered worldwide, but some of these are broadly used by the farmers. Yeah. This is a summary of all fungicides where registered elsewhere in the world and where Frug has knowledge too. So a lot of fungicides, but fungi can counteract, like bacteria, bioresistance, and there are also a lot of publications available because there are a lot of problems with fungicide resistance in the world with different pathogens, Alternaria, Phacophthora, Botrytis. Botrytis is always one of the first ones, but also such pathogens as Septoria, for example, and others, many, many others. I would like to give a short introduction on the most important resistance mechanisms in phytopathogenic fungi. So this is the normal biochemical pathway. We have the target and we have a substrate, and the fungus is happy because everything is fine. The fungus is not happy anymore when we treat him with a fungicide, which blocks, for example, this binding site of the fungicide, and then this biochemical process is inhibited and the fungus is dying or is at least inhibited in his growth or in his development. So, but the fungi can counteract, for example, with a target site mutation. Yeah, I illustrated like here, yeah, that then the substrate is can still bind, but the fungicide not anymore, and that is resistance. Yeah, the fungus is then resistant, and these target site mutations is in phytopathogenic fungi the most important resistance mechanism. We have also something like target site overexpression. You need then more fungicide to inhibit all these target sites, and that leads to higher concentrations you need, to higher EC50 values, yeah, reduced sensitivity. Or the fungicides are metabolized, yeah, or what is also important, in some fungi, not in all, but in some, is an enhanced efflux, yeah, which is normally unspecific. This is a detoxification mechanism of a plant, which could be increased, enhanced, overexpressed. I would like to go in more detail with these two important or some important mode of actions. One important mode of action in fungicides is clearly the respiration inhibitors. Here, the strobilurins belong to, also QII inhibitors belong to, you might know, thiazofamid. And of course, the very important SDH inhibitors, which were recently introduced. And this older class, but with a lot of fungicides, these are the sterol biosynthesis inhibitors. In this group, the demethylation inhibitors are of more importance. I will come back to this. There are many, many other fungicides which are of importance. The multicides, chlorothalonil, copper, sulfur, mangocept, all these things, or the acrylphenylketones, these powdery mildew compounds, for example, metraphenol. I would go first to the respiration inhibitors. And this is the site where the respiration inhibitors are acting. What you see here is the respiration chain here. In the inner mitochondrial membrane, you see the different complexes, complex 1, complex 2, 3, 4, and 5. And we are now looking at the complex 3 because this is the target of the QOIs, you know, maybe better than with the word strobilurins, yeah, and there are also the QIIs here. Why are these so named? Quinone outside inhibitors, QOIs, and quinone inside inhibitors, QIIs, because we have one binding site outside and one binding site inside. Okay. The main resistance mechanisms for this successful class of the strobilurins, for the fungi, is the target site mutation G143A. Nearly all who are acting with fungi know this mutation, because this is a very strong mutation in the cytochrome B, which make up the complex 3 for fungi and strobilurins. This is the reason why many pathogens cannot be controlled anymore by strobilurins. For example, Salmonella triticea or Venturia has problems, even in Japan, Pyricularia, rice blast, and many others. Many pyromildews in grapes, in wheat, and so on. Makes at least this resistance is present in some regions. So, but some species, and that is very interesting, cannot make this G143A mutation because of the genetic structure. What do I mean with that? After the codon 143, there is in some fungi directly an intron, and this intron has to be spliced out through the messenger RNA maturation, and if there is a wild type, then everything is fine, because this codon 143 belongs to the recognition sequence. But if there would be a mutated codon, then the splicing process would be inhibited, and this would be a lethal mutation. And this is very conservative. We call these pathogens the intron pathogens, and these are, for example, all the rusts. So you do not find in any rust so far a G143A mutation, even over 20 years of use. Another resistance mechanism with really minor importance is the alternative oxidase. This is a bypass of the complex 3, is often accompanied with fitness penalties, and as I said, of very minor importance. And the G143A is the resistance mechanism of choice for every fungus. Who can do it? Which one can do it? Here is an example, Septoria leaf blotch in wheat in Europe. Here in France, you see blue dots are fine, sensitive. Red dots means high resistance in a field. 2003, it begins with a strobilurin resistance, the QOI resistance. And in 2004, 1 year later, the efficacy was gone in most of these regions. And here from 2017, we had still a monitoring. All Europe looks not good, and but in the eastern parts, where the wheat production is less intensive, there is still a sensitive situation in many fields. Totally different in wheat brown rust, 2003, everything fine, of course, still in 2008 to 2018. Why? Because this fungus is not able to produce the G143A, and that is good for us, it is bad for him, but it cannot overcome this handicap for many years, as we see. So, this is a timeline of QOIs, of important QOIs. These are the natural compounds, which you can find in the nature, strobilurin A and odomenzin A. The first here, strobilurins were grasoxymethyl and azoxystrobin, then trifloxystrobin, picoxystrobin, paclystropin, fluoxystrobin, and there are several more which are not listed here, were introduced. But then it was stopped because of the G143A mutation. But then a Japanese company made an invention, and this was methyltetraprol. And this methyltetraprol binds at the same site as the QOIs in the outer side, same binding site, but this is active against G143A mutated isolates. Here we see the complex 3. You see here this parallel alpha helices. These indicate that this here is anchored in the membrane. So here is the membrane, and we see here the QO site, and here we see how this methyltetraprol lies in this QO site. Here we have a comparison of strobilurins, of a normal strobilurin. It might be vasoxystrobin or pyridoxystrobin. In the wild type, it fits nice. Also, methyltetrapol fits nice, yeah, in the binding site. But when we have a G143A mutation, that means this glycine is made to an alanine. That means one methyl group more, yeah. And this causes then a steric clash with the normal strobilurins, but not with methyltetrapol. Methyltetrapol is not affected by this mutation. It is also good described by the Japanese researchers here in pest management science. So if you are more interested here, you can find more. And in the last time, there came even up more publications on this. So interesting, an old group, and there was nothing happening over years, but now methyltetrapol, a new compound, yeah, which is acting against resistance. So, and now we have also another complex 3 inhibitor, but in the other side, the QI side. These are not cross-resistance to QOIs. I mean, QIIs are not cross-resistance to QIIs because they bind at different sites. They bind at the same complex, but at different sites. And this new invention was fenpigoxamid. This is by Corteva, by the company Corteva. It has an interesting activity against Thymus septoria, Tridice, and other true fungi, not against oomycetes. And this is also an interesting new invention. Okay, these were my examples for the respiration inhibitors with newly developed fungicides. And now I would like to come to the sterol biosynthesis inhibitors. The sterol biosynthesis inhibitors, as the name say, they inhibit the ergosterol biosynthesis by inhibiting this enzyme here, C14 alpha demethylase, also called CYP51 or ARC11. I prefer the name CYP51, it's shorter to say. So CYP51 is inhibited by the DMIs, the demethylation inhibitors. On the right side, you see a mess of products, yeah, which are copied out of the FRAC poster. A lot of DMIs. You see, it is an old class. It is a very successful class. But why there are so many DMIs? Because the fungi adapted always to the DMIs. And then new DMIs were developed. Yeah, then you find again an adaptation, not a really strong assistance. Yeah, we call this slowly increasing adaptation. We call this shift to a lower sensitivity, a shifting. Yeah. And this sensitivity to DMIs is mainly reduced by target site mutations. But these target site mutations are not so strong like the G143A with the strobilurins. Yeah, there are mutations which have a little effect, and there are a combination of mutations which might then have a little bit a stronger effect. Yeah, and then we have, for example, found in Septoria Tridice. I think this is the one with the highest adaptation to the DMIs, at least what the target site mutations concerns. And there we found more than 100 different CYP51 haplotypes based on all these different mutations and combination of mutations. Yeah, but also other pathogens showed CYP51 mutations. We found now in apple scam, powder mildew, in wheat or so. We found also CYP51 mutations, but not with this many mutations as we did with Thymus septoria Tridice. If you want to know more on that, yeah, it's really an interesting topic. What are the fungi doing to adapt? Yeah, and especially if they are not able to make one strong mutation in the target site, you can have a look on this publication or on several other publications on Thymus septoria Tridice from our group and other groups. Yeah. And the landscape of the DMI is changing. So here, the development of new DMIs are triggered by two things. First, by the toxicity of the compounds, yeah, by unknown effect, unwanted effects, yeah, to humans or to animals. And the other trigger is to have a very good activity, yeah, because of this adaptation to the azoles, yeah. And here we see epoxyconazole. The future is quite uncertain. In Europe, it was phased out already, yeah, and also other triazoles are discussed, yeah, propiconazole, for example. And then there was time to develop a new DMI, and this is called methane-trifluconazole. This is made by BASF. And the registered DNAs are under pressure, as I said, from an efficacy and a regulatory point of view. And therefore, I would like just for explain this. Yeah, we have the desired effect. Yeah, we want to inhibit this target, the CYP51 in the fungus. But we do not want to have any effect here on the CYP19 because we see of mammalians, because we see here sometimes side effects of the DMIs. And to keep this this effect high here, but here low, yeah, this is what's the target of our research. And therefore, we did not only in the development process look for the efficacy here, we looked even more that our products have no efficacy or a very, very low efficacy on this off target, on this mammalian CYP19. So this was really triggered, this development of methane-trifluconazole from a regulatory, but of course also from an efficacy point of view. We call methane-trifluconazole, we call it also Revysol. Yeah, you might remember this word here. This is very complicated. Yeah, it's the first isopropanol azole. Has a good regulatory profile, no classification on cancerogene mutagenic reproduction, no ED, that means endocrine disruption, and it has a high efficacy and nice target selectivity. Also, you can spray it on the crops and you do not see selectivity problems, that mean phytotox, yeah. And this means it combines favorable regulatory profile with superior activity and excellent selectivity. It can make different conformations, and I think that is the secret behind its good activity also on the fungal target. It can have this unbound conformation like you see here, but when it goes in the pocket, it can also form it like here. That means it finds its way to the pocket very nice, and then it is very nice fixed in the pocket. And even if there are some soft target site mutation, it binds very good. So you can see it like this, yeah. And other DMIs are not so flexible, and that might be the reason why with methane-trifluconazole we can now control many shifted strains in a very proper way. This is a picture how methane-trifluconazole binds in the binding pocket. And okay, sorry, yeah. And this is just a greenhouse trial with Septoria tritici, where we infected, yeah, when the plant were young, yeah, we infected these leaves here. Then you need to wait 2 to 3 weeks, rather 3 weeks, until you see the symptoms, because the incubation period is very long for Septoria, and then you have a high growth of new leaves. So we have to focus on these leaves, and you see in untreated the leaves are dead. Here with the first DMI, you see some green parts, you see some efficacy, but it's not so nice. Here methane-trifluconazole, what means Revysol, full controls this isolate, and this DMI not. And this is an isolate which is highly adapted to the most DMIs. But here Revysol has still very good activity. And that is also the reason for the success of this development of this compound. So The development of novel compounds, of novel fungicide is important because of different things. I would like to summarize it here. New regulatory requirements are, and that is good. We always want to have green products, yeah, for ecotox, for human tox, and so on. We have the problem of the development of fungicide resistance in many pathogens towards the COIs, for example, the DMIs, the CAAs. These are oomycete compounds like mandipropamid, dimetamorph, so the SDHIs. These are boscalid, fluxapyroxate, bixafen, and so. And there we have always problems with resistance development in some fungi, in some regions, maybe also worldwide. Yeah. And the pathogen spectrum and the importance of diseases might change also in an unpredictable manner. So new solutions are necessary. Yeah, but we have also to take care that we do not throw away old fungicides very fast, because old fungicides might have a good efficacy on new challenges. Yeah, I have here one example. I could give some more, but one example is the efficacy of the old compound fenpropimorph on soybean rust. And we introduced fenpropimorph in soybean rust some years ago, and it is a valuable tool in soybean rust because soybean rust developed resistance, or at least reduced sensitivity to all the different compounds which are used against some soybean rust. Yeah, not the multicides, but for example, for the COIs, the DIs, and the SDHIs. So there is every tool needed. Yeah. And new fungicides have developed and are under development with new mode of action. For example, this oxatiapipolin. This is a very high effective compound developed by Syngenta on, no, not Syngenta, it was Corteva, on oomycetes. Yeah, this is a really good compound. And but it is also possible to develop new fungicides, as I showed you with the different examples, with an existing mode of action, or with then no or incomplete cross existence. And the examples were fenpicoxamide as a QII, methyltetrapol as a QOI, and mefentriflucolanzol as a DMI. Okay, I think this is my last slide. We have to maintain the efficacy of old and new products by appropriate resistance management. Yeah, we should use all measures first, which reduce disease and therefore selection pressure. So good agricultural practice is mandatory before we think about using fungicides. Then we have to plan our spray programs and consider the use history. We have to limit the number of spray with fungicide with the same mode of action to reduce the selection pressure. We have to have the knowledge about cross resistance of the compounds. We need to alternate with fungicides with different mode of actions. We can combine fungicides with different mode of actions, especially with fungicides with multi-site action. That means we apply a mix of compounds. Yeah. That helps for resistance management, but also it can broaden the disease spectrum. We have to check that we apply at the optimal timing, maybe rather preventive than curative, because when there is a lot of disease on the plant and you come with curative applications, then you have a higher selection pressure because more fungal cells are then under selection pressure. And we have to make resistance monitoring, and we have to know what is going on in your field, and then we have to adjust the spray recommendations with this knowledge. And that is my last slide, blooming orchards. And I would like to thank you for your attention, and I would like to thank my group, yeah, who participated in the preparation of this presentation. Thank you very much.
Thank you, sir. Thank you so much. Thanks for the wonderful presentation. You have series of messages in the chat box, sir, appreciating your presentation. There's a lot of messages from the participants.
Okay.
It will be shared.
Appreciating your presentation, sir. Okay.
Okay.
Benagi sir, now, yes. Now the session is open for discussion. Any questions? Benagi sir? May I have the chairman, please? Okay, sir. Thank you so much, sir. Dr. Jared Samler, thank you for having taken your time and giving us a wonderful presentation. We are very glad to have you today for our international e-conference. Thank you so much.
Thank you very much. Very nice conference. Thank you very much.
Thank you, sir. Thank you.
Thank you, sir.
We'll move on to the next presentation. Dr. H.M. Venkatesh. Sir?
Yes, I'm here.
So good afternoon, sir.
Good afternoon.
I welcome you to the International E-Conference for the session, Recent Molecules in Plant Disease Management.
Thank you very much.
Benagi sir, with the permission of you, I would like to introduce Dr. H.M. Venkatesh. He has 20 years of experience in crop protection and seed health management. He has started his career with Syngenta Seeds. He has worked in various capacities in crop protection, research and development with EI DuPont India Private Limited. He has worked as Asia Pacific Regional Development Manager and has led projects in the field of disease control and seed treatment across Asia Pacific countries. He also worked as Vice President Agro R&D in Pepsi Cola. Presently, he is working with PI Industries, heading Discovery Biology.
Rajeshwari madam.
Mute. Rajeshwari, please unmute.
So it is automatically muted. And
okay.
Hello, sir.
We can hear you. Yeah.
Okay, sir. You can start your presentation, sir.
Can you all see my presentation?
Yes, sir. It is visible. Can you please put it on to slideshow mode, sir?
Now?
Yes, sir. Yes, sir.
Yeah. Good morning, good afternoon, and good evening. I think it's an international conference. People participating from all across the world. Good afternoon to my teachers here, my friends and teachers, especially the chairman, the co-chairman, and the rapporteurs. Thanks for giving me an opportunity to present, the recently developed new mode of actions in fungicides. I guess, Judge Stamler has already introduced the subject very well, and he has explained the global scenario on the disease and resistance and need of a new molecules. I guess, that I feel is a good introduction for my subject, but at the same time, there could be some overlaps. Sorry if there is a repetition, but I'll try to close those slides wherever a little overlap very quickly. Okay, let me move to the next slide. I think, the way pathogens evolve, the fungicides are also evolving over a period of last 5-6 decades. I mean, since two big famines across the world, we have not seen another new famine, thanks to all the fungicides invented and used by the growers to contain those diseases and still supply the food to all of us who live in cities. So if you look at 1940s, 50s, 60s, 70s, 80s, I think the focus was like to bring a broader molecules, broad spectrum molecules, multi-site molecules. Then the use rates were very high. The targets were also to reduce the environmental loading. So as we move forward, the use rates, if you look at the use rates which were 2,500 grams per gram active per hectare, today we are very, very close to 10 to 15 grams or 15 to 30 grams depending on the crop. Means this is a great evolution, and I'm sure going forward all the new molecules which are coming are going to be really more effective at very low dose and less harmful to non-target organisms, and they are more or better or safer to human health and environment. The safety profile of the compounds is increasing continuously, and thanks to all the great efforts done by the industry and also the academia across the world. I'll move to the next slide. Similar to the discovery of new molecules, the regulatory system across the world is also evolving. As there were concerns of the toxicity or non-target effects of the pesticides, so-called fungicides or insecticides, the entire regulatory system went on to evolutionary now, because the new products are tested very rigorously, and now I can say the crop protection products, including fungicide, are among the most regulated substances in the world. Means before it reaches the farmer, the industry has to generate more than 150 human and environmental toxicology studies to make sure that the product which is used is safer for the applicants and also for the consumers and environment. Means the effort to bring a new molecule has increased. Just to give an example, the number of molecules synthesized to bring a new molecule in 1965 was 100 molecules. And then, in 1995, it was 52,000. And today's environment, if you want to bring a new molecule to the marketplace, you need to screen 160,000 compounds. Means, entire biology team, whether it is in vitro, it starts with in vitro, followed by in vivo, then, the field studies, microplot and field. There is a large amount of, work which is done prior to bringing the molecule, thanks to all the, the regulatory environment which is evolving. And also the duration of to launch a product today, you need, from discovery to launch of a product, you need almost 11 years. And the total cost is also like, more than $200 million, depending on the country and the company. So in our age, people spend $260 million to bring a new product. Means, the rest assured, the new products coming into the market, they have much better safety profile than the previously discovered and invented molecules. Some of them are still continued in the toolbox, and some are discontinued. But I'm sure, the combination of all these things will bring down the environmental loading, going forward. Moving on to the next slide. Basically, my lecture starts from here. I think, we have already seen this, FRAC, chart there. FRAC 2020, lists around 11 mode of actions, and it has 230 overall active ingredients in it. Means, so broadly, the mode of action, which is the specific the process of the entire pathway. The pathway is the mode of action. Many times within the same pathway, there are different sites of actions. I think you heard Jared talking about QOI, QII, and within QOI there could be different. Within QOI there could be two different sites. So overall, there are 11 mode of actions and several sites of actions, and both are many times referred as mode of actions. Okay. And as already understood, respiration, because of multiple mode of actions there, it leads from the front. Almost 57 molecules are there. SDHIs, QOIs, QIIs, all these molecules belong there. Some complex 1 molecules are also under research, followed by sterol biosynthesis inhibitor, ergosterol biosynthesis, 49 compounds. So the list goes on. Yeah. I'll take you to the next important inventions in the recent time, which was also mentioned by Jared. Oxothiopipranyl it is also called as Zovec, is the newest mode of action invented and brought to the marketplace. This is an oomycetes molecule invented by DuPont and being sold by multiple companies in multiple names. And depending on the country, this product has been sold from 2016, and certain countries have not received this registration of this molecule yet. But it's a great molecule. Again, I can say in terms of potency, in terms of low use rates, this is achieving climbing Himalayas. So means very very low use rates, and that to a controlling oomycetes diseases with 5 to 10 gram active ingredient per hectare. It was unimaginary few years back, thanks to a great research done by a company like DuPont, which brought this molecule. This is listed in FRAC under group F9, number 49. These oxysterol binding proteins are involved in movement of lipids between the membranes. Hence, it is under lipid synthesis and transport. So inhibiting this oxysterol binding protein may disrupt many other processes as well, such as signal transduction, maintaining the cell membranes, and formation of more complex lipids that are essential for the cell survival. Means at the organism level, it inhibits the mycelial growth, sporangial formation, and zoospore germination. Once it is applied, it protects longer duration of control depending on the dose and the crop and the pathogen, and it is highly effective against most of the oomycetes molecule, especially downy mildews and late blights. But it has less activity or no activity on PTM. Overall, it's a great invention, a new mode of action, and a great addition to resistance management, for especially oomycetes diseases, which are known to develop resistance very quickly, and need of a new mode of action is always very urgent in these cases. And it also revives several other old molecules which are about to develop resistance. So it helps overall resistance management to a great extent. Hence, a new introduction of a new mode of action is a great benefit to for the food security of the world. I'll move to the next invention, which is like the butyrtubulin assembly inhibition, which is also very well known for us. It's a world mode of action. Benzimidazoles, we remember carbendazins, but within this there is a novel site. There is a novel site. It is called as vinblastine site. This vinblastine is a cancer drug, and many companies are working on this site, trying to bring one molecule from this site of action, this mode of action. And I have seen excellent activity from these molecules, and and it is very broad spectrum disease control, and very important diseases like rice blast, zymoseptoria, rust pathogens. Most of those are covered. We know that benzimidazoles are one of the hall of fame mode of action, and also very broad spectrum systemic. Entire fungicide profile really was very good. Hence, this mode of action is really very important. Also because benzimidazoles, because of the resistance and also because of the regulatory concerns, they are moving out of the market. Any new molecule from this site of action which has no cross resistance, new mode of action with the least regulatory concern, if it is brought to marketplace, it is great benefit again for the resistance management and also for the disease management. Right now there are no molecules in the marketplace, but this mode of action has been explored by multiple companies across the world. Different companies have made efforts. They are at different levels. Some have given up, some are still exploring this, hoping to get a new molecule in another 5 years from this mode of action. I'll move to the next slide. This is another important mode of action. I think you saw sterol biosynthesis inhibitors. These are also called as ergosterol biosynthesis inhibitors. And historically, the morpholins and triazoles, these are always very, very popular compounds in this area. It is a long process here. The entire ergosterol biosynthesis, the pathway has several stages, several steps, and at any of these step, any one gene or any one protein or enzyme is inhibited, the process is inhibited. So the ergosterol is a very, very important hormone for the fungi. Hence, this is a very interesting site of action and mode of action. So companies have been researching to discover a new mode of action in this area. They have found a new site which is called as C24 demethylation inhibition, and this mode of action is works at ERG6 gene, where zymosterol is converted into ophicosterol. Even this process is inhibited here, and the ergosterol biosynthesis will not happen, which means it still acts on the ergosterol, but still prevents the ergosterol synthesis, but there is no cross resistance to any other triazoles and morpholins. Another excellent idea and a site of action to research. No compounds yet in the marketplace. It is still under research by multiple companies. Many patents are being published in this area, especially the target pathogens are rust pathogens in soybean and multiple other crops. There are some toxicological concerns. Okay. There is some disturbance in the background. If the organizers can request them to, mute, that will be helpful. Yeah.
Ramchandra sir, Dr. Ramchandra sir, can
yeah. Is a very important new mode of action. Many companies are researching, and any new molecule from this by 2026, 2027 will be a great addition for the resistance management and disease management in total. Okay, I'll go to the next new mode of action, which is under discovery. This is called as HDOC inhibition, or these histone deacetylase inhibitors, HDOC inhibitors is a very, very new invention and a new mode of action invented very recently, and being considered by many other companies as well. So multiple companies are working on this mode of action to bring a new molecule to the marketplace. These enzymes again, are the histone HDOC. There are multiple classes of HDOC in all organisms, and these are responsible, enzymes that remove the acyl groups of amino acid on an histone and allows DNA to rewrap around the histones. By inhibiting this, wrapping of DNA, the transcription continues, which results in abnormalities in the cell, which prevents the pathogen from infecting the plants. Means the organism is killed. This class of compound is called as oxadiazole benzamides, and which have a very good selectivity to HDOC class 2A, which is very important. Otherwise, this class of compounds in human medicines or human pharma, these are used for cancer control. The innovation comes from there. These are very, very safe to the crop. We don't foresee any mode of action related toxicological concerns as of now. Again, the main target pathogens are rust pathogens. Soybean rust offers a big opportunity. Hence, the need is also very high. There is a high focus on the rest pathogens there. You never know, it is started with the rest pathogens. These can also be explored for rest of the pathogens once it is optimized for one target pathogen. Certainly, the broadening of the spectrum will certainly happen for other pathogens as well. Yeah, I'll go to the next mode of action under research. I think this part was again shared by Judd a few minutes back. So many a times we believe that the new mode of action should be very new, otherwise it is not a new mode of action. No, it is not like that. Many a times if you bring a new moiety, like for example here, tetrazolinone moiety for a strobilurin resistance management, especially the G143A, which is a rampant and and very ubiquitous, widespread, and it is killing all the strobilurins. Simple, bringing a new moiety in terms of tetrazolin and tetrazolinone, and the way it binds and the way it changes the position and angle of binding from the central linking ring, the entire steric clash at the site of action is avoided. Means it is lock and key. The resistance is lock and key. When you bring a new key which can fit in spite of having a modification at in the lock, it works, and it is working very well to control G143A mutant Zymoseptoria. And certainly this is a great innovation, very scientific in nature, and we are all, as an industry, we appreciate such effort with bringing physicochemical properties and still making this mode of action lively. So this is another QI molecule with a different thinking, different moiety in the same molecule in pyraclostrobin. So it's a wonderful invention again. Yeah, I'll go to the next invention. Again, the respiration, the QI, it is in the complex 3. The molecules in QIIs, the quinone inside inhibitors, were known, but they were known in only homeicetes. Amisulbrom and cyazofamides were known for the homeicetes control. And then Corteva, right, Dow Chemicals comes out with a picolinamide chemistry and a fermentation chemistry, basis, a product from Japan. Fenpicoxamide comes to the market now. This is a fermented product, followed by they also have a synthetic molecule now. That's another great innovation to reduce the cost, because fermentation process is very expensive. Any molecule coming from fermentation becomes very expensive, and sometimes it is unaffordable. There will be fluoryl picoxamide which will be coming to the marketplace, which is synthetic and which will be affordable again. So basically the point here is the mode of action is known, but it is known in one segment of a diseases, whereas it is not known in another segment of a disease. Another important segment of disease management in septoria. Zymoseptoria is again very nasty pathogen which develops resistance quickly. At the same time, we also have heard about the black sigatoka, which is spreading all across the world and developing resistance. These molecules, both the molecules, are very effective against Zymoseptoria and Mycospirilla, and this is a new mode of action for this segment. Hence, I wanted to bring it here and present to this august audience. I'll go to the next mode of action, which is being invented, discovered. This is called dehydroorotate dehydrogenase inhibitors. In short, it is DHODH. Again, influence is from the pharma, which we had never heard about this kind of a target sites. There is a huge advantage now. We have a lot of nomics which is helping it. This enzyme is involved in pyrimidine biosynthetic pathway, Again, there is a chemistry called quinoline amines. These amines, quinoline amines, are very new in nature, and there are two molecules in this in this series, quinofamilin and iflufenoquin. These two molecules are extremely efficacious against botrytis, the rice blast, and a few other pathogens, alternaria. Means apple scab, especially anthracnose, gray mold, and rice blast. These are very good chemistries. This is a new mode of action, and most probably it is part of the the respiration, or it could be very close to the nucleic acid as well. The grouping will happen later. Again, wonderful innovation. DHO-DH inhibition is a new site of action for the fungicide research. I'll go to the next slide, which is again a new site of action under research, under discovery, under cell wall biosynthesis. This is called as GWT-1 is the site of action. Glucosamyl phosphatidyl acyl transferase is the enzyme, and it is a precursor for the biosynthesis of glycosyl phosphatidyl inositol, GPI. Okay, there is a molecule called aminopyravin, which is inhibitor of this GWT1, which is involved in the biosynthesis of biosynthesis of the GWT1. Okay, it inhibits the mycelial growth. Basically, it is not a spore killer; it is a mycelial killer. Accordingly, the application will happen. Again, the spectrum is like gray mold, scab, anthracnose, and septorias and powdery mildews. Yeah. Not much information is available on this. Only a couple of companies are working on this. Hence, not much. The crystal structure is not available publicly. Hence, there is limited information available on this site of action. There is another interesting, the mode of action which industry is exploring. This is again not very new to the industry. This is called as ACC inhibition, acetyl CoA carboxylase inhibition, which was very popular already in the herbicides. ACC inhibitors are very well known, and Bayer Crop Sciences also brought this mode of action into insecticides and acaricides. Many insecticides, three insecticides, acaricides came into the marketplace, but there was none in the fungicide marketplace. Now, there were multiple target sites within the ACC. So the domain, the BC domain, the biotin carboxylase domain, is the domain where fungicide, new fungicides are inhibiting the BC. They are actually binding in BC domain, which catalyzes the ATP dependent carboxylation of biotin. By inhibition of this domain, it prevents the dimerization of the BC domain. Hence, it kills the pathogen. Means, there is a new opportunity. Again, very broad spectrum in nature. Multiple diseases, including rust and other important pathogens, are targeted by this mode of action. So you can see here, like the mode of actions being considered, which were available but not available in our disease control. Okay, I'll go to the next slide, which is my last slide and the concluding slide. Discovery and development of new mode of action fungicide is a continuous activity for the management of diseases as well as the resistance. And the rapid emergence of the resistance is growing threat for the sustainable agriculture and for the food security of almost 8 billion people. And improved activity and potency of these new fungicides combined with environmental and human safety is a great benefit. Again, and use of new innovations are the tools available like genomics, proteomics, and bioinformatics for the fungicide design is helping us to bring the new mode of action which were not known. With that, I'll thank you for the thank you, Chairman, Co-Chairman, and Deputies for the patient hearing. I conclude my presentation.
Thank you so much, sir. Thanks for your wonderful presentation. You have a series of messages. You have one question, sir, in the chat box.
Sorry, I cannot access chat box because I'm outside
Sir,
now.
I will read for you. I will read for you.
Yeah.
I will read for you, sir. Is there any substitute for carbendazine? The question is there any substitute for carbendazine?
Yeah, carbendazim is a very old molecule. This factor, there is no alternative. China factor is always helping certain molecules to continue because of in spite of their, the regulatory concerns. So this is a broad spectrum molecule. There are many innovations, many new molecules which have come after that within the same mode of action and also from the other mode of actions, depending on what pathogen you are targeting. If you are targeting, certain fusariums in the soil, there could be limited options. If you are targeting any other pathogens, there are many, many alternatives which are better, which are safer, which are very good in nature. Unfortunately, developing world still depends on carbendazim a lot, to control certain diseases. But I'm sure, you always find an alternate compound for carbendazim. If not within the same mode of action, certainly from modes of action, there are multiple molecules available which can do similar job. And which is, again, they are also called as Hall of Fame mode of actions or Hall of Fame chemistries. Yeah.
Sir, we'll take up another one question. Isoxazoline compounds, whether available in India?
Right now, it is under registration process. It would have been available for the Indian farmers if they had decided to sell solo product, because they upgraded the resistance risk from medium risk to high risk. They started developing the combination products later. Hence, the combination products are under registration. The compounds from Syngenta and Cateva should be available sometime soon. Right now, it is not. But certainly 2021, 2022, it should be available for the Indian farmers for grape disease, potato disease, tomato disease control.
So we'll take up only one. There are so many questions in the chat box. We'll take up only one question. So last question. Please suggest any good fungicide for Ambrosia fungi group Ascomycota, which grows inside the tunnel.
Yes, certain these pathogens, difficult to control. There are some old molecules like calaxin. Calaxin is also very well known in palm. I mean, to control these kind of a tunnel growing fungi. Certain strobilurins are also being, tried, but, super systemic molecules like calaxin should do a good job. But, these specific questions should be addressed, by one-to-one discussions. I can give their number and we can work on it. I guess, some of the old and good super systemic molecules are systemic molecules, which are many systemic molecules are available for ascomycetes, and we can explore them. Yeah.
Thank you, sir. There are series of questions in the chat box and a lot of accomplishments also, sir. So we will definitely pass on your...
I'll try to address them.
Due to paucity of time, we will definitely pass on your contact number and email ID to our participants so that they can have you on discussion, sir. And a message from Judge Stambler is very good overview of the information has been passed on by Dr. Judge Stambler, sir, for your presentation. Thank you so much, sir. And for the information to the participants, Dr. H.M. Venkatesh has served for 20 years in different capacities, and he started his career with Syngenta Seeds, and he worked in various capacities in crop protection, research and development with EI DuPont India Private Limited, and he has worked as Asia Pacific Regional Development Manager and has led projects in the field of disease control and seed treatment across Asia Pacific countries. He has also worked as Vice President Agro R&D in PepsiCo. and presently he is working with PI Industries, heading Discovery Biology in crop protection, and is involved in new product development for the global markets. Sir, thank you so much for your enlightened presentation. Thank you, sir. Thank you for being a part of our conference. Thank you.
Very much.
Thank you, sir. So we'll move on to Benagi, sir.
Okay. Very good.
Okay,
Very
sir.
good presentation.
Okay,
Yes,
sir.
yes.
Yes, sir. Thank you very much. Guruji, thank you very much.
Oh, thank you, Nitesh. Very happy to see you. Very wonderful presentation you had. We had not known the different mode of site action. Really, it is very useful for future management of the plant diseases, which are very much complicated in nature now.
Benagi, sir, you can switch on your video, sir.
Video. I'm on. Now I'm back.
Yes, sir.
Yeah.
Okay, so you can stop your presentation. Yes, sir. We can take the next keynote speaker. You can take up the next keynote speaker, Dr. MC Ranganath, sir. Ranganath, sir. Dr. MC Ranganath.
Good afternoon. Good afternoon.
Good afternoon, sir. You're audible. Yes, sir. So you start sharing your slides, sir.
Yeah, I will do.
So I take this opportunity to introduce Dr. MC Ranganath. He is presently the head new product development Indophil Industries Limited, Mumbai. He has served in different capacities as field R&D on product development, marketing manager, product manager, fungicides, rice crop manager, chief technical manager, and currently he is the head who is leading the new product development. And he has total 22 years of experience. So we are happy to have you in this conference. Over to you, sir.
Yeah.
So can you please put the slides onto slideshow mode?
Yeah, I put it.
Slideshow, sir.
Yeah. It is audible, visible.
Yes, sir. You can proceed, sir.
Yeah. Slides are visible?
Slides are visible.
Okay. Echo is there.
Yes,
I don't know.
yes, it is visible.
Hello?
Yeah, there is a little bit of echo. Yeah.
Yeah. Hello?
Hello. Yes, sir. Audible, sir.
Echo is coming. I don't know. Hello?
You can start presenting, sir.
Yeah. Respected chairman and my guru and co-chairman and rapporteurs. I don't know. Some repetition. Echo is coming. Hello? Hello?
Most probably YouTube live stream is near to you.
Sorry?
YouTube live stream is near to you. Please switch it up.
I cannot. Echo is coming. Hello? Hello?
Sir, you have YouTube live next to you on mobile?
YouTube live. No?
Any other systems next to you, sir?
No. The mobile is with me.
So can you please switch off your mobile?
Okay.
Switch off your mobile, sir.
Okay. One. Yeah. Hello. Hello.
Hello, sir.
Still that echo is coming, disturbing a lot of this. So, shall I disconnect and again join once again?
Yes, sir, you can do that.
Yeah, I will do it. I will do it. Sorry, give me 1 minute.
Shankaraba.
Sir.
If you mute everyone except the speaker, then it should be okay.
I guess it was the problem was from Rangnath himself. The moment he is out, the rest there is no echo at all. Let him come back. Let's see what happens.
So we have muted all, sir.
Okay, MC is back now. Can you speak, MC, so that we understand a little bit?
So Rangnath, sir, can you please unmute yourself? So please unmute, sir. Sir, no, sir, you have to unmute the mic.
Hello?
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 Am I audible, sir?
Yeah. Okay, sir? Yeah. Okay.
You can share your slides, sir.
Yeah, I'm doing it. Okay. Yeah, sorry for the inconvenience. Hello? I'm audible?
Yes, sir. Yes, sir,
Yeah.
you're audible. You can proceed,
Yeah.
sir.
Yeah. Now it's clear.
Yes, sir.
So respected chairman and my guru, co-chairman, rapporteurs, scientific community, and industry friends and students. Today I will be taking on the topic changing disease complex scenario and with respect to the crop protection market in India, special reference to India. I skipped this slide already. Just I'm trying to cover up on the global and Indian agriculture importance of agriculture and its importance of agrochemicals, and global and Indian crop protection market, and with a special reference to fungicides. As we all know that one is the population is the biggest problem, especially countries like India and developing countries like especially in Asian countries. When the population is growing, the farmland per capita is coming down. So there is a pressure on the small unit area to produce more. Other side, there's increasing demand for the nutrition. Now there is a few shift, maybe change from a carbohydrates to the proteinaceous diets and the calorie intake also. So overall, the globally the nutritional requirement is increasing, but the per capita land availability is reducing. So here, the technological innovations in agriculture is going to play a significant role, which will help to meet the quality and quality challenges. Just if you can analyze actually overall the crop production market for global in respect to the cropping area, percent crop area. When you look at our Asian countries, we have the 34% contribution of cropping area, wherein 26% contribution to the global crop production market. In other case, you can see the American country like NAFTA and Latin America, where the in NAFTA the cropping area is 16%, where the contribution to the global market is 20%. In case of Latin America, it is cropping area is just 13%, but contribution to the crop production market is around 24%. So here, the Europe, USA, Canada, Brazil contributes more than 60% to the global crop production market, which is having around only the 30% of the cropping area. why I'm stressing is, so continents like Asian and African, if you see the cropping area is 34% and the Middle East is around 19%, wherein the usage of the agrochemicals is very, very low in case of Asian and African Middle East countries. So here there is a lot of scope and opportunity to protect the crop from the diseases, to increase the farming income to the farmer. There are so many ways to increase the farming productivity. Anyway, now the land is limited, but we can, through mechanization, we can increase the farming productivity, and through application of a fertilizer, increase the farming productivity, which are very limited potentials nowadays. Coming to the breeding and the crop productions, which are the ongoing good potential for increasing the crop productivity. And the next upcoming field, the plant biotechnology, is the upcoming field. It has got a vast potential. So here, if you just go back to 40 years in China and India, so we are known for the food insufficiency and hunger. But wherein the 30% of the global population present in these countries, and agrochemicals with other technologies have played a significant role in the increasing the food self-sufficiency in these countries. Yeah. Just you can analyze the what is the
Hello,
just
sir. 1 second, sir.
Yeah.
Hello.
Yeah.
Deepshika. Can you please mute yourself?
Yeah. If you see the just
Okay, sir, you can proceed.
yeah. Thank you. If you analyze the area of some over 30 years, some 50 years, if over the production, but in last 50 years, the global crop area is increased in some developed countries, but overall increase was very, very insignificant. So approximately threefold, where in other case of production, approximately there is a threefold increase in the agricultural production is attributed to increase the productivity through adoption of the modern technologies, like I already discussed, mechanization, fertilizer, agrochemicals, and the biotechnologies. When you come to in India, actually, there is a demand for the overall food will continue to rise as the population is increasing. There is always a demand to increase, produce more productive food in terms of increase the productivity. When you analyze the gaps, actually, from 2000 to 2030, so there is almost 114% increase has to be done in the pulses, 48% has to be done in wheat, and almost 93% we need to increase the productivity in rice, and 156% we need to increase the production kind of fruits, and 94% in case of vegetables. So again, the land is a limited resource, so we cannot expand it. So we have to today's business, today's topic is that management of the diseases through the plant production chemicals. It's going to play a significant role in case, especially in the coming years, maybe another 10-15 in the coming years, in countries like Asian countries are going to play a significant role in increasing the productivities of the produce. Yeah. So here, actually, just I got some reports. So without any crop production, still the crop can grow around 30% yield. 20% yield in case of rice, depending upon, in case of wheat, maybe 50%, in case of corn, maybe 30%, soybean. And through the extra crop production care, we can increase from 20 to 60% in case of rice. And we are limited in rice, wheat, and corn. There is a lot of scope is to increase the productivity through disease management. I'm just to try to give the just a global scenario and a comparison with the Indian market. In case of a global market is around around $50 billion. $50 billion is there. 42% is contribution is coming from the herbicide, and 28% is coming from insecticide, and 27% is coming from the fungicides. Whereas in case of India, the 55% of the contribution is coming from the insecticide, and 25% is coming from the fungicides, and some from the herbicides. What I mean to say is, so especially in India, there's a lot of scope is there to develop these fungicides to produce good quality produce. So in coming days, there's a lot of opportunities to develop fungicides and the herbicides. And if you can look at the distribution of the molecules, if you can see the major product molecules are occupied by the strobilurins groups, which is contributing the major contribution, followed by the triazoles around 18%, and the SDHI chemistries around 11%, which is just to enter, which just started entry in the Indian market. Still in Indian market is strobilurins and the triazoles and dithiocarbamates are ruling this in Indian market, and the rest of the other molecules are contributing not so significant, but which are also very important for the disease management. And if you see, and I try to make again an analysis on this global versus the Indian. Actually, in case of a fungicide, the major contribution is coming from the rice and the fruits and vegetables, followed by the soybean, cereals, maize, and other crops. Actually, so again I'm stressing this. There's a lot of scope is there to improve on the fruits and vegetables and the rice crop in Indian conditions. And industry is also working on to bring out the very safer molecules, molecules and the safer formulations, which should be using for the applicators and the users and the environment. When you see the data, actually, if you can show that, so EC formulations are slowly declining, and which are replacing by the SE formulations, which are very, very safer and water-based formulations. Similarly, in case of you can see the WP formulation, also the WDG formulations. So WP formulations are slowly declining, wherein WDG formulations are again increasing, which are again the safer molecules. So I mean to say that, so a lot of efforts are made to improve the safety with this addition of this formulation, development of the formulations. And the biological crop protection is also emerging out from, especially from last few years. This segment is also coming in globally. It is around $2 billion. India is also started coming up, especially the plantations, and in even the polyols also these applications of the biological started. Among the biologicals, microbials and the macrobials, the predatory insects and the bacteria, fungi, viruses, which are the living organisms, there are some still the technological and regulatory limitations impacting the globalizations actually of these in India also. There are a lot of good technologies, biological technologies are available outside the India and within the India, no issues. But bringing from the outside the India, there is still some regulatory limitations are there actually. And in case of the in India, in case of again, I already told actually, strobilurins are taken a significant in a bigger cake share, followed by the triazoles, then the bendimedazoles, then oomycetes, fungicides, then So dimethomorph, these type of molecules are taken the shares. So almost established 30% market share in Indian and triazoles, 25%, carbendazim around 15% market share, whereas the acids like metalaxyls, these products are 11% market share. If you can see the Indian market is highly scattered market, especially in fungicide. So if you see the top 20 molecules are on the Indian market are the trifloxy plus in terms of the value wise, trifloxy plus tebuconazole, which is on the fruits and vegetables. Then they followed by the mancozeb. Mancozeb is the still the leading and second leading product, and which is accepted and it's available throughout the many parts of the in India, and it's going not more than almost 74 crops in India itself actually. So still the product is in the second importance in the Indian market. Then the followed by the carbendazim plus mancozeb, then the azaxi plus tebuconazole, carbendazim, then the propiconazole, then propineb, hexaconazole, azaxi, copper, like this is going on. These are the top 20 products which are almost contributing to the 65% of the fungicide market. Yeah. When you just dissect our Indian market pathogen wise, actually, it's of more of importance to the even scientific community. Maybe persons who are working on this scientific community was working on these pathogens. When you see this pathogen wise, the foliar spots like leaf spots and blights like anthracnose, alternaria, sarcospora, which is contributing around 38% of the Indian fungicide market, and the followed by this downy mildew and phytophthora, which is around 27%, and powdery mildew fungicide is contributing around 10%, and the magnoforte, that is the rice blast fungicide, contributing around 8%, which is almost it's the 400 in Indian value, and rhizoctonia is around 7% contributions. So which is also around 350 to 375 crores to the on these pathogens. Hello.
Hello. Sir, you're audible, sir. Proceed, sir.
Yeah. Slide stuck up. Hello. I don't know. Slide. Slide movement, please. Hello. Hello.
Okay.
Yeah.
Be cool.
Yeah. No, slide is stuck actually. It's not moving. I don't.
Okay.
Yeah.
No problem.
Yeah. Okay. Yeah. Just a minute. I'm connecting again.
Okay.
Yes, sir.
Yeah.
Yes.
So here there are the changes. You can see in the rice agroclimatic climate scenarios. So there is a changes in the climatic conditions on there. Especially, I've taken the rice crop as the reference crop. The changes in the climatic conditions, changes in the varieties, hybrids, changes in the planting dates, depleting soil health, non-ability of micronutrients, which are all going to lead to causing the disease complexities and the crops. So if you can see that in India, actually in 1945, we have only one disease, brown spot. So later on, 1965, the blast is gone to that bucket. Then in 1975, BLBs, then 85, the sheath light and other tumor diseases added. Now, as of now, there is 11, there are economically important diseases are increasing. So here what I mean to stress is there is a complexity of the diseases are increasing in these crops. Not only in rice, actually, in many crops, you can see the chili crop, maybe you can see the crop like a pomegranate. So there are the complexities are increasing. There are coincidence of the diseases are many times happening. So it is a difficult for a farmer to choose the product. So farmer is looking for the broad spectrum fungicides. So that now the issue is the labor is the issue. Again, the rainy season, monsoon is a rainy season. The frequent rains are the issues. In this situation, the farmers are looking for the broader spectrum molecules. So just I'm trying to give it in the rice actually. This how the spread of the increasing the number of diseases or more. Now, although in the rice now presently the blast, sheath light are important, but there are so many other diseases like a grind discoloration, false smut, brown spot, and in places like Punjab and Haryana, the Bakane diseases are increasing in the rice cropping system in India. Special reference to India actually. So these are on the leaf diseases, and these are on the stem and the grind diseases. These are the just to try to, if you map it actually, we know where it comes to triggering and so on. Okay. So another important one is coming the false smut. If you see it actually, it is coming up in a very bigger way in from last 3-4 years. This product, this disease is increasing, and it is beyond the economic threshold level actually. So farmers have started losing the crop because of this disease. Disease. It is from Kashmir to Kanyakumari. This problem is there, and the sheath rot, grind discoloration again. These are the complexities are increasing. Similar, I discussed already. The chilies, pomegranate complexities are increasing. So in this regard, actually, we bought and when launched the one product, the combination of tricyclazole plus hexaconazole on these crops, rice crop with a dosage of 200 gram to manage the disease complexities in the case of rice and the chilies and pomegranate. And also we have launched the one new more product like thioflutamide, which is again the SDH chemistry, upcoming chemistry in India actually. These are the specialty products for the sheath pet of rice. And in India also a lot of efforts are made to bring out the safer molecules like WDG molecules and SG molecules. These safer molecules which are helpful for the applicators and the environment. Okay. So some of the game changers in Indian agricultures as well as in agrochemical industries. So there are a lot of mobile-based social networking is there. Micro banking, the finance is now easily available. Use of app informations, and there is a lot of virtual communities like a grape association, pomegranate association, and this type of groups are coming up. For them, their bioefficacy is more important than the less PHI product with the software benefits than the product price actually. Knowledge bank on the crops and diseases available and the fingertips. Now farmers started using it, especially horticulture farmers, and now the drone applications are also coming in the light in agriculture. We have to look for the suitability of the formulations, and now the present days, the toxicity and metabolites are the important and safety for looking for the safety formulations as the export trends are increasing from India actually. Coming to I think so. What this slide? Just I want to stress actually the whatever the product, since the lot of scientific community is in the conference, and students are there actually. This is the one thing is very important. Whatever the product may be, if the application is not proper, time of application, method of applications, and the type of sprayers, the product efficacy is gone down by, although if the product efficacy is 100%, if any of these things is not proper, then naturally the efficacy of the product will come down actually. So I mean to say that whatever the product may be the good, if the things are not application, but in India, the application technology from last, I can say that some 10 years, there's a good improvement. Now every day, there is a good improvement. We can able to see the our application technology. Again, we need to, especially for the extension plant pathologists, this is very important to highlight the spraying things. And we should not forget the safety also of these products. Anyway, we are training on the farmers, dealers, on the safety aspects, how to use it. There is a lot of wrong perception about the crop protection chemicals. If you are using at the right time, right doses, right way, it is not going to harm the environment. It's not going to harm the applicators or end users actually. So this is also one of the stressful things we need to. I request the scientific, especially the plant pathology or the extension things. We need to try and stress on this aspect also. And this is the last slide. I think I have some submissions for the scientific community actually. But nowadays we could not able to see the good scientific community on the people who are working on the resistance management. There is a lot of I think previous speakers explained how difficult it is bringing the new molecules, what is the expenditures on the new molecules. We need to have the maintain the products which are having in our hand. So resistance management tools and spraying modules should work at the top schedule level. And one more other thing is that disease forecasting for the existing and the new disease. This is also a very important area. Somebody, it's helpful for the industry and the farmer also. So if you have a robust disease forecasting models, and which are the upcoming diseases, like we are not aware of the rice wheat blast in India actually, but see the last 2-3 years, it is the news started coming. The rice blast is coming in the West Bengal, from Bengal, all those things. We should have this type of a new this one, and drone applications with the viewpoint. What type of formulations are required, dosages, water volumes, and application. Another thing is that drip irrigation is increasing in many in most of the crops nowadays. Even I could able to see in maize crop like maize, farmers started using the drip irrigation. So can you give us plant protection chemicals to drip irrigation? Then what is the dose and what is how we can do it? So this type of things are not properly evaluated still, even for industry also actually. So these are some of a few submissions to the scientific community, the extension plant pathologists, and people who are in the even the students community who are doing PhDs. I think. Thank you very much. Thank you very much for the organizers, especially for Dr. Devappa, Dr. Sangeeta, and Shankarappa for this opportunity. So yeah, I also attended last 2 days sessions that are very good. So thank you very much once again. Thank you.
Thank you, sir. Thank you for your wonderful presentation, sir. So now the discussion is open for, now the session is open for discussion. So there are 2-3 questions in the chat box. I'll just raise it. Which is the best fungicide for rice grain discoloration, sir? The question is, which is the best fungicide for rice grain discoloration?
Okay. Rice, that is now propiconazole. Earlier we are doing it. Now apart from propiconazole, trifloxy is also better. Trifloxy plus that's a nativo, and even product like tricyclazole plus mancozeb. These are also good product on the rice grain discoloration.
So the next question is, For blast disease, tricyclazole is one of the most effective molecule, but in Punjab and Haryana government not recommend tricyclazole in paddy because export of rice. So there is any other molecule to control the blast disease effectively?
I think it is a very big debatable issue. Actually, it will take maybe long time to explain it. In a short term, actually, I can say it's of a more of a PHI and residue related issues. Actually, so this product was not defended in the European market. Anyway, now the data is ready. Actually, we are in the position to defend this product. Once the product is defended, so maybe we don't have any problem for usage in the basmati rice also, tricyclazole. As of now, because of the residues issues, MRL issues, this problem is there. I think maybe then we can able to address that issue. We are already working on that. Yeah.
Sir, thank you so much, sir. There are few couple of questions for which we will be passing on your contact number. Thank you, sir. Thank you so much.
Yeah. Thank you very much. Thank you.
I now thank all the keynote speakers who have given the wonderful presentation. Dr. Benegi, sir, shall we move on to the next invited talk session? Invited talks? Okay. So our next speaker is Dr. Ravi Kumar. Sir, can you please share your slides?
Yes.
Dr. Ravi Kumar will. Yes, sir. Please share your slides, sir. So if you have any difficulty, please let us know, sir. Next to the video. Next to the video icon, there is one share icon. So you need to click on that. Yes, sir.
Okay, madam.
Yes, sir. It is visible. You just put it into slide mode show, sir. Slideshow mode.
Hello?
Hello, sir. It is not visible.
Yeah.
It's. I think you have poor connectivity, sir.
Yeah, connectivity is poor.
So actually, I'm in hospital.
Okay. Okay, can you try after another speaker?
Yeah, sure.
Okay, sir. Thank you. I'll call upon Dr. Suresh Egbote, sir.
Yes.
Sir, you can present your slides. Yes.
Okay.
Suresh Egbote, sir.
Yes.
Please, sir, you can share your slides.
Okay. At the outset, good afternoon, Dr. Chairman, and Organizing Secretary, Dr. Deopa, for giving me an opportunity to share my views. And I also congratulate Dr. Deopa organizing this international e-conference. So I wanted to share my views on the effect of recent molecule a little bit of fruit rot and leaf spot of pomegranate. Of course, what presently the area, of course, in India and Karnataka, next to the Maharashtra, Karnataka is the largest pomegranate growing state. And next to the, of course, the heavy problem in pomegranate means and wilt say that for the new growers and the fresh growers, whatever it will be, up to 3-4 years, the problems are maybe due to the various areas once. And earlier the pathogens were different because we conducted survey and we come to that there are the many pathogens. They are also causing the spots and the rots. Not only the which the regulatory or traditionally we found that Pulpotrichum or Alternaria something else. So in our laboratory we have identified them through molecular characterizations. So this is the one Pulpotrichum, of course, is the pomegranate growing, you have seen, and also Alternaria also. That also we come to know that the Curvularia geniculata is the species we have identified the new one. And also Pathology apsis microspora. Earlier we know also like in case.
Dr. Suresh, please near to mic. Please near to mic. Please come to near to mic.
Yes, identified through molecular identification of the Pulpotrichum gliosporidis, Alternaria alternata, and Curvularia geniculata. Pathology apsis microspora. So we have conducted in vitro evaluation because we have identified the very few species of the Pathology apsis or Curvularia like that. That's why we come to know that. So we tried some contact and combi products and the systemic products. Okay. So of course the Pulpotrichum gliosporidis, we have tried the many strobilins and triazole compounds and combi products also. If you see the combination of the triazole compounds and the combination of the strobilins and the triazole compounds, alternator, sites are systemic fungicides except azoxystrobin and all the systemic fungicides they have better in case of in vitro studies, even control. And, as far as the combi products, it's the carbendazim or the mancozeb or the metloxyl mancozeb, they didn't work. Otherwise, all the combi products, what we tried, they are all very effective in vitros. And against the Curvularia geniculata, none of the, except the mancozeb and propinum, other fungicides, they are not shown very good control in vitro. And likewise, even that, except the tebuconazole and hexaconazole and propiconazole, that worked. And even the combi products, all the combi products, they have given very good control. Okay. And the Pathology apsis microspora blight, none of the contact or contact fungicides. However, systemic fungicides like different systemic fungicides, they have given very good control. Okay. And even the except the metloxyl and mancozeb, all the combi products, they have expressed very good control. And we conducted the field trials of the tricyclozole plus hexaconazole field trials. That is the impression that 1.25 gram per liter water, which has given very good control against Pulpotrichum and Alternaria and the Cercospora, different leaf spots and the fruit rot. And come and also the alternary and flora, come to know that isopyrogen control all the leaf spots and the rot. Strong is also for the very good control of the now. And this is what actually I wanted to share, and I very thankful to Dr. Devappa giving me the opportunity to share my views. Thank you, sir. If you have anything.
Thank you, sir. Thank you for your presentation. Sir, we'll move on to the next speaker. Dr. Ravi Kumar, sir. Yes, sir. You can share your slides.
Suresh, please exit.
Sir, Suresh, sir, you please end your presentation, sir. Ravi Kumar, sir, you can share your slides.
No, sir.
Sir, click on the share button, sir.
1 minute. Share ಆಗ್ತಿಲ್ವಾ?
No, sir. It's not visible. The slides are not visible.
Now, madam?
Wait.
Yeah.
Yes.
Okay?
Yes, sir. It is visible.
Okay. Good afternoon.
Sir, please put on to the slideshow mode, sir.
So, good afternoon, respected chairperson, Maya sir, and delegates and friends. Today I am going to present management of rhizome rot complex disease of ginger in Karnataka. So, ginger is one of the important crop for aromatic rhizomes, and ginger is used for both spice and medicinal property. And India is the largest producer of ginger in the world, and in India, major ginger growing states Kerala, Karnataka, West Bengal, Andhra Pradesh, Orissa, Meghalaya, Mizoram, Arunachal Pradesh, Sikkim, and Himachal Pradesh. So we conducted survey on major ginger growing districts, and Uttar Kannada and Coorgs are highest incidence of the disease, followed by Shivamogga and Haveri, and the less incidence was recorded in Mysore. So based on survey, we isolated the pathogen. Nearly 2016-17, we have visited 66 places. Out of that, we have recorded PTM 20 species, means 20 places, means different type of pathogens were recorded: PTM, Ralstonia, Fusarium, Sclerotium, Meladocine, PTM in combination with Ralstonia, PTM in combination with Fusarium, PTM, Ralstonia, Fusarium, and these are a combination with disease and causes more incidence of the and influence of the cultivar with soil type. So here we have evaluated different genotypes or cultivars. So among the genotypes, Himachal shows little bit resistance compared to the Sirsi local and Rio de Janeiro. So here the Sirsi local is the highest incidence, followed by the Rio de Janeiro. So to know that the phenol content on the effect of the disease. So we have collected the inoculated and healthy samples and subjected for phenol content, and highest phenol content was estimated in Himachal, followed by Varda compared to other genotype. And these Himachal and Varda variety, they are shown moderately resistance to rhizome rot complex disease. Next, estimation of chlorophyll content on different genotypes. Again, the Himachal and Varda shows moderately resistance to disease, and Varda shows highest chlorophyll content, followed by Himachal. So based on that survey work, we have conducted a large-scale experiment in farmers field with 9 treatment, 3 replication, and important diseases of ginger. We all know that soft rot, bacterial wilt, dry root rot, then leaf spot and root knot, and different type of PPM is associated for soft rot, and important symptom, soft rot causes pre and post emergence rot, water soaking lesions near the collar region, whole plants are appear yellow, ultimately rotting and produce a foul smell coming from the affected rhizomes. So dry rot causes Fusarium species, and 2 type of Fusariums are involved: Fusarium oxysporum form species gingerberry and Fusarium solani. So important symptoms of Fusarium. So here mainly the pathogen is soil-borne, invade the rhizome. As soon as pathogen invade the rhizome, yellowing, wilting of the plant, creamy discoloration, and ultimately rotting symptoms. So next, another disease is the sclerotium rot. This disease is appear only at the time of maturity, December to January. Here the yellowing followed by drying. Ultimately on the rhizomes, we can see the whitish mycelial growth. Later on it produces a sclerotial body. Next important disease is the combination with root knot nematode. As usually the pathogen invade the roots and galls on the plant, and those affected plants, they look like chlorotic, stunted growth, and you can see bead-like structure on the root. So very important disease, Ralstonia. Bacterial wilt causes Ralstonia solanacearum, and this disease is more severe in September to October, and almost all wherever the ginger is grown, this disease is very common. So important symptom, the leaves show flaccidity, bronze discoloration, drooping, and those affected plant remain green in color, and the rolling of the leaves, and ultimately the plant show drying or wilting. And if you uproot the affected plant and you press, you can see the bacterial ooze is coming from the affected plant. The symptoms of the soft rot and the bacterial wilt and rotted of the rhizome. So this is the pure culture. Based on that, we have evaluated different antibiotics. Streptocycline followed by K-cycline is found effective compared to other treatment. And this is the pure culture of VTM. So in vitro evaluation of different molecules. So among the chemicals, we found that the carboxyl combination with Thiram, Vitavax power controls 100% inhibition in 0.1, 0.05%, 0.1% and 0.15%, followed by the Luna experience and metloxyl. So next, another important pathogen, Sclerotium rolfsi. So here also we have tried different chemicals for management. So under in vitro condition, 100% inhibition was noticed in again Vitavax power followed by a Luna experience. So in pure culture. So here the integrated management practices. So to know the, you all know that different pathogens are associated. For that we have evaluated different integrated management practices. So here the integrated management showing soil application with the Trichoderma hargenum plus Pseudomonas fluorescens plus neem cake plus pre-sowing rhizome treatment with streptocycline combination with COC for 15 minutes, followed by as and when the diseases appear, go for drenching with bleaching powder, then copper oxychloride, metaloxyl combination with streptocycline, 3 to 4 drenching at 15 to 20 days interval controls and gives highest yield. So here one is avoid the growing lowland area or should not select a soil ill drained area, avoid monocropping, avoid growing of ginger in the field in the solanaceous crops or intercropping with solanaceous crop, deep summer plowing and add bleaching powder before planting 8 to 10 kg per acre. Planting should be done February to March in epidemic area and before storage go for treatment with mancozeb combination with chlorpyrifos and we have to avoid the planting in low laying. So have to go for raised bed so that we can minimize the moisture and selection of healthy rhizomes for planting and pre-sowing application with FOM enriched trichoderma plus Pseudomonas, then pre-sowing soaking rhizomes with streptocycline combination with COC for 15 minutes and drenching with bleaching powder plus metaloxyl, then COC followed by streptocycline found effective. So this is in brief for management of the disease. So this is the you can see the photographs for treatment and this is the overall view of the ginger plot and this is the severely affected ginger plot. Thank you.
Thank you, sir. Thank you for your wonderful presentation. So
Thank
we'll
you,
take up one question from the chat box.
Thank you.
What are the conditions that prevail in Mysore and not in Uttar Kannada, which led to this incidence of diseases in ginger?
Madam, ginger pathogen requires high soil moisture. So if the high soil moisture there, the incidence of the disease is more. That's only the less soil, less rainfall is there, the incidence is less.
Okay, sir. Thank you so much. We'll move on to the oral sessions.
Madam, can I ask a question? Hello.
Yes, sir. Yes, sir. You
Hello.
can ask, sir.
Ravi Kumar, sir. Good afternoon.
Yes.
Actually, we could able to manage most of the ginger diseases except bacterial wilt. Are there any really workable solutions for this? Because most of the farmers are struggling even till today in many states, sir, including Karnataka.
So
Even we have seen in Mysore also a lot of Ralstonia has been reported of late. It's very serious.
So I have taken large scale demonstration even till today. So best is bleaching powder 3 gram per liter of water, combination with COC 3 gram plus half gram K-cycline or streptocycline, 3 to 4 drenching at 15 days interval manages 100%.
Is it preventive or curative, sir?
Preventive, not curative because bacteria oozes. It spreads to even irrigation water also.
Okay. Okay, sir. Thank you.
Thank you.
Okay.
Thank you.
Thank you, sir. Thank you so much. We'll move to the next oral sessions. I request Mr. Mahesh HS.
Yes, ma'am.
Please present.
Ravi Kumar, please exit.
Yeah. 1 minute.
Okay.
Okay, sir. Thank you.
Slides are visible?
Yes, slides are visible. Put it onto slideshow mode.
Yes, ma'am.
Yeah, you
Shall
can
I-
present now, and you have only 10 minutes. You have to stick on to the time, please.
Right, ma'am, right. Very good evening to respected chairman, co-chairman, and dear fellow scientists and student friends. I'm presenting on the title Colloidal Silver Hydrogen Peroxide, a novel tool for management of soil-borne plant pathogen. So,
You're not audible, my sir.
slides are not. That's right. Am I audible?
Yeah, you're audible. You can present now.
But slides are not moving, ma'am.
Try to re-upload.
Slides are visible, ma'am?
Yes, it is visible. You can proceed.
Once again, good evening. Actually, these are the product we, as a pathologist, we are continuously searching either for a resistant source or for the new chemical for the management of plant pathogens. In such case, we find one commercial product is available in the market in the name of Silvax. We evaluated that product against some of the pathogens. So, coming to the introduction, for every year, almost 20 to 40% of crop were lost. We are losing by biotic stress. It may be pathogen or fungus, pathogen or insect or weed. Among the pathogen group, most important ones are soil-borne pathogens. They are very difficult to manage because of their broader upstream as well as they survive longer duration in soil with resistant structures and resistant development against insecticide or fungicide. So presently we are solely dependent on either chemicals or physical methods for management of this soil-borne infection. So most of the farmers are dependent on chemicals. That means formalin or methyl bromide or dazamate, extra other chemicals. They are unstable and also they are phytotoxic to the plant. So with this regards, we are searching for same molecule for soil disinfectant as well as for management of this pathogen. In that, we got one commercial product that's available in the trade name of Alstasian Silvax. It's a combination of silver nanoparticles that are 490 to 510 ppm with hydrogen peroxide. The minimum concentration of hydrogen peroxide is 49.5 to 50%. We evaluated against both fungus as well as bacteria by using poison food technique, and for bacteria we paper disc method. Again, the phytotoxic studies were studied on cotton plants to know the phytotoxic effect on the plant. Later, to check whether the product is actually nano or any other form, we characterize the particle by using particle size analyzer. These are the target pathogens. Initially, we tried for soil-borne pathogens of ginger like Pythium offender metal, Fusarium solani, and as well and the bacterial wilt is caused by Rostoni solanacearum. And with curiosity, we also tried under for citrus canker, that's Xanthomonas axanopodis petaur citrus, as well as coconut blight. these are the two major bacterial diseases in horticulture crops. So these are the results of antifungal efficacy of silver hydrogen peroxide. We actually tried from minimum concentration, that is 100 ppm for against fungus, but we represented the results here from 100,000 ppm. But the minimum concentration required to inhibit 100% inhibition of both the fungus, that is Fusarium solani as well as Pythium apoendermatum, was 5,000 ppm. That means 0.5%. So these are the photographs. Coming to the efficacy of silver hydrogen peroxide against bacterial pathogen, that is antibacterial activity. Here we got good result that in case of Ralstonia solanacearum, we got 19 millimeter zone of inhibition at the rate in the 50 ppm concentration, which is equal to our standard antibiotic, that streptocycline, which are given 18.33 millimeter inhibition zone at 500 ppm. That means this product is having same efficacy at lower concentration as compared to our positive check. Thus, similarly, in case of Xanthomonas punicae as well as Xanthomonas citri, at 100 ppm we got inhibition of 22.67 millimeter, which is on par with our positive check at 500 ppm. So these are the photographs showing the inhibition zone of the different pathogen, that's pomegranate blight and citrus canker, and these are the Ralstonia solanacearum photograph. Coming to the, we also studied whether our effective dose, that means 500 ppm or less than that, it affect any, show any phytotoxicity or not. In that we spot inoculated our product to product on 30 days old cotton leaves. At 0.5% we didn't find any phytotoxicity on cotton leaf, but at 1% concentration we find toxicity on cotton leaf. So for bacteria, we got inhibition at 10 ppm to check whether the product is nano or any something else. For that, we characterize the entire hydrogen peroxide silver hydrogen peroxide particles under particle size analyzer, in which almost 70 to 80% of the product particles are distributed and having size more than 100 nanometer and to 500 nanometer. That means the annual average of the product is 322 nanometers. Coming to the conclusion, the minimum concentration for inhibition of fungus, the silver hydrogen peroxide is 5,000 ppm, that is 0.5%. And the product is showing good antibacterial activity as compared to antifungal activity, because at 100 ppm we are getting good result as similar standard antibiotic at 500 ppm. The effective doses that our 100 to 5,000 ppm was safe for application, and not only for soil borne management, this product can be used for management of soil-borne pathogen, as well as we can include this as one of the component in the integrated management of diseases. And finally, I acknowledge my chairperson, Dr. M.R. Ravi Kumar, and Dr. V.P. Nargu, Emeritus Professor of U.S. Dharwad, and Dr. M.S. Patil, plus Dharwad. Thank you.
Thank you, Mr. Mahesh. Now the session is open for discussion. We can take up 1 or 2 questions, if any. Okay, if there are no questions, we'll move on to the next speaker.
Madam.
With the permission of the chairman.
Yes, good afternoon to everyone. With the permission of the chair, we had a morning left with one keynote speaker in the session, plant microbial interactions and signaling. Now I have the opportunity to welcome Professor Louis A. J. Murr, who is a professor in the Institute of Biological, Environmental and Rural Sciences, Aberystwyth University, UK. Professor, are you there?
Yes, I'm here. Can you hear me?
Yes, sir. Yes, sir. You can start sharing the screen. Meanwhile, I have an opportunity to introduce you to the August gathering and participants participating across the different nations. Professor Louis A. Murr will be speaking on applying of understanding of plant signaling from model pathosystem to real life crop situations. I hope we'll be benefited from this. Today I have an opportunity to introduce Professor Louis A. Murr, who works at the Institute of Biological, Environmental and Rural Science, Aberystwyth University, UK. He is basically a researcher in pathology and working on screening of novel antimicrobial agents for the last so many years. As I could get his biodata from the university website, I was really fascinated to read some of his novel contributions, especially in the field of understanding the genetic and biochemical events associated with plant responses to stress and in identifying sources of tolerance bar resistance. He has worked on the models based on the models of Arabidopsis and also the crops like wheat, oats, tomato, and barley. His major interests are focusing on respiratory tract and identifying drug leads from the natural products, especially to deal with some of the deadly diseases like cancer. He is working on the bioactive natural products, focus on identifying the antimicrobial metabolites against human pathogens such as MSRA and Pseudomonas aeruginosa from invasive plant species and extramural microbial strains. He has published his research. He has got more than 255 research articles to his credit in high impact factor journals, and he has numerous international and national collaborations. To quote a few, funding agencies from Biotechnology and Biological Sciences Research Council, European Union, Royal Society, Wellness Assembly, and AU research funding. So the profile goes like this. So now I have the opportunity to welcome once again Professor Louis A. J. Murr for his fascinating talk on applying models of pathosystem to real life situations. Professor, the floor is yours now. You have 30 minutes time. You can present your keynote speech. Thank you.
Okay. Thank you for that very full introduction. Hopefully you can see the presentation on screen.
Yeah.
Yes, sir, it is visible.
Great. So, as introduced, I have many interests, but I want to focus on really the application of the signaling molecules that we all have spent a lot of time working with to real life situations, particularly with a focus on India. I do work with collaborators in India, and so I'm going to highlight how our understanding of signaling in Arabidopsis or in things like wheat or model species like Brachypodium distachyon can be transferred into a context that means something for people in India who and their major crops. Okay. So this is what I want to do, hopefully in under 30 minutes. I really want to show where we are on the kind of work we do. You've never heard of this very unusual university. It's based in the United Kingdom, and it's based in one of the countries of the United Kingdom called Wales. There they have their own language, and this is called Aberystwyth, and this is the mouth. Aber means mouth of the river Ystwyth. Okay, so I'm going to talk about the work we do briefly, overview of plant defense, and focus on really two interactions that are of interest. One is the work I did with North Bengal in Darjeeling with tea interaction with this very important pathogen called Exobasidium vexans, and the work I did I'm doing with RGCB in Trivandrum, which is looking at black pepper interactions with the oomycete Phytophthora capsizi. I don't have time to talk about sesame, But some of the everything I've talked about with tea and black pepper also applies with sesame in different types of interactions, alternaria and fusarium. Okay, so this is where we are. Okay, this is the United Kingdom. Northern Ireland is there. This is Wales, England, Scotland, and Aberystwyth is by the sea. Okay, so there is the river Iswyth, hence its name, and we're by the sea, and we're an enormous city of 12,000 people. Very small, very small. But we're the oldest university in Wales. And this is where we were born, looking very much like Harry Potter and Hogwarts. Okay, so that's where we were born, and we're the oldest university in Wales. So we are a BBSRC institute. So we're like RGCB. We have DBT funding. And so we do a lot of work on improving forage grasses, oats as well. For instance, this is one project we is very successful. This is a forage grass where we increase the sugar and protein content of the grasses by traditional breeding to improve the diet of sheep. In Wales, there are 3 million people, 12 million sheep. So we have lots of sheep. And so we do a lot of work on engineering the plants and also the microbes in the rumen of the animals to improve the diet and digestibility of sheep and cows as well. So because we have a lot of infrastructure, we can do things like automated phenotyping. So we host the National Plant Phenomics Center. So this is a way of actually phenotyping a diverse population for size differences, flowering time differences, drought tolerance differences. So our breeding populations are on trolleys like this. They go round and round and round. And there are imaging stations. They go round and are imaged. And they're imaged for fluorescence data, visible light differences, near infrared, and we generate computerized models of the plants showing how well they are growing under drought, salt, infections. So this is the kind of work we can do. Work that I do a lot more is more at a molecular level. So we have a lot of equipment here that I will do metabolite profiling, so that in say 200 microliters of sample, we can measure 20,000 metabolites in 2 minutes, and we do this in high throughput. So we have all this equipment here for metabolomics or lipidomics, or also some gel-free proteomics. So we do this to match genetic variation with phenotypic variation and also metabolite and protein variation in high throughput mode. So this is the kind of thing you know already. I'm going to talk about defense. And so in particular, I want to remind you of these are pathogens. These are pathogens and they're recognized by resistance genes. These are leucine-rich repeats. You can see there, nuclear binding sites, and they are recognized effectors or PAMPs, pathogen-associated molecular patterns, to generate reactive oxygen species, salicylic acid, nitric oxide, and things like salicylic acid and nitric oxide can contribute towards systemic resistance. We all know that. I'm sure it's featured many times in this conference, but a reminder that we have receptors that recognize effectors delivered by the bacteria, in this case, or fungus, or oomycete, and also molecular patterns on the pathogen, which all feed into a defense response and in some cases leading to programmed cell death. Now, what I did a lot of work on was looking at how these different signal patterns are actually coordinated. So all the recognition I've talked about is here, pathogen detection. And now I've got some jokey images here. Here we've got a biotroph. These are pathogens that live on living organisms. And here we have a necrotroph, which are pathogens that live on dead tissues. So I've done a lot of work on salicylic acid and how it actually interacts to generate resistance to biotrophs. Done a lot of work on jasminates and ethylene and how it interacts with to control necrotrophic resistance. And what I remind people of, and I'm going to not talk about it here too much, certainly not this. I do a lot of work on nitric oxide and how it actually generates increased resistance via salicylic acid, jasmonic acid, and ethylene. So it's very much a driver of both forms of resistance. This is in green. In red is ABA, abscisic acid, which is a suppressor usually of defenses in Arabidopsis, usually. And I think this is the most important point I want you to remember from these two slides. Okay, so salicylic acid, jasmonic acid, ABA. So to talk a little bit about work that's relevant for India, so probably about 10 years ago, I started working with North Bengal University in Darjeeling area. And we were looking at diseases that infect tea, Camellia sinensis, particularly in the Darjeeling area there. And I obviously went there and saw these people picking and saw the pathogens in situ. And the ones we focused on first time was basically tea blister disease, which is Exobasidium vescans. And this is one of the phenotypes. Okay, it's a Basidium mycete. I can see here the sporulation from the lower leaves. So it's a major disease affecting the worst tissue from the point of view of harvesting, which is the tender leaves at the top. So there's no real fungicides that do an effective job of controlling the disease because there's a lot of resistance out there. But there is some native resistance that's associated with things like phytoalexins, polyphenols, catechins, and things like methylxanthines. Okay, and what I joined was that there was biological control experiments underway in North Bengal, looking particularly at Pseudomonas as a biocontrol organism from the rhizosphere. So to actually understand, because I like to understand the situation, what we thought we'd do is we'd start to understand what are the chemical changes associated with disease development. Okay, and we did this from the field. So what happened was that we went out into the field and picked leaves that had different stages of development. So we did an artificial time course, if you want. So this is how the disease progresses from healthy to phenolic based to the classical Exobasidium sporulation events there to necrosis. So these samples were picked, and it's effectively a time course. Okay, so these were picked, sent to me as stages, many replicates from the field, and we analyzed what were the chemical changes that were going on with a view to trying to understand what was the basis of the disease development. And so this is something that you may actually be familiar with. It's like a principal component analysis. And so these are the stages. Hopefully you can, this is stage 1, stage 2, stage 3. So when we looked at that and we saw these are the healthy stages, okay, and then when we got stage 2 and stage 3, they were already very different. There's a big change already. And then as it progressed to necrosis, again, they were very different. There were different stages. These individual names like S2H were individual samples from the field. And you can see they're grouped together. Stage 2 and stage 3 were very similar, which is quite surprising when you see the phenotypes. But we were very interested to know what was going on very early on this change here between healthy and infected material, because that is when disease is being established. So in this two-way comparison, we just looked at healthy H versus stage 1s. And what we did was we tried to find out, they're clearly different. What were the chemical changes that were occurring between those blocks of tissue, those types of tissue? And so we looked at the sources of variation, and probably unsurprising, we found the major changes were associated with the catechin group. Okay, so we looked and identified and measured directly all of the catechins. Okay, so, okay, I'm getting some noise on the line. Anyway, I'll carry on. So here we got some healthies. These are just looking at the catechins. See the healthy groups, these are 90 and 95% confidence intervals. There's the mean for the group in the middle. So just looking at the catechins, we were looking at a difference between the healthy and infected. But see here, along the catechins, we've got major changes in caffeine. But a lot of the catechins were actually going down. We looked at the phenolics. All of these are major defense hormones or defense compounds. These were often going down. You can see a lot of the quercetins were going down. Again, look at them all together. Those are the control groups, and these are the other groups which are infected. So these are all going down. So we're looking at caffeine going up, but caffeine is not associated with defense as a major compound. These other ones which are associated with defense were going down, including some flavonoids, which are very important, some important ones being quercetins. That's okay, but the breakthrough came when we looked at what was happening with the hormones. Now, this is not a PCA. This is just a biplot where I'm looking at salicylic acid levels and jasmonate levels. Okay, and so there is, see, in the healthy material, in the absence of infection, as far as we can tell, there's a fair spread of high JA and high SA levels. Also true of stage 1. But as you progress, particularly say stage 3, where you've got this very good sporulation going on, what you find is basically there's a collapse in the salicylic acid levels and the jasmonate levels. Okay, so this is being targeted. So it looks like disease progression is associated with the suppression of defense hormones. So what is going on in North Bengal now is a whole range of rhizosphere organisms, and they're all being assessed for their ability, there's trichoderma or pseudomonas, to actually increase both SA and JA to counter the suppressive effects of the Exobacillium infection. There, obviously, you're well aware is that there are other people are trying direct applications, say, for instance, SA and salicylic acid or benzothiotriazole, all these things, but there tends to be a yield penalty or a loss in the quality of the tea. So the ones that are being most actively looked at are these biocontrol organisms. Okay, so that's one example. The next example is much more in depth. And this is the one that was done with the Rajiv Gandhi Centre for Biotechnology in Trivandrum. It's been funded from the Commonwealth, okay, and involved exchange of personnel. I went to Trivandrum a couple of times, and Divya Katupalli came to work in my lab for nearly 1.5 years. And so you can see here, I'm sure you know this, black pepper. It's a very important crop in southern India. Very valuable. You can see it's called black gold. But the problem is that quick wilt disease is a filamentous oomycete caused by Phytophthora capsicii. It's basically can devastate the crop, and there is no really good cultivar which suppresses disease development. So we're in the same position as the tea. Okay, so we together decided to involve an omic analysis involving transcriptomics as well as metabolomics to try and work out what is the feature of the disease which leads to perhaps suppression of defenses. So here we go. So first thing we did was look at RNA-seq data. Okay, and we looked at RNA-seq data over 48 hours, and also we looked at local responsive RNA-seq, but metabolomically we looked at local and systemic. I'm going to talk only about the local here. Okay, so we did the RNA-seq, we looked at differential expression of the genes, and we did gene ontologies and looked at what was that showing us, and we did a lot of obviously effects that you can see here, a lot of it associated with, say, for instance, growth, cellular growth. That was one of the major changes. Infected versus control, that's the top graph there, and this is the fold change down below. But when we looked at these fold changes, you can see here there's carbon utilization differences, very interesting there in terms of fold change, but the level was very, very low. So we've got a high level of expression or highish level expression with a good fold difference. Unsurprisingly, we got the defenses. Okay, so we decided to look at those in more detail. And this is a snapshot. You can see it's from KEGG. And we looked at the genes that are differentially expressed directly. And we started to see a preponderance of nuclear binding site leucine-rich repeats, which are the receptors for both PAMPs, pathogen-associated molecular patterns, and also resistance genes, which recognize effectors. And these were basically changing, and we noticed they were going down. So these are all receptors. I put them in pink. Okay, and these are the serine 3 kinases, which are usually outputs of recognition. So they were also being modified by expression. And you can see here things I mentioned, reactive oxygen species, nitric oxide, all these outputs were all being affected. Okay, so of course, those are RNA-seq data, and these are the kind of genes we got out. To remind you, we've got various classes, nucleotide binding sites, leucine-rich repeats. We have TER, toll-like receptors. We have coil domains. All of these we found and classified from within the transcriptome of black pepper. Okay. Same with the serine 3 kinase. There are many of them. Now, the ones I've highlighted here are the different classes. Some of them you may be working on them. And these was a very predominant class. These are the genes. We've got this IRAC class, this is very interesting because this is induced by a PAMPs. MAP kinases down there and various other types. The reason I've highlighted certain genes here in particular is that we use these as representative of the whole class when we did qPCR. Okay, so we actually confirmed the expression of qPCR. Okay, so we've got this. Now, what we did with the coil and nucleotide and TERD class is we managed to get some primers which we could actually report everything that was going on in with that class. Okay, so there's lots of information here, but let's circle this. These are the controls. Those are 24-hour infected, 48-hour infected. So you can see here, this class of resistance gene, the genes are being shut down. They're shut down. This is all roots. Don't worry about this. This is what's in the leaves, which are being infected. Here again, with a coiled class of resistance genes, that was going down. Again. Now look at this one. These are the IRAC serine 3 kinase domains. Looking at 2 examples. Controls, 24 hours, 48 hours in the infected leaf tissue. Down it goes. Suppressed. The other example, suppressed. Over here, for another class, the MAP3 kinases, here there's no difference at all. No difference at all. Maybe a little bit there. You can see that's going down a little bit, but compared to this is rapidly being suppressed. Okay, so we got that, and please just remember this because I'll come back to it. Now, of course, what we did was as well is looked at metabolite profiling. Lots of information going on here. And so what you see in front of you here is the KEGG map. So every one of these dots is a metabolite. All of those which are highlighted are differentially accumulating when we have an infection of quick wilt or Phytophthora capsicii. So you can see here a linolate, that this is a jasmonate pathway effects. You can see here the nucleotides are being this is nucleotides pathway. Over here, the nucleotides are being massively affected. This core here, you can probably make out the names. This is TCA cycle. So fumarate, you can see here, we've got here pyruvate, PEP above here. And here we have the amino acid pathways, all of which are being affected, including things like glutamate and glutamine, which are indicative of assimilation differences. What we're seeing here is the assimilates, particularly being moved out of the host into the pathogen. So that's very descriptive. but we have to link this through to the transcriptome data. So equally important, and that's why I actually highlighted it, is we looked at what was happening in the hormones. So this is control 24 hours, 48, ACC, which is our surrogate for ethylene, salicylic acid, ABA. And what we notice here is that these are box and whiskers, that, yeah, salicylic acid was changing, but at 48 hours. But ABA, which you remember from a few slides ago was a suppressive event, was rising very quickly, very quickly. Okay, so ABA was a very quick responder. Jasmonates and linoleic acid, they're changing, but they're quite slow, and ABA was the rapid effect. So you can see here the metabolite profiling data suggested that ABA was an important variable. Okay, so let's link the two together. So we can do that by looking at the transcriptome data. And what we did was try to confirm the ABA effect in the transcriptome by looking at the receptor gene expression. So this is qPCR, and these are two receptors for ABA. And look, you can see here, they are the controls. Within 6 hours, 12 hours, 24 hours, very rapidly, we're getting a rapid response to ABA. It goes down, through the receptor for going down. And this is indicative of a ABA response that's very, very rapid. So we've said, okay, let's apply ABA to the infection. So you can see these are the normal symptoms for controls. ABA sprayed. Look, this is the same. This is the infection site. There's the infection site. Here we're putting a very low amount of ABA, about 50 micromolar. And look at the disease. The disease has gone everywhere in the leaf, and notice how there's chlorosis, and this is indicative of altered nitrate assimilation. And that's because the pathogen is assimilating the nutrients. Okay, so now we can see this is the microscopic area, this is the infection point, and this kind of area here is where the pathogen has received or gone to. That is the infection site, there's a little wound there. But here, look, this is the spread of the fungus, and this actually is nowhere near the infection site. So you can see it spread everywhere, very advanced spreading of the, excuse me, the oomycete. Sorry, there's the infected one, and there's the oomycete ABA treated. My apologies, that's my error. That's the control on infected. This is the one spreading everywhere, and this net here is from over here. It's spreading very, very far. My apologies for that error. So ABA is basically really important for progressing the disease. Now, what about salicylic acid? It's a delayed response, and it's too delayed. But why not prevent or preempt this by applying salicylic acid directly to the leaf? And look straight away, it should look like that, but there you can see only a few pinpricks. Okay, so we look at the site of infection, you get a little bit of fungal development, probably a little bit like that. But what you get a lot of is this cell wall changes. And this cell wall changes tell me that there's reinforcement of the cell walls leading to increased resistance. So again, salicylic acid or rhizosphere bacteria could be used to suppress this oomycete. Now let's complete the logical flow. Because what we did was we actually said, okay, what's the effect of ABA application on the nuclear binding sites? Okay. So this is the relative expression. One is the control. One is control. So in the absence of infection, basically, if we apply ABA, we can see the IRAC goes down. IRACs, MAP kinases are not very affected. Remember that one was a bit different. Serine kinase going down. This neck is massively induced. Now, this is a kinase we know to be induced by ABA, so that is a nice control. But your nucleotide, your resistance genes, TER class and the CCL class, are also going down slightly. So ABA by itself can mimic a lot of the effects of infection. And when we did a correlation analysis here, I mean, there's a lot of information here, so I'll just summarize it. In the correlation analysis, we looked at ABA effects and those of the natural infection, you've got a strong correlation. You can see there, that's a ABA at 24 hours, strong correlation with ABA treatment and infected at 24 and also 48 hours. And that's what that little red green box shows. Okay. So a lot of information. I've actually tried to show you how very basic understanding of salicylic acid, jasminic acid, ABA can be applied usually to important crops using omic approaches to give new insights and guide what could be markers for showing the efficacy of a rhizosphere approach. To a certain extent, you're thinking, Lewis, I already know this. You've just spent a lot of money proving what I already know. But by having these molecular markers, you can then screen more effectively in the lab a whole range of elicitors, rhizosphere bacteria, and show efficacy. Okay, so that is basically what I'm hopefully going to leave you with. But the last point was beware the cost of resistance. Of course, I do lots of work. So these are the group. Here's Divya, okay, who came over. Lots of people from around the world. I have a very international group. And I work with NIPGR, RGCB, obviously some with Pakistan, which I've not done here. Hopefully I've not overrun too much. And if there are any questions, I'm very happy to deal with them, even with my English accent. I'm sorry.
Thank you, sir. Thank you for your wonderful presentation.
Thank you,
There
Professor,
are few
for your
questions.
excellent. Madam, 1 minute. Thank you, Professor, for your excellent presentation, taking into the examples of two Indian most important, economically important diseases. One is blister blight of tea, and another one is Phytophthora foot rot of black pepper. And these two diseases are of national importance for the last so many years. And we have really set it to the insight into understanding of the phytopathogen interactions, the molecular basis of the effectors, the PAMs, the MAMs, that we can really go into. The last slide that what you said is really apt, because I worked on Phytophthora foot rot of black pepper for my PhD program 25 years ago. And there was a recommendation of use of the rhizosphere microflora for the management of the Phytophthora foot rot of black pepper, including the trichoderma harginum, which is a millennium microbe. I feel your talk has really opened an understanding of how the pathogen behaves at different levels of infection and what are the effectors that really matter in understanding the molecular basis of the plant microbial interactions and signaling pathway. Thank you very much for your wonderful presentation. We have a very good number of appreciations in the chat box and also a couple of questions. I will read one of the question. Will ABA and salicylic acid have same effect on the Phytophthora cassia bell pepper?
So we have done some limited assessments in other species, and it seems to have similar effects. But we haven't done the molecular biology yet.
Okay. Anybody is having any question? We can take a quick 1 or 2 questions. I have a clarification. What are the reasons for sudden rise in ABA?
So what we think is that what some of the effectors that are delivered by the Phytophthora will trigger the ABA synthesis in the host.
Is it going to vary with the susceptible host and the resistant host?
The resistant host, there haven't got a resistant host of that species. So we just got a range of susceptibilities.
Okay, thank you. So if there are no questions, I profusely thank Professor Louis Gamer for his excellent presentation highlighting the Indian context, the work. Manjunath, you have some questions?
No, sir. Actually, Louis is our good friend, collaborator. Just I said hi.
Okay, no problem. Very good. Very nice. So we have there is one more Somnath. Morning we left in the morning session. Somnath, now you are ready. Now you can give your presentation on Somnath. Dr. Somnath.
Hello. Yes, sir. Hello.
Now you can share your slides and start your presentation on transcriptome analysis of heterodox reveals its adaptation to parasitize the monocot host. Morning we could not have. Now you can present.
So thank you, sir, for the introduction. And I supposed to say good morning, but now I will say good evening to all respected researchers. So my topic of today's presentation is transcriptome analysis for heterodox reveals its adaptation to parasitize monocot plant. And this is the outline of presentation. I will start with introduction and in vitro life cycle of heterodox which I will introduce after 2 slides. Then followed by transcriptome of heterodox and its adaptation to parasitize monocot host, followed by conclusion and acknowledgement. So to start with the presentation, I guess most of the researchers now knows that what are the nematodes, what are the plant parasite nematodes, what are the free living nematodes. But I just want to highlight here is till today more than 25,000 nematode species has been discovered, but only 25% are of human concern, that they infect the human beings and their belongings like animals and plant species. And even though if they are microscopic animals, they have most of the organic all the systems except the respiratory and circulatory system. So here I will mention some of the important human parasite nematodes. The first photo which I show is the disease called elephantiasis or filariasis, which is also caused by a nematode called Wucheria bancrofti. Then followed by the second photo I'm showing here is called onchocerrosis or river blindness, which is very prevalent in West Africa. And the third photo which I showed here is the giant intestinal worm, which is also called as Ascaris lumbricoides, which is quite prevalent under a low sanitation condition, and we can, due to these nematodes, central government has started National Deworming Day on 10th of February and 10th of August. Coming to the plant parasite nematodes, even 4,100 species of plant parasite nematodes discovered by now. But among them, most economic important nematodes are root knot nematode. Here I'm showing 2 photos. Left one is showing root of pomegranate infected by root knot nematode Melodogyne incognita, and another photo is my right side showing tomato roots heavily galled due to Melodogyne incognita. And the second most important nematode are cyst nematodes. On the left side we can see the field of potato infested by the cyst nematode, and on the right side we can see the roots of the potato plant infested by yellow and brownish females, and both of these nematodes are endoparasites. So now comes to the heterodox, the nematode which I worked. It's called a sugarcane cyst nematode, and it's primarily infests sugarcane and rice crop. And it's compared to cyst nematode, its reproduction is very unusual because it reproduces by mitotic pathogenesis like root knot nematode. And nowadays it is coming as an emerging pest of upland rice along with the root knot nematode, causing yield losses up to 50%. And due to its presence, it aggravates the effect of drought and other stresses in rice. So before going to transcriptome, we need to establish the system where we can go from soil to plates or in vitro culture. So after lots of efforts, we were finally able to establish an in vitro culture of this nematode heterodox under petri plate condition on the gel called pluronic gel, and we found that this nematode can complete its life cycle within a span of 7 to 9 days, and these are the different life cycle stages we were observed, and this was published in Nematology last year. So after that, after this in vitro completion, we choose 2 stages. First is second stage juvenile, that is pre-parasitic, just hatched juveniles, and second one is 15 days post-infective female, that is young female, for transcriptome analysis. I will not go into detail how we made a transcriptome, but after all the initial cutting and procedure, we could pull the transcript, and we found that 44,230 transcripts with the Illumina MiSeq technology. And from that, we need to search for the putative effectors. I guess a lot of previous speakers also told that about effectors. Just one sentence, the effector is nothing but any molecule which is secreted by pathogen inside the plant, which manipulates its host for the benefit of the pathogen. So from the transcriptome, we looked for the candidate effectors by checking presence of signal peptide and absence of transmembrane domain. And after this initial screening, we compare our transcriptome data with previously published root knot nematode, Archaeanis root knot, and cis nematode transcripts, and we targeted 3 groups of effectors. First one is HGT. HGT is horizontal gene transfer event. Second one is Sprycek events, which are important during the host's defense suppression. And last one is CLE, that is clavata. So I will touch first and last group, which are today's of concern. So when we checked the transcriptome of Heterodactyly, we compared this one with another cis nematode from potato, that is Globura rostokiensis, which is golden nematode of potato. And we found that there was sufficient similarity between the effectors molecule identified from the Globura rostokiensis and that have copy in it in case of Heterodactyly. But if you look at the histogram of all gene versus effector, you can see there are very less similarity, or in other words, you can say effector molecules which are from the Heterodactyly accumulated more mutations rather than the housekeeping or consort genes. And this reflects the need for co-evaluation from the respective hosts, because Globura rostokiensis is a pathogen of potato and Heterodactyly is a pathogen of monocots. In order to confirm that one, we also checked the cell wall degrading and modifying proteins from our nematode, that is Heterodactyly, and to our surprise, we found that the Heterodactyly, that is sugarcane cis nematode, and other its relative, that is Heterodactyly, that is cereal cis nematode of wheat, do not have this type of family transcript from this family, that is GS53, and the other cis nematode like Heterodactylia, the sugar beet, the potato nematode, 2 potato nematodes, they were reported to have 1 or more copies of this GS53. So we explored more literature, and we found that this GS53 is nothing but endo-beta-1,4-galactanase, which metabolizes galactan and arabidogalactans from the plant cell wall. And these monocot roots contain very less pectic polysaccharides and no arabidogalactan in their primary cell wall, and that is the probable reason that these 2 nematodes lost or deleted this family of effectors. In order to confirm that our observation, we did core 96 core eukaryotic genome with 18 species, and this analysis showed us that Heterodactyly and Heterodactyly, both monocot pathogens, are clearly assembled together, and they are nested within the other cis nematode species, showing they are having this other cis nematode species having GS53 enzyme, and Heterodactyly and Heterodactyly, in both the cases, GS53, that is this GS53 family of enzyme is absent. And in order to, we found that there are other plant peptides which are being mimicked, secreted and mimicked by this nematode. One of these are the CLEs, or endosperm surrounding region. And this photograph or this image shows that at the left side there is signal peptide, N-terminal signal peptide, followed by canonical CLE domain at the C terminus. And in our transcriptome data set, we found 6 transcripts which were formed in 2 groups among the transcriptome data set based on their single amino acid changes. And this was our observation. In order to confirm that, we created all by all similarity matrix, taking all the published CLEs from 20 plants, including rice, Arabidopsis, Populus, and other nematodes like Heterodactylus, Globura rostokiensis, Heterodactyly, and Rotolunculus reniformis, in order to know that whether the Heterodactyly CLEs really mimic the rice CLEs. And in this image, you can see the orange dots are represented by Heterodactyly CLEs, while the green ones are the CLEs from Oryza sativa, that is rice. And you can see clearly that first 3 CLEs from Heterodactyly well connected to the rice CLEs. They are the group of 14 CLEs, 8 from rice plant. and another one CLE from the another 3 CLEs from the heterosaccharide, which are also connected with the rice CLEs. And we concluded from this image that the heterosaccharide CLEs are more similar or identical to the rice CLEs, and there are no other nematode CLEs which are as similar as rice than heterosaccharide. And in order to confirm that, we did the in vitro experiment in order to prove that we artificially synthesized heterosaccharide CLEs which were strongly have a strong similarity with the rice CLEs. And we were knowing from the previous publication that this overexpression or overdose of this CLE produce short root phenotype. And we took a control of heterosaccharide scrambled CLEs and heteroglycinous CLEs which infect the soybean and not related to rice, and we could clearly see that in case of the plants were supplied with the heterosaccharide artificially synthesized CLEs, we can found that short root phenotype, and these were experimented repeated like 25 times in order to confirm that. And from these two above experiments, we could conclude that GS53 enzyme has been lost in the monocot pathogen, which is heterosaccharide, and heteroaveni, which is cereal cyst nematode. Heterosaccharide CLEs are more similar to rice CLEs, and they are adapted for function in rice plant because it's a specific host, because its host range is very less. And the effector co-evolution that occurred from the transition of dicotyledonous parasitic nematode to monocotyledonous parasitic nematode, especially true in case of heteroaveni, which is cereal cyst nematode, and heterosaccharide, which is the sugarcane cyst nematode. And I did this work at the University of Bonn, Germany, and James Hutton Institute, Dundee, with the help of ICR International Fellowship and National Rice Research Institute, Odisha. And I also thank NRC Pomegranate Solapur for permitting me to present my previous work. And last but not in the least, I thank the co-organizers for giving me opportunity, which I missed in the morning. And thank you one and all. I will be happy to answer one or two questions. Thank you.
Thank you, Dr. Somnath, for your excellent presentation on the transcriptome analysis of heterogeneous infecting and adaptation to parasitize the other monocot horse. Excellent presentation with slides and very good work. One or two questions, quick questions. From anybody, if there are one or two quick questions, we can take. Otherwise, I have a clarification. Dr. Somnath, did you do cross-inoculation studies in this case?
Yes, sir. We used cross inoculation methods where the lab which I was working, they are working at Shaktiya, and we try to infect the Shaktiya in case of rice and heterosaccharide in case of Arabidopsis, but the cross inoculation didn't mention. And there are several mechanisms. Most important mechanism is non-host resistance, because I see.
What was the extent of infection rate? Cross inoculation studies?
No, there was no infection, but I checked.
There was no cross inoculation.
Yes, there was infection process started, but nematodes were dead just after half.
So, what is your opinion? These nematodes, they co-evolved? That what you want to say?
Yes, sir. These nematodes are special, very much specialized, in my opinion, and they evolve to parasitize the host. As host is getting resistance, these are also evolving to counteract the resistance and starts infecting, make it susceptible again.
Okay. Thank you. You can stop sharing your screen.
Thank you, sir. Thank you.
Thank you for excellent presentation. Now we have the last presentation in the previous session. I hope Dr. Benigi sir is there, the chairman of the previous session. Otherwise, with the permission from the organizing secretary and the organizers, I invite now the last oral presentation.
Kavanashri.
So, Kavanashri, are you there?
Yes, sir. Am I audible?
Yes, you can start sharing your screen and present.
Yes, sir. Is my screen visible?
Yes, it is visible. You can present.
Thank you. Good evening all. I'm Kavanashri and I'm here to present on topic exploration of nanotechnology in managing anthracnose disease of soybean under scheme 5. Soybean is an internationally recognized crop which is also called as golden bean, and it is high in protein and also oil, that is around 40% and 20% respectively. The major constraint in the cultivation of soybean are biotic and abiotic stresses. Among the biotic stresses, disease cause major loss. The crop is known to affect more than 100 pathogens, out of which 35 are economically important. Anthracnose disease caused by Coledotrichum truncatum is turning out to be a very serious disease, especially in northern Karnataka due to the changing climate. The initial symptom is brown spot, which will coalesce and turn to be a necrotic lesion, and also we can observe the concentric acerbuli on pod. These are the mycelia that is ash colored, and the fetae aseptate spore, and they are truncate. Usually, management of anthracnose is mainly through chemicals, which is non-eco-friendly and hazardous. In this view, exploration of nanotechnology in managing anthracnose was attempted. Nanotechnology is that idea where we can synthesize the particle all the way down to the nanometer scale, which is billions of a meter, that is 10 to the power minus 9, that is about half the width of a human DNA. So the particles which are of 1 to 100 nanometer are called as nanoparticles. Due to its small size and high surface volume ratio, the precision will be more and it can be used effectively in phytopathology. Green nanotechnology is more safer than other methods like chemical or physical synthesis, because here we use plant or microorganism extracts as reducing agent, and it also has more advantage over the other methods as it is simple, convenient, eco-friendly, cost-effective, and it requires less reaction time. And these are the four green nano formulation that were synthesized, and cytosine-based zinc nano formulation and Pseudomonas fluorescence-based zinc nano formulation. For this, we use zinc oxide as the precursor, and water-soluble cytosine at the rate of 1% and Pseudomonas fluorescence at the rate of 20% was used as grease-reducing agent. And the pomegranate aryl-based sulfur nano formulation and silver nano formulation was synthesized by using the precursor sodium thiosulfate acid and silver nitrate respectively, whereas the reducing agent used was pomegranate aryl. It was irradiated by the microwave heating, and also it was stirred continuously in a magnetic stirrer. Finally, the sonication was done. The color of the final product was cytosine-based zinc nano formulation and pomegranate aryl-based sulfur nano formulation was off-white precipitate, whereas Pseudomonas fluorescence-based zinc nano formulation was white to dull yellowish color precipitate, whereas the silver nano formulation was chocolate brown to black in color. The characterization of these nano formulation, green synthesized nano formulation, was done through particle size analyzer to know the mean diameter of the particle, and the shape was observed under scanning electron microscope, and the analysis for the elemental conformation of nano formulation was done through energy dispersive X-ray spectroscopy. The chitosan-based zinc nano formulation recorded 73.6 nanometer diameter, and the sun images showed that it is of rod shape, and EDS confirmed the presence of zinc element. Whereas the Pseudomonas fluorescence zinc-based nano formulation recorded the least mean diameter, that is 9.9 nanometer, whereas the SEM images revealed that it is of rod to irregular shape, and that the EDS confirmed the presence of zinc. And the sulfur nano formulation recorded 79.8 nanometer. The shape of the particle was spherical to irregular, and the element was also confirmed. The silver nano formulation recorded 83.5 nanometer, and the shape was spherical. The element was also confirmed through EDS. The in vitro evaluation of the green nano formulation against the Coledotrichum truncatum was done in the laboratory along with the base material that have used for the synthesis, and also commercial zinc particle through poison food technique. The result revealed that the cytosine-based zinc nanoformulation at the highest concentration, that is 1,100.
We are unable to see your screen. Just check. Unable to see your screen. You can once again stop sharing and start sharing again. There is a problem.
Sir, is it visible now?
No, you can start sharing the screen. It's not visible as yet. Yes. Go for slideshow mode.
Is it visible now, sir?
Yes, it is visible. You have to go for slideshow mode.
Sir, I have already in slide. I have already in slide.
Okay, maybe we'll wait. You can start.
Is it visible?
No, double click on the slide, it will go to slideshow mode. I feel you can continue the presentation. We'll see.
Sir, now is it visible?
No, it is not in slideshow mode as yet.
Okay, again I will put. Hope now it is. Slideshow mode. Sir, is it in slideshow mode now?
Not yet, honestly. You can explain. I feel we'll see how it is possible.
Shall I one more? Shall I the PPT?
You try once again, otherwise.
This is the last chance. Put it in slideshow mode. Professor Jagirdar?
Yes, sir, I am there. Kaunasri?
Yes, sir, I'm there. Because it had
Open your slide.
that's why it got delayed, sir. I will not access that.
What has happened?
I have shared my contact.
No, it is not visible. Nothing is visible here. You go that share, open your slide, minimize, share mode, then go to this slide, double click, automatically it will go to the slide share.
Yes, sir, I'm doing like that only.
Kavanashri, can you please check your network?
Hi, yes, sir. Yes, ma'am. It is. It has the strength, ma'am. It do not have any issue.
If not possible, It is better to complete. Only few slides are there.
Try. Otherwise, you have to complete. Don't go for slideshow mode. At least show the slides.
No, it seems a poor network problem.
No, sir. Network is good only because I have done this before also, just 1 hour before also.
Just open the slide and share because nothing is seen. No screen.
Sir, but whenever I'm clicking on the share the content, it is not getting shared, sir.
Professor, see whether it is permitted or not.
It's from here. It is not possible.
No. Whether you are permitted to share the screen slide.
No. Our presenter, we have permission.
She's permitted, sir. She was presenting.
Yes.
Sir, it is better to take the decision.
Yes, sir. Kavanasri, what you will do?
Sir, give me 2 minutes, sir. I will try.
No, 2 minutes is not possible because many people are listening.
Only 30 seconds I can give you.
Okay.
Hello? Devappa?
Sir.
Better to ask her to conclude the presentation.
Yes, sir. Better to conclude, Kavanasri.
Kavanasri, you conclude. Otherwise, it will be difficult. So many people are waiting.
If not, automatically your slide will be disabled.
Okay, sir. I will continue my presentation, sir.
Yes.
Okay.
In case of chitosan-based zinc nanoformulation, the maximum mycelium inhibition was observed at 1500 ppm, that is 49% inhibition was observed, and in Pseudomonas fluorescens-based zinc nanoformulation, it was 58.17%, and the for sulfur nanoformulation, it was 27.06, and the silver nanoformulation was 84.71, and it was found to be best one out of all. So I have the picture of the in vitro evaluation also for that, and further I have also evaluated under the glasshouse condition where I have treated the seeds of the ZS335 variety and also the DSP21 variety with the nano formulation along with the base materials and also commercial zinc nano formulation, whereas it was also followed by the spraying at 30 and 45 days after spraying. So in this case, we could not observe any disease in the plant sprayed with silver nano formulation at 500 ppm concentration and also the silver nitrate that is at 500 ppm concentration. And in case of Thyram, it was showing around 1.23 disease severity against 60%, 59% of the disease severity in the control. In case of DSB21 variety, the silver nanoformulation as well as the Vitavax power was showing 1.23% disease severity, and the control was showing around 42% disease severity. And the percent reduction over the control, like in case of that, the pomegranate aryl based silver nano formulation, 15 days after the second application also, it showed 100% reduction in the disease in case of JS335 and 96.97% reduction in case of DSB21 against the 95.74% in case of JS335 in the Vitavax power sprayed pot, and 96.97% in case of DSB21. So I have also I have the photographs of the glass house evaluation, wherein I have till now I have also told that silver nitrate is also best and effective in controlling the disease. But in case of glass house evaluation, we have found that it has showed some phytotoxicity effect, that is tip scorching, and also in case of JS335 and the veinal clearing in case of DS21. And out of all the glass formulation, the silver nanoformulation, that is pomegranate aerial based silver nanoformulation, was found to be very effective, and it was found to be very best because the percent mycelial inhibition was about 84.71%, and the mean PDI, irrespective of the DS335 or the DSB21, was 0.62, and because of the phytotoxicity of the silver nitrate, it has been ruled out. I conclude my topic saying that size does matter and nano is even better. Use of green nano formulation in phytopathology is not only eco-friendly, and also it suppresses the disease effectively, and it has also major impact on precision agriculture. So, as Schwartz says, impact of nanotechnology expected to exceed the impact that the electronic revolution has had on our life. I also want to thank my guide and department and also nanotechnology lab for helping me in conducting this research. Thank you for the organization, and sorry for the inconvenience.
Okay, thank you, Kaunasri. Now I invite Dr. Benigi sir, because I intervened in between the session because the morning it was because of the time constraint, Professor Luis Mar could not present in the morning. Now I request Dr. Benigi sir for his chairman remarks. Dr. Benigi sir.
Benagi sir, can you please unmute yourself?
Yes, sir.
So totally there were 9 presenters. The first one presenter, Gerd Stamuller. So he has presented very well about the different target sites of expressions like DMI, SBI, and the mode of actions, and isopropanol.
Benagi sir? Hello?
Most probably network disconnected. Professor Shamrao? Tamay sir is there? Co-chairman Tamay sir.
Tamaya sir.
Okay. Shamrao, please.
Yes, sir. Thank you, Dr. Devapaji, for giving me an opportunity to chair and co-chair the session on the plant microbial interactions and signaling. We had totally in the morning 2 keynote speakers, and in the evening Professor Lewis spoke on the applying understanding of plant signaling models, pathosystems, real life crop situations. Totally, we had a very good flooded action on the PAMS, MAMS, PRR, all those aspects related to the host pathogen interaction. It was a very good informative session. And also we had the oral presentations by Dr. Manjunath, who spoke on metabolomics as a reciprocal defense system in case of the pomegranate bacterial blight, and Ms. Chippy, she spoke on antioxidant enzymes playing a crucial role by using endophytes in targeting and managing the bendy yellow and mosaic virus. Sampath Kumar, he presented on the virulence pattern and the role of AVR genes in suppressing the Xanthomonas citri Pathava malvacearum, causing the cotton bacterial blight. And Somnath, the last speaker in this session, he presented a new approach on transcriptome analysis with respect to the nematodes infecting in the monocot system. Totally in and all, it was a very good session on plant microbial interactions, and we can draw very good recommendations out of this. And I request the rapporteurs to please send the proceedings so that we can send the recommendations and the proceedings of this session to the organizing secretary. And once again, I thank Dr. Devappa sir for giving me an opportunity to co-chair the session on plant microbial interactions and signaling. Thank you.
Benigi sir is there?
Please. Hello.
Please. Sir.
Can you hear?
Yes, sir.
Okay. Okay. Just I will continue my opinion. Hello, Venkatesh has presented very well, and he has told about the isoxazoline fungicide, which is very much effective for oomycetes, and some of the new mode of actions like OSBP, and some of the new SCDAC and ergosterol biosynthesis inhibitors, and also there are QR sites. Their new sites has been promoted, and another one target site is dihydro DHODH. Then cell wall biosynthesis GWT1, and his information was the benzimetazoles has moving out of the market. What his opinion, and this work should be continuously active for our future disease management. Then Rangnath has expressed the total market structure about the fungicides, and he was in the opinion that EC formulation is declining, and SC formulation is very much popular. Similarly, WP formulation is declining, whereas WTG formulation is very much increasing. And he has also told about the microbials and microbes role, and there is a lot of scope for resistance management, and also there is a lot of scope for disease forecasting. And he is in the opinion of the strobilins have got the 30% share in the market, especially for foliar spots, blights, and downy mildew, and phytophthora and powdery mildew. Then Dr. Suresh Yekpote to express about the opinion about the Combi products, which are very effective for foot spots and also the foot rot of pomegranate. Then Ravi Kumar has very excellent presentation on the management of ginger rhizome complex, and he was in the opinion that the Vitavax power, that is carboxyl plus Cyram, 100% inhibition. Of course, in our trials in case of pomegranate also it is performing very well. So integration of both the biologicals like Trichoderma and Pseudomonas along with the neem cake, so they have found to be very much better in the management of the rhizome rot of ginger. Then Mahesh, he has presented on the colloidal silver hydrogen peroxide for management of soil-borne pathogens. He was in the opinion that the Alstastan Silvex, which is a combination of silver nanoparticles and also the H2O2, it is found to be very much effective. Even at 0.5 percentage concentration, 100% inhibition is noticed, and at the rate of 50 ppm, he could be able to manage the bacteria that is caused by the Xanthomonas axanophilus pathology punica in case of pomegranate. And Louis Mar already this has expressed about by the Dr. Jagirdar. He has given the feedback about his chair. Then Somnath Pokre, he has told about assassination, and he has in the opinion that the effective genes was more diverted than the non-effective. because of the more mutation, and the monoclonal group do not contain GS53 enzymes factor. And heterosaccharide CLEs are more similar to that the rice CLE. Then last one, Kavanasri has there is a problem in showing her slides at the last moment. So she has told about the nanotechnological management of anthracnose of soybean. So silver nanoparticles is found to be very effective for managing the most of the diseases, she told. So finally, what my suggestion is, in order to compare the resistance management, there is a need of newer and newer combination of new molecules which are having the different mode of actions, and also there is a time to revive the old fungicides. Of course they are old. For example, our Dithion M45 and Dithion Z78, still they are very effective because they will completely affect the sporulation of the so many pathogens. So in order to give the recommendation to the farming community, we should not leave the old fungicides, and we should not forget the use of the new and the most very promising new molecules which are having the different mode of actions, that is a combination of the products. So this is our last conclusions. So I am very much thankful to Dr. Devappa for giving me an opportunity to share the sessions. So thank you very much. So I wish all very good luck for our all plant pathologists. Thank you very much.
Thank you, sir. Thank you for your concluding remarks. I thank all the speakers, participants, chairman, co-chairman, and the rapporteurs of this session for participating in this session and giving a wonderful response. Thank you, sir.
Rajeshwara madam.
Yes, sir.
Tomorrow session will going to start by 9 o'clock. Please announce. 9 o'clock.
So, tomorrow the session will be starting at 9 a.m. So kindly we will be sending the link tonight to all your emails. Email IDs will be sent. So please kindly log in at 9 o'clock, sir, for the tomorrow's session. Thank you, sir.
Thank you.
And also
Thank you, sir.
the
Thank you very
feedback
much.
form will be sent. Thank you, sir. Thank you.
ಇಲ್ಲಿ ಬನ್ನಿ ಪ್ರವೀಣ್. ಇಲ್ಲಿ
It's okay. I will also go for that. यदि तुम अगर लीगल लीगल हाँ बोल रहे हो आप लीगल है तो मुझे बोलो है कि हाँ है बस शरई अगर मैं फुगी में बोलूँ बस बनूँ लीगल शो بحق Asciutto che ci cata ma ne si sente?
Katie band, love to get kind of good band. I know. उन्होंने तो सभी आप ही लागू हो गया आपके लिए नहीं मुझे
इतना शोर है मतलब एक इसमें से भी ये मुझे आराम से बोलना प्यार से तो मैं क्यों नहीं इस बात का ख्याल है कि अगर अशरफ बटन पर थोड़ा सपोर्ट हो करो ना I just said there was a teaching center so you said yeah क्या ऐसा रूल बना रहा है आईपी तक कुछ तो मैंने कुछ मार्क क्लास जल्दी कर वन एसोसिएट क्योंकि ये आईसीआर का रूल बदलने वाला है कुछ और होने वाला है सही कर अगर जैसे जो हमारा पीरियड निकल गया होगा उसको आप बचा नहीं सकते हैं अब देखो बैक टू बैक का भी मतलब दिन से तो आप कैसे पॉइंट बना सकते वो देखते हैं ना कानूनी हिसाब है यार
ಲೀವ್ ಮಾಡ್ಬಿಟ್ಟಿದ ಮೇಡಮ್ ಕ್ಲೋಸ್ ಮಾಡೋದಾ
ಅರಿವು ಕಳಿಸ್ಲಾ